Patience. Cool. Did he go to the back or?
My son go that way. Oh,
All right. So first our talk is by Sam Sherrill called What is a Sponge? All right.
All right. Before we begin, a content warning for trypophobia. If you're not a fan of irregular holes, there will be some in this presentation. So, sponges. What are sponges? Sponges are a diverse group of animals that are found in waters all over the world. Bottoms, the ocean, shorelines, rivers, and lakes. There are a few places in the world where you can't find at least some type of sponge. However, because of unique elements of their biology, combined with the fact that most people don't get to interact with sponges the same way they might get to interact with a dog, bug, or even a fish, sponges have a bit of an air of mystery or even simplicity to them. I don't blame you if you didn't even know sponges were animals in the first place. Usually, when people talk about sponges, they focus on the things that sponges don't have. Sponges don't have a heart or circulatory system. They don't have a brain or nervous system, and they don't have a gut or digestive system. However, the things that sponges do have allow them to circumvent a need for any of these systems that we often assume are required. Most obviously, sponges have holes. These large holes that are visible are known as the oscula. These holes are where the sponge will spit out water it's taken in. However, in addition to these oscula, are hundreds of thousands of microscopic ostia that dot the sponge's body. These holes are so small that each one is even smaller than one of the sponge's skin cells. If we look under the surface of a sponge, we'll find that these ostia are all connected through a series of complex canals that are much more weaving than this diagram shows. Each ostia takes in water, which will then be brought to one of these chambers. These are the pumping chambers that actually do the job of pulling water into the sponge and pushing it out through the atrium and back out the sponge's osculum, or one of its many osculum. This process, known as the aquiferous system, means that sponges don't need a circulatory system. Since all this water is being brought directly into the sponge and against all of its cells, those cells can get oxygen directly from the water rather than relying on gills or lungs. Now, each of these chambers does its pumping thanks to a cell known as the choanocyte. Each choanocyte has a little swinging tail on the top, a flagellum, which pumps the water through the chamber, pulling it through the sponge. This process not only brings oxygen-rich water into the sponge, but is also how the sponge gets its food. Sponges are one of the many animals we consider a filter feeder. So to eat, they filter bits of food out of the water too small for us to see. These choanocytes are also the ones that do the job of eating, thanks to this collar around their flagellum. This collar can capture bits of food as small as bacteria, which are then brought into the cell and either digested there or passed along to other cells in the sponge. That job of moving around food is done by a different cell type known as the archaeocyte. Archaeocytes are mobile cells that climb all around the insides of a sponge, bringing food, nutrients, minerals, and even microbes along with them to wherever they're needed. If a cell is in the wrong place, an archaeocyte will also be the one to drag that cell to somewhere new. These archaeocytes are even more important than that, though, as they are also the stem cells of the sponge. They're the cells that become other types of cells. When a sponge needs more cells, these archaeocytes can become more of the choanocytes, the skin cells, the skeleton building cells, and when it's time to reproduce, they're even what become the egg cells. And since these archaeocytes can reproduce throughout the sponge's entire life, they're rarely in shortage of cells. Now, getting a bit bigger from the cells, sponges in general have a skeleton. This skeleton is usually made of silica, though can also be calcium carbonate, silica being glass. Here we have a bunch of different structural spicules. These larger spicules are about a tenth of a millimeter to a millimeter big, and the sponge is filled with these. They're what give the sponge their shape. In some sponges with glass skeletons, These spicules will fuse together to form complex lattices that make the sponge even tougher. Smaller than these structural spicules, most sponges have tiny spicules that help with all sorts of tasks. Some of these spicules help with prey capture, others make the sponge less appetizing for predators, but most of these spicules, we actually don't know what their use is. If you have any guesses, feel free to talk with me after the presentation. So, putting all this together, what does it actually look like when a sponge is living in its environment? Since sponges are so reliant on water for pumping, the sponge's body is very muscular, able to expand or contract depending on if it needs to pump water or not. They'll also react to animals around them and environmental conditions. This sponge in particular is currently hunkering down as it's during the winter, at which point there's less particulate food around and it doesn't want to waste as much energy pumping water when there's less food. So now that you know the basics of sponge biology, it's important to remember that, like any living thing, sponges don't live in a vacuum. Sponges live alongside other organisms and animals that they'll interact with, the smallest of which being their microsymbiotes. There are lots of tiny organisms that will live in sponge cells. Some, like algae, are able to photosynthesize to help give the sponge food in exchange for protection. Others are able to digest certain compounds the sponge can capture, but cannot digest itself, meaning the sponge is able to eat more foods if it has these sorts of bacteria. Other microbes in the sponge are predatory microbes that are able to eat things dangerous to the sponge, helping its immune response if it gets sick. Going to something a bit bigger than these microbes, a lot of sponges have other animals that live on or in them. Here we have some worms that are living in the canals of this sponge, along with some anemones that have taken residence in its oscula. Here we'll also have a little shrimp that has taken residence in this sponge. I also notice a lot of particulate matter collected on this sponge. For these animals that live on and in sponges, sponges are an important source of food, as the stuff the sponge cannot digest will accumulate on it, which other animals can then eat. Animals that wouldn't be able to get that food if it was floating around in the water. And, unfortunately, like most animals, sponges are food, and there are animals that love to eat them. This is actually very important for marine food chains, as sponges are a source of food that help connect different parts of food webs. For someone like this sponge-eating slug, it's an animal that doesn't have access to particulate food floating around in the water. But if a sponge is eating a whole lot of particulate food in the water, that slug can benefit from food in the water column without actually being a filter feeder, thus connecting benthic and mid-water food chains. On the other hand, there are some very large animals that love to eat sponges, like this hawksbill sea turtle. Hawksbill sea turtles are much too large to benefit from food as small as bacteria, but by eating sponges, they're able to be connected to bacterial ecosystems rather directly, rather than having to eat through, say, 5 or so layers of the food chain to be connected to this important food source. So, sponges both connect microscopic food chains to macroscopic ones and midwater food chains to benthic ones, allowing nutrients to flow through the ocean much more efficiently. And then, to go a step above, sometimes sponges are able to create whole new ecosystems. Sponges that have tough enough skeletons will leave these skeletons behind when they die. And over generations, these can accumulate into sponge reefs, a version of coral reefs instead formed by sponges and can exist in deep water environments that corals might not be able to thrive. These sponge reefs are important habitats for all sorts of animals and can take generations to form. So now you all know a bit more about what sponges are, how they live, and why they're important to our ecosystems. If anyone has any questions, feel free to ask. Otherwise, thank you. I think I saw a hand here first.
Yeah, so you said they sometimes can live in rivers and.
Yeah, there are freshwater sponges too. There are less freshwater sponges, but they definitely exist. I think I saw a hand here next.
So if an animal eats them, do they have any defensive mechanism?
They do have some. Usually things that eat sponges are specialized at eating certain types of sponges. So they can better deal with those defense mechanisms. One of the defense mechanisms are that certain spicules make a sponge less appetizing, but there are also lots of sponges that produce special compounds that make them less likely to be eaten. And then?
What are lifetimes without predators, and then how do they reproduce?
Lifetimes without predators. I don't know off the top of my head, but there are some sponges that definitely could live over 100 years. Part of the problem is that sponges are a diverse group of animals, so different ones have different lifetimes. The longest ones are going to be over 100 years. And then, what's the second question? Sorry. Reproduction. Most sponges use internal reproduction, where the male sponges will release sperm into the water column. A related fun fact, the archaeocyte doesn't directly become the sperm cell, but in fact, the choanocyte will become the sperm cell in most species of sponge, at which point the sperm will enter another sponge, and most sponges are able to produce both types of gametes.
You mentioned sponge reefs. I've never heard of that before. Are they very common?
They're less common than coral reefs, but the bigger reason you probably haven't heard of them is that sponge reefs are most often in deep water environments. But yeah, generally, generations of sponges can accumulate glass that create a whole new type of environment.
Say deep, how deep?
We
Still the light.
dark, like pitch black.
I think we have time for about one more question. Anybody got one?
What aspect of sponge biology are you working on right now?
So for my thesis project, I'm actually describing a new species of sponge. Because sponges are somewhat hard to study in certain ways, there are a lot of sponges, especially in the deep sea, that have yet to be described. That is categorized, identified like by science, where they are in the tree of life and all that. And I'm currently doing that for a carnivorous sponge, which is an annoying sponge because they do not have that aquaphorous system that most sponges find so important.
Great, well, let's give Sam another round of applause. Thank you, Sam. Please stick around if you'd like. There's another talk in 5 minutes by Haley about kelp forests in Monterey Bay. Yeah.
All right. But I know. I got everything from but that was great.
Yeah, most sponges have silica skeletons. Sponges independently of silica skeleton spice. So the demon sponges that we classify both have silica skeletons, but they don't have common ancestors. Yeah. I don't know. However, it probably has to go on in certain environments.
Certainly.
Now.
So crustaceans are this big, but huge.
Since it was live streamed, is there gonna be any kind of this stuff?
Is there gonna be what? Sorry.
Are you gonna watch it online?
Yeah. It has all of our past live streams. And it's also masters in strong defense and live history as well. Lots of people. Yeah.
Yeah, one of the interesting things that is the potential concern is that.
I love everyone watching me on live stream. You're seeing the ins and outs. Okay.
So top of my head, that's like would be, if I had to name one of the environmental concerns, that would be one of them.
Oh, yeah. I know, it's like, sure.
They all, oh, because I see some of that.
Yes, there is. Okay.
Yeah.
I said, yeah. Put in five.
They're stunning.
That's.
I told her.
Yeah. Exactly true.
I have like the own place now.
Oh.
But just one thing, like, I worked at, like, it's also, I do, but I actually don't understand. It's even a little more.
He looks serious.
That's sure that, I know the things that.
Yeah. Oh, let me just.
Let me just. That is. Okay, sure, yeah. Hello, everybody. We have our next speaker coming up. This is Haley Bregoff, and she will be talking to you about the kelp forests of Monterey Bay.
Great.
Too much to do. Okay. Give everyone a second to settle in. Right, hello everyone, I'm Haley Bregoff and I use she, her pronouns, and I'm going to be talking about the kelp forests of Monterey Bay. So here in Monterey Bay, we have two dominant local kelp species that are canopy forming, and that's going to be Macrocystis pyrifer, or giant kelp on the left. We have our bull kelp, Nereocystis luteana, on the right. So today I'm mostly going to be talking about giant kelp, and that's because here in Monterey Bay, as a perennial, it usually outcompetes some of our bull kelp, but we still do have stands. So mainly on either side of the peninsula, you're going to see stands of giant kelp, unless you are on the Carmel side, where we do have a few stands of bull kelp. But you are right here. So right now we are at Moss Landing Marine Labs, and yeah. So what are kelp forests? So before I describe this large community, I'm just going to break down kelp as itself as a species. So here we have subtitle angle of kelp. So we have the stipes, which are kind of synonymous to a stem on a plant, but giant kelp is not a plant. It's actually an algal species. So they have their own kingdom and phyla. And we actually have holdfast, which are synonymous to roots. So there's lots of invertebrates that will live in the holdfast. And this is mainly how giant kelp is able to withstand high wave energy and our winter storm events. So that's how they're able to have populations all year round. And then here we have our blades. So this is where the photosynthetic reactions are happening. So they absorb light, which allows them to grow. And as you can see, there are these long towers of our stipes. So these nematocysts acts as an air-filled chamber to keep them up from collapsing and actually create this long canopy structure. So this is kind of what I was describing. You have these long plants, long stipes. Giant kelp is actually one of the largest growing kelp species in the world and can grow about 2 feet per day. So these individual plants will develop a population, and they either grow on the sandy bottom, as you guys can see, but mainly we see giant kelp stands on rocky reefs where there is a lot of rugosity for lots of organisms, so complex hiding spaces for invertebrates, juvenile fish, and a myriad of other organisms. So I kind of briefly touched on that there is kelp forest population. So as you can see, all those stipes coming together. So I'm really briefly going to be going over kelp forest ecology versus phycology. Most of the talk's going to be talking about kelp forest ecology, but I really can't get into the nitty-gritty without talking about phycology, of course. So phycology is the study of algae, a very diverse group. There are three main phyla. We have our green algae, our red algae, and our brown. And I'm only going to be talking about brown because kelp is in the Laminariales group, family, pardon. So essentially, when you think of phycology, I want you to think of beautiful architecture, structure. You're really thinking about, well, how many buildings, how large are they? And really, what is that building there for? And what's the purpose of it? But I take a very different approach to it as a community ecologist, and I take more of a landlord doorman mindset. So who's really living in that building? How many people are there? And what are those individuals doing and contributing to their community? So I've kind of talked about the main differences between kelp forest ecology and phycology, but why do we even care about giant kelp forests, right? Well, there's a lot of benefits to giant kelp. So not only is it a foundational species, an ecosystem engineer that allows a habitat to form for a diverse group of organisms from marine mammals to fish to invertebrates, but it also removes carbon from our systems. And we actually can see that it buffers wave energy. And also kelp, because it has been because it grows so fast, we can actually harvest it and has a plethora of uses. Like actually the ice cream you guys eat, a lot of that has kelp derivatives, and you can actually try seaweed ice cream later today. So just something to look out for. But mainly what has allowed giant kelp forests to be so important is the biodiversity. And if you guys aren't aware, Monterey Bay is actually one of the most biodiverse marine habitats in the world, and we wouldn't have that biodiversity without our giant kelp. So I have this slide, which is most of our sessile-benthic invertebrates, sponges, anemones, snails, abalone. We do have some pelagic jellyfish, and mainly our grazers. So in that bottom right-hand corner, you can see our purple urchins surrounded by some brittle stars. And all of these organisms have really, really important roles that allow the system to be so productive. Additionally, we have more of our charismatic organisms like rockfish, sharks, and of course the lovable marine mammals of Monterey Bay, harbor seals, sea lions, and who can forget the sea otters? So essentially, I just wanted to discuss how important the biodiversity of our Monterey Bay. And for my master's thesis, I actually did kelp forest monitoring with scuba-based visual surveys. And what I did, it was actually take transects out, which if you can see in the photo on the left, is that tape. And we would actually do 30-meter transects and try to quantify all of the organisms with species identification and counts to really get an idea of how the community takes shape and structure over a year-long period. As you guys can see, that water is really green, and that's how it looks like normally in the bay. Sometimes you get photos like this and you're like, wow, oh my God, that's beautiful. But the reality is it's looking a lot like this. So how do you tell what a fish is when you can see about 3 feet in front of you? And that's when environmental DNA comes in. So I know you guys came in for a kelp forest talk, but I tricked you. It's a molecular talk. Nobody comes into the molecular talk, so sorry. But what is environmental DNA? So environmental DNA is a short fragment of DNA that's shed from organisms and is free floating around the water column. If you were to jump into a swimming pool and I was 100 miles away, and then I got some of that swimming pool water and I did a lot of cool DNA molecular work, I would be able to tell you how many people were in that water and try to identify that person's genetic information. And so that's the beautiful utility of eDNA. And so we have these genetic fingerprints based on the reference genomes that we're able to create these tools called primers, which are short DNA fragments that allow us to assign to those references. So how does eDNA detect multiple species? I kind of alluded to it, but each of those colors will respond or coordinate to a different primer. And I have some of the main universal primers listed on the board. So as you guys can see, the blue would detect fish, and the red would detect more of our invertebrates and algal species. The yellow would be more of those planktonic, tiny little microscopic organisms, and the 16S would be just bacteria. So when it's dark and green and I can't see a lot of things and I have limited visibility, I use those water bags to collect water and filter them, and I put them in a preservative so I can do DNA extractions and I can compare to those sheets of paper that I took notes on and say, well, what did I really miss in that low visibility? Was I seeing more rockfish? Maybe there was more, maybe I missed a ton of barnacles and it was just so low visibility, or maybe there was growth over them. So I'm able to use not only visual tools, but also molecular tools to give us the overall insight of how our community is changing, as most of our data comes from the summer months. When I go back, summer, 3/4 of the year. This is how it's looking when you're going diving. So like I said, have been doing high frequency, long-term kelp forest monitoring at Hopkins Marine Station at Stanford's satellite campus right next to the Monterey Bay Aquarium, if you guys aren't familiar. And so if you guys have any questions about my research, I would love to connect, because as I said, I'm an eDNA nerd and nobody ever wants to talk to me about molecular stuff, because they're like, but show me the kelp photos. And I'm like, but what about the cool little stuff? So if you are interested in the nerdy talks, I tried to keep it light because I was like, ooh, don't want to trick people, catfish them too much. But if you want to learn about the molecular stuff, I'm happy to talk your ear off and go into more depth about it. And those are my photo credits. Sorry to give really brief insight or a really brief talk, but if you guys have any questions, I'm happy to talk about it. Yeah. Yeah, so if you guys have any questions, yeah.
Based on all the things you've studied, how optimistic would you be that you're going to see the same level of biodiversity in Monterey Bay in 20 years' time, 50 years' time?
Yeah, that's actually a great question.
The question, please.
Yes, so the question was, based on the trends I'm seeing over this year from my data, what is the potential of 20 to 50 years, the long-term standing populations of giant kelp in Monterey Bay? So just as a little bit of brief background, in 2013, there was an anomalous heat wave called the blob, which really led to a decline in kelp. Giant kelp love cold, nutrient-rich waters. And so when you had this increase of high temperature, less nutrient water, we saw diebacks around 50% of giant kelp stands around the peninsula. So there are cyclical populations where we see large stands of healthy kelp. We see a big El Nino event, which can have these warming waters where they die back. But as a foundational species, there is a set level where a population can rebound. So a lot of work is being done to control grazers like urchins and try to protect our bigger predators, so we never fall beneath that standing population. So when the summer comes and we have big recruitment events, it can rebound. So I say, as long as we're monitoring and not letting these populations fully crash, there's always going to be enough of a standing population, and as long as there's enough genetic diversity to keep the population growing, 20 years and 50 years, as long as we're monitoring it, I have lots of faith, especially with the huge efforts going on at Tinker's Reef to kind of identify the interactions between urchins and other grazers, and outplanting of other kelp like bull kelp in Northern California, which saw giant crashes, kelp completely wiped out, and this year they're saying over a 900% comeback. So there's always going to be positivity and hope, as long as we do our roles as stewards and try to understand. Yeah, great question. Yeah.
Once you take your sample, we're doing that, how long does it take to do the analysis?
Yeah, so I was asked, after I take my sample, so after I take this bag of water, how long does it take to do my analysis? It can take anywhere from a week to months at a time. So once I get it out of the water, I put it on ice and I'm running to the lab. I'm like, oh my God, the DNA is degrading as we speak. And then I go into a lab and then I put it on a filter. So I put it on a PVDF 0.22 micron filter. So it captures all the little fragments of DNA, and I put that in a fixative. So it can sit in a -80 freezer for years. Or I can go and filter it right away and do my DNA extractions and PCR based on the number of samples I have. It could be 2 days, I finish all the extractions and I do the PCR and it's sent out for sequencing. Or it could take me, if I have hundreds of samples, it could take about a week or longer. So it's just based on the amount of samples I have and the amount of primers. 12 to 20 primers, that's a lot of rounds of PCR. If I'm looking at one very specific organism, like a taxon specific primer, I could have data back in 2 weeks. Yeah, great question. Yeah, I saw him back here and then.
Does kelp farming, is that a 30% thing?
Yeah, kelp farming. So where I go is actually a protected area. So along the Monterey Bay National Marine Sanctuary, we have our conservation areas and our state reserves. So kelp farming doesn't impact me and my research, but there is still a standing kelp port, I believe, in San Diego. So I'm not really sure, but I'm assuming that it would stir up a lot of DNA, right? Because they're actively pulling kelp stipes, they're pulling out plants. So that would send a lot more fragmentation of DNA into the water column. So I would assume, like, we would see even more DNA. Yeah. Then I saw a question
Yeah.
up here. Yeah.
Can you get a sense of, a reliable sense of the population densities? I know you can get the diversity from.
Yeah.
But you can still get it with just a.
Yeah. So for eDNA analysis, so you would need qPCR of that very specific kelp primer. So what I'm doing is more of a presence absence, where I'm just detecting if I'm seeing an organism. If I really wanted to know the quantity of that, I would then do another round where I use a very specific qPCR primer that would tell me the number of loci present. Yeah.
Would you like 2 more questions?
Yeah, 2 more questions. Yeah.
Sunflower star, how that's affecting the kelp forest, and like, I know the sunflower star died.
Yeah.
How the research around that.
Great question. Talking about sunflower star and my research and just kelp forest comeback. Well, here in Monterey Bay, they are still locally extinct. Unfortunately, we have not seen a sunflower sea star in the kelp stands, but historically they used to be abundant. So, people are leading to think that maybe the urchins were able to expand at the population rate they were because we didn't have an abundance of predators keeping that grazing pressure down. So we are definitely seeing, there's a great paper by Josh Smith that talks about, Dr. Josh Smith at UCSC, that talks about otters kind of filling in that role, otters being able to keep the grazer populations down. But really what we need is more predators on grazers to keep that balance between, urchins and sea cucumbers from grazing down healthy kelp stands. But yeah, great question. Definitely, there's a lot of work trying to get them back in Northern California where they were historically more dominant. Yeah. Yeah, one more.
The farming of the kelp.
Yeah.
I thought you said they're pulling out the roots, but then how does it propagate? How does it?
Well, this is like, there's controlled farms in San Diego, so I'm not sure, but I have seen they will have these boats that will have large, like, propellers, kind of similar to a tractor, where they can pull out the kelp. But that's because they own those stands and they're like farming it very specifically. We don't have any kelp farms up here.
Right, but how does it grow back?
Yeah, well,
How does the kelp expand, I guess?
I don't wanna leave, I don't actually work at a San Diego kelp farm, so I would assume they're either seeding or they're only pulling out a certain percentage of their population to allow the natural recruitment.
So then how do they expand here?
We don't have any, just natural recruitment. Like,
In life.
oh, sorry. They actually have an alternate generation, so they will release spores, and then those spores will propagate, and then they will turn into juvenile kelp once they've been able to settle. And that's like a sporophyte to gametophyte transition. Sorry, pardon. Yeah, I'm more focused on the community, but we do need to always consider the phycology aspect of how they reproduce and how all those physical parameters affect them.
Thank you.
Great questions, yeah.
All right, guys,
Sorry.
you guys, if you have more questions, feel free to find Haley in the hallway and let's give her another round of applause. Please stick around if you have more time. The next talk will be about blue water diving in about 3 minutes.
Hi, Aiden. How's it going?
Are you running the box right now?
Am I what?
Are you running the box?
Yeah.
Okay.
Yeah. Okay, so if I put this, beautiful. Okay. Okay, so I'm doing these four, right?
Yeah.
Okay, I'm doing these three. And then I'm doing these two.
Yeah.
You're doing these two.
Yeah.
And then I figured with these ones, we can just kind of like talk about what they are and then.
I just have like one bag.
Hey, what are you doing here?
Need to see you.
Yeah, sorry, I was literally like up here talking to you already. Yay, and then I figured when you get to this one, since it's Rise of the Sun. that's
Yeah,
when you
yeah.
can talk about-
I'll just talk about the trail. Yeah.
And then try to keep it to like this two. But, okay. Also, I feel like we should change the light.
Sorry.
Yes. So what is actually there? Yes, but
My mom's.
yeah, shopping my mom. No, we're speaking. I don't think I have. Oh. But I just.
I'm not sure.
Yeah, I'll just say, yeah, she's in there right now, yeah. I walked in there, we both went, thank you. You know, there's like. Walking down the hall. I know, and the part on the wall that has the history and pictures and all that. They're quite history. Yeah. Yeah. And all that equipment, see all the equipment.
Hello, everybody. Our next presentation will be about blue water diving, and it will be presented to you by Jess Franks and Aiden Young. Let's give them a round of applause.
Hi all, welcome. Thanks for being here today. I'm Jess. This is Aiden. We're both divers in the Moss Landing Scientific Diving Program. I'm the Assistant Dive Safety Officer and Aiden is a student here at Moss Landing. And we're going to talk to you about blue water diving today. Raise your hand if you have heard of blue water diving before. Okay, so some of you. Okay, well, I think everyone's gonna walk away here learning a little bit more. So today, just to give you an overview, we're gonna talk about blue water diving, the what, the why, the how. We'll go over some existing methods for answering some open ocean research questions, as well as the history, some of the history of blue water diving and at Moss Landing. We'll show you some organisms you might be able to see underwater and how you might sample them. And then we'll end off by talking about the reason that we've been conducting so many blue water diving operations recently, this NSF funded grant. So to begin, what is blue water diving? Blue water diving is a branch of scientific diving that takes place in the middle of the ocean, away from coastlines, reefs, and artificial structures. So it's in the open ocean. It's not open water diving refers more to just any sort of entry-level certification, whereas blue water diving is that open ocean, no reference diving. Now, you might think it implies deep diving, but it doesn't. What it implies is that we are over very deep water. So if we were to go offshore right here in Moss Landing, we would be over hundreds of meters in the Monterey Bay Submarine Canyon. However, the diving that we're doing is actually pretty shallow. So we're only at about 30 to 60 feet of depth. It's just that we're over a canyon, right? We're over something really deep. Now, the key challenge with blue water diving is that there is no frame of reference. So when you're diving in the kelp forest, there's a kelp right in front of you that is going to tell you which way's up, which way's down. When you're diving in the open ocean, everything is blue. So there's no frame of reference, and that can be quite disorienting. Could lead to vertigo, and it can lead to unsafe ascents or descents if we don't properly train. Here I have some pictures of what that has looked like. These were taken just a couple miles offshore here in Monterey Bay. Sometimes it's blue water diving and sometimes it's green water diving. Very Monterey Bay. And we'll go over in a little bit what this contraption is with these down lines. But this is sort of what it looks like. And I remember the first time I did an offshore blue water dive last semester. As soon as I came up, I told the boat crew, I think that's the closest I have ever felt or ever will feel to being an astronaut. It's truly special. There's nothing like it. It is in some ways really frightening. And you also get to focus on all of these little microorganisms right in front of your face that you would never otherwise see. And why do we do it?
Why do we do it indeed? We are interested in that upper layer of the ocean in the pelagic, where there's a lot of flux of nutrients and materials from the surface and you focus onto the deep sea. And so we're interested in the magnitudes and kind of how the transport operates. So we need in situ experiments and collections of these fragile, members of the community that are responsible for processing nutrients and, marine snow, which is the detritus, which is very important for nutrient transport to the deep oceans. And so some existing methods of collections other than blue water diving, we have your traditional CTD, which takes measurements of certain variables in the water column, and we have net tows, but net tows, as we talked about, can be harmful to these gelatinous organisms that are very fragile. And so blue water diving is a way to observe them in situ and to collect them without causing harm, so we can manipulate them in experiments and figure out answers to scientific questions that we have. So exactly, sometimes we don't have, the tools we have are not adequate, so you just got to go yourself. And it's super fun and exciting.
Cool. So Moss Landing is a really cool place to do blue water diving because we have a rich history of it. So there's some key pioneers in the use of blue water diving, one of them being Kenneth Cole, who was our director. He created the equipment that we use for blue water diving, and we still use it to this day. Some other pioneers are Alice Aldridge and Mary Silver that are pictured there in the top photo, as well as John Heine, who is our diving safety officer, and Steve Haddock, who works at MBARI. Those two gentlemen wrote the scientific blue water diving manual, which is basically the only, piece of work that we have to go off for how do we conduct these operations. Alice Aldridge was a professor at UC Santa Barbara. She was interested in how do I quantify sort of these marine snow or particles floating down while I'm underwater? And she was actually able to use this quadrat that you see in the picture on the left to quantify sinking speed of different particles in situ, which is so cool. And the two of them, Alice Aldridge and Mary Silver, won the Bigelow Medal in 1992 for their research. And the quote here says, "In recognition of their creative contributions to biological and chemical oceanography, particularly in demonstrating the importance of marine snow as a major contributor to the vertical flux of particulate matter throughout the world's oceans." So that marine snow is one of the biggest things that we can study. And if you've not heard of marine snow before, it's often, detritus or fecal matter, things that just fall throughout the water column that look like snow, that, help that flux continue. And it's just also really awesome to see pioneers of diving be women. So that's a win. Okay, so what does it look like? So this is from that manual that I shared with you. This is what a typical dive might look like. So on the boat, we have a small boat, and then likely you have a mothership that we sort of have, come off of. On that dive boat is a line that connects out to a sea anchor. We used one yesterday. They're a little bit complicated, but it's basically, if it's really windy, the sea anchor helps the vessel stay in line with the wind, and it helps maintain your position. So then for the dive boat, it connects to a surface float that connects to what we call the down line. That down line has a wave damper because if there's a lot of waves on the surface, that rig is just gonna be bouncing up and down, which it did yesterday and made me really nauseous. So then on that down line, you get all the way down to where the divers are. Now the divers are hooked in to this trapeze, which we actually have one out in the courtyard that you should go see at the dive booth. The trapeze has tethers that each diver is connected to. So blue water diving is very different than normal diving, right? Normal diving, you don't want to be attached to anything because you don't want to get entangled with anything. But in this instance, we need to all be together and act as one big buddy group, so that we don't, drift off and separate into this vast open ocean. So everyone's connected into these into their tethers with a quick release, because in case something happened, you need to be able to get out really quick. And then this diver right here has a special role that Aiden will share with you next.
Yeah, so one of the more important roles, I suppose, would be the safety diver, who kind of serves as a buddy for all the working divers, checking in with them. They kind of keep track of all the, the safety variables like time and depth. They make sure no entanglements happen, and they can resolve it by detaching and reattaching tethers, solving problems. They keep an eye out just on the surroundings for predators or signals from the ship or other ships in the area. And yeah, they're kind of just the safety, as the name implies, they keep track of everyone and the safety of everyone involved. And so the working diver is their jobs are to handle collections, do the science, but also to keep contact with that safety diver, as they're the kind of the core of the whole operation. And then obviously they have to be juggling complex, like all these tasks, like handling collection devices, which can sometimes be ungainly to use underwater, while also maintaining buoyancy, and be efficient and make good use of the time underwater. And then here's some cool photos we have of that were collected using blue water diving techniques. We have. Yeah, so you should check out in the hallway on the way down. We have tons of these kind of pelagic art photos. And they're really, really pretty and show a lot of just the diversity of life that can be found in this environment. Some examples of organisms we can find are salps, which are organisms that form chains of like 100+ identical copies. There's pyrosomes, or this is a siphonophore, which can actually contend for the thought of longest animal on Earth, as they can form colonies of specialized individuals, and they can be up to 40 meters long, these chains, which is really, really cool. Another example is like a ctenophore. They've got these rows of cilia that refract light in an interesting way, so that they kind of appear rainbow to the eye, rainbow color. And tons of other jelly species and like juvenile fish, juvenile like mollusks and things like that, which are super pretty to look at. And yeah, like Jess said, it is the closest I'll ever come to being an astronaut. It does feel like you're in space, especially when it's like super snotty in Monterey Bay and it's very green. Kind of just looks like you're swimming through stars, which is very cool. Yeah, here's some more cool photos. This is kind of the reason why this blue water project was started up again at Moss, is for these diatom mats, these Rhizochelenia diatom mats. And Jess and I are part of an NSF funded project where we're going to the oligotrophic ocean off Hawaii to kind of look for these mats, because there's an interesting story that hasn't been resolved where basically they've been known to sink to deeper depths to refill on nitrate and nutrients, because the oligotrophic oceans are poor in macronutrients like nitrate. And so they sink to these depths, refill on nitrate, and then go back to the surface to do photosynthesis and grow and stuff. And so they also have been found to have a unique nitrate fixer that is not typical for other species of phytoplankton. So we kind of just want to do some incubations in situ to figure out what's more important. Is it the sinking to depth to get nitrate? Is it the fixer? And kind of resolve the big picture there. So we're both part of that project. I will be heading in the summer, which is super cool and exciting. Yeah, and then here's a picture of marine snow. Not much to look at, but super, super important.
Much to look at.
It is very cool. Yeah, but when you think about just how sparse nutrients are in the deep ocean. Like this is very, very important for life. Cool. Yeah, and then here's a photo, more photos of the diatom mats. Yeah, thank you.
Thank you. We'll be happy to answer any questions y'all might have. Yeah, with the fish shirt.
How do you maintain buoyancy in the water? Does that make you think?
Yeah, can I take that? Okay, the question was, how do you maintain your buoyancy in the water column? So the biggest thing is just looking at that dive computer the whole time. I have mine on my wrist here. And you can also keep the downline in your sight. So I always try to keep the downline somewhere in my line of sight because they do have little tabs on them with the depth. So that's a good frame of reference as well. But truthfully, you know, building up your like basic dive skills before you take on this blue water task loading is definitely like a big part of it, right? You know, dialing in your buoyancy, dialing in your gear before you go out into the open ocean is huge because it is so different. Like the senses are so different that it takes a lot of centering yourself to really keep that neutral buoyancy. Yeah. Yeah.
This kind of action if somebody gets lost or an emergency happens and.
Yeah. Yeah, can I answer this?
Yeah, sure.
Okay. So if someone is lost, typically what we assume happens is that the safety diver will notice that their tether will start to fall down because the safety diver should always be watching the divers in front of them. In which case they can either make an audible sound for a diver nearby, or they can pull on a tether of another diver to get their attention and point and say like, go find them. And we don't want people to unattach. So it sort of depends on like how far that lost person would be. But that's the general protocol. And then if you are not able to find them, you see that someone's gone, we would abort the dive, come up to the surface, wait for 5 minutes, and then enact EMS. Because if they were lost, they would hopefully realize and then also ascend and then find us. Other emergencies operate very similarly to any other dive rescue. The only added component being that they're tethered in, and so just managing the entanglement as you're conducting that rescue. But yeah, it's a good question. Yes.
I just manage drift with the rig in relationship to the mothership.
Yeah. Do you wanna talk about that?
Sure, yeah, well, the rig is attached to the boat, so you're kind of drifting all together. And then hopefully you just have a competent captain who knows how the winds are flowing, right? And so that they'll put the rig like off to the bow where the wind is blowing in the opposite direction so that you don't cross lines and.
Additionally, the sea anchor should help that a little bit. But we have had a dive where we dropped the rig down and you could see it just like this way with the current, and we're like, no, that's not worth it. So you don't want to be swimming your whole dive into a current, right? So assessing conditions continuously throughout the day.
Last question.
One last question. Okay, I don't know who raised their hands first. Raise them again, guys. Okay. Blue long sleeve.
How frequently would you encounter a large marine mammal or a fish or shark or whatever?
Yeah, how frequently would you encounter a large mammal? Well, for us here, bless you, we likely wouldn't know they're there because the visibility is so poor. So that's good in a way. But where we're going in Kona, there have been oceanic whitetip sightings. And we have shark billies. There's one in the courtyard as well, if that, you know, is the case. But typically they don't care about us that much. So like if you see one, we have like a protocol where we'll all come to the center, you know, have our arms out and be looking. And if they become more aggressive, or if a second one arrives, then we will call the dive. Or if for any reason someone is like, I can't do this anymore, we'll call the dive. But yeah, I mean, some whales breached right after we got out the other day, but we wouldn't have known they were there. So okay, thank you guys.
Thank you.
Thank you very much, everybody, and we have a puppet show coming up next, so sit tight for that puppet show.
It's not a toss. Okay.
Get somebody lost and left behind.
They might come back. Yeah, that's true. Okay, well, I guess I'm done. It's fine, so that. I need to ask really quick.
I think so, I mean, yeah.
Everybody's here for the puppet show. Our puppet stage is behind all of us, so we're gonna come around and start turning those chairs. But you are also invited to turn the chairs if you would like. Another lost item, left item. Okay, so that's really fun. Like, start like 11:15. Yeah, that was super fun. Are we able to have a document I'm just gonna step out for a minute.
Yeah.
Yeah, that was from my ear. So like, I wanna sing.
Wow.
Yeah. I was supposed to, I was supposed to like this. I didn't. Was there?
don't think it'll take us.
Yeah, the idea. I was starting right into sticky note, but they felt it's too big, so.
Yes. So before we begin, I want to thank a few people. I want to thank Dr. Amanda Khan for her encouragement and continuing this excellent tradition. I want to thank the Child for constructing some really beautiful puppets. And lastly, I want to thank James, who helped us set up this stage back here. And without further ado, Moss Landing Puppet Show.
Hi everyone, I'm a scuba diver. Wow, it's so beautiful outside. What a beautiful day to explore the Monterey Bay. Are you ready to go? Yeah. Awesome, let's start in the intertidal zone. Let's go do some tide pulling before we go diving. Hey, watch what you said. Who is that?
Anemone.
Can you say anemone? What is an anemone?
I am an invertebrate, which means I don't have a spine. I can be found in coastal habitat. It's quite cool.
This is such a cool place to live. What other animals live in the tide pools?
Tide pools are home to many amazing animals. We have urchins, mussels, barnacles, octopus, and sea stars. Here's one of them now. This is my good friend Arthur. Lydia.
Hey guys, are you enjoying your visit to the tide pools?
Yes, we are, but I almost stepped on an anemone.
Sometimes critters in the tide pools can be hard to see. They can blend in with the surroundings using camouflage. That is why it is so important to watch where you step.
I promise to be more careful about where I walk. I love the tide pools and I want to keep them safe.
That's what I like to hear. I would love to find some tasty mussels for lunch, but I hope you enjoy the visit.
We are so lucky to be able to see a small but intimate life in the tide pool. We have to be careful to protect that. The best thing to do are to watch where you step and leave animals where they are. It takes effort from all of us to keep this habitat the way it is.
Don't worry, I won't forget and I will definitely be back for another visit. I can't wait ready to go diving in the beautiful kelp forest. Oh, I love the kelp forest. Look, there's an abalone. Wow, look at all these fish. Oh my goodness, a sunflower star. It's so nice how everyone works together to create this beautiful environment.
Is it just me, or is it getting a little warm? Actually, it's making me pretty hungry. So hungry I could eat an entire kelp forest.
Too hot. It's making me feel a little sick.
Over water, here we go again. My, how could I survive you? Over water, urge is coming in. Must find another place to move to. Yeah, warm and hungry. Since the tap is rising, my mind, we can never stay at home. Warmer water, here we go again. My, how could I survive you? Wait a second, am I in the right place? Where's all the kelp? Where did all the fish go?
Yeah, so that might have been my bad. I kinda ate all the kelp.
Oh no, why didn't anyone stop you?
The only thing I'm scared of are sunflower sea stars, and they couldn't handle the heat. With the sunflower stars gone, us urchins aren't afraid and we come out from our hiding.
Wow, I didn't know that a single species could make a big difference in making sure this kelp forest stays beautiful. Even the little guys like sunflower stars mattered in maintaining a healthy ecosystem.
Yeah, even us small guys matter. Big, small, squishy, crunchy. Oh no, I see a sunflower star coming. Run away, run away!
Wow, there's no place like home.
Okay, guys, now it's time to go a little deeper. Odysseus, I need to find my way to something called the Mesopelagic Zone, and I've heard you're quite the diver. Can you help me out?
What I'm playing. Where all my favorite snacks are. Let me take a big breath. Let's go.
Wow, Grandma, you're pretty just wait for me.
Then, all right, some people call the Mesopelagic Zone the Twilight Zone. And that's because it gets so dark. I can use my echolocation to see where we're at. But you might need the flashlight.
Got it. Wow, Grandma Chrissy, this is really cool down here.
Sure is. But I got a thick layer of blubber to keep them nice and warm. Other species swim to shallower areas during the night to stay warm. One sec. No. It's my favorite snack. No, look at squid.
Oh, where did those guys come from?
Like I was saying, some species swim to the surface at night. It's called vertical migration. Can you say vertical migration? Vertical migration.
Okay. So we gotta swim down with all we've got, because it really matters if we make it or not. See, the dolphins depend on us for food. So what do we do? Wow. Nation, wow. From migration, is that the same? From the dolphins up there. Wow. Woo! No food is getting past you. Chop, 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 chop. Grandma Chrissy, I'm not sure I can go down much further.
Humans aren't meant to dive this deep. You can catch a lift on my submarine.
Whoa, nice.
I've got some squid on my name.
Sweet deep ocean, here I come. And I finally made it to the very bottom of the ocean. So how does anyone see down here?
I could answer that.
Whoa, your eyes are, sorry.
Big, enormous, gargantuan. Yeah, you wish you got orbs like these. Because my eyes are so big, I can see things in the dark that your puny eyeballs can't.
Cool. Do your eyes help you see at the surface too?
The surface? Why'd anyone want to go up there? It's way too bright. I got all the light I ever need right here.
Lights? Down here?
Want to see something cool? My favorite place is the deep sea. There sure are some fancy lights down here. Look, here's some of my friends now.
Whoa, what makes you all look like this?
It's a process called bioluminescence. Can you say bioluminescence?
That's right. Some fish use their lights to talk to their friends. And some use them to trick their dinners, like me.
We like to use our light for socializing. Just don't ever die in the deep sea, my friend. I use my lights to partay.
Wow, you guys sure do know how to party. You even have confetti.
That's not confetti. It's called marine snow. Tiny bits of dead plankton, poop, and leftovers from the surface.
They're for us. Delicious. Would you like some?
Would you look at the time? I think I'll have to skip the plankton poop for today.
You're not.
From the surface. So, what did you think?
I learned so much about our home in Monterey Bay. I learned that every single species in the ocean is important, and I want to make sure I do my part to protect it. Thanks for the ride.
That was beautiful. Wow. Okay. That was so big.
There's a sticker.
It's open behind the room. What is open behind the room? I downloaded it as a PowerPoint, so things would be different. I thought I looked. I mean, it's fine. You guys are great. All right.
I don't know. Actually, I'm not. Well, I'm in here.
Right, and then class is open today as well. Yeah. Cool. I haven't gone back. Oh, no. It's very stressful. And then. Like, that's your comment, and it's like, you can get my talking. Yeah. Like, once I pull it, it's hard, I go back to that. It's really short, my friends, it's like, it's hard, it's not by Monday, I'll see you on Monday or something. I was in, I was like, yeah. You like,
Next year, there will be.
He says something about what.
So here I can come here. There's no reason that was there.
Yeah. Yeah. Now, it happens and. Yeah, but, like last. And so we decided. Yeah. I'm glad, thanks for coming. Do you like?
No, I live in.
Oh.
Oh, it's good, because I want to do it. I think
Yeah. I think you have to be afraid of that person. Yeah, but I'm okay. Yeah, good luck.
We're gonna go.
Oh, I know, yeah, my desk looks right out there, yeah. Our study, this has dolphins. Yeah. Here, when I was a student, like, yes, there. And just, and yeah, those are successful. Stingrays, that was that. Not stingrays. So they're, you know, you have to, they're, but it's not like a blind, big price. But no, and they're also really thin, so they're huge dolphins, and they're kind of like a dark gray color, and they're really scratched up. They're so cute, they like, so if you'll see like, dolphin, and they'll be like, small, yeah. So, like, a lot of research. They have, yeah, so I just that's probably, yeah, so I'm doing a study on figuring out how to diet.
So you doesn't have your.
I think someone knows what actually it is. I actually was saying and then versus skins and they said, wow, not studying, you know, I was like, yes. You know. Like, you're so we're gonna remember. Yeah. Okay. Sunflower stuff. Yeah.
Second they saw me, you know, they're.
Yeah. I mean,
Coming up for.
oh, wow. We're here, great, and then it's. It was so much underwhelming.
Yeah.
Coming for so many years.
I've written flyers that you guys gave them, yeah, and Actually, I thought people, you gotta go.
But. So, you know, just know that it's, so it's not a thing, so it's, yeah, it's, but no, overall, it is. So. If I don't trust. Yeah. Going to the puppy show.
Oh, you're next, like that.
Yeah.
A lot of people were saying that it was on. What's it called? Said it was actually more elaborate than I thought. I saw it.
It was, I didn't know, soft, but.
Mother really had different. No, oh, the zoo was just.
I'm just giving something for.
Yeah, of course.
I've been told I have to be looking at.
It's absolutely so.
Yeah, I look at this every time I'm in the room.
And
Oh, yeah.
I always notice something new every time I want to speak. Like, I wonder if we can get to it. Yeah, every time I look at it, I notice like a new detail that I didn't see the first time. It's stunning. Yeah, that's insane. Really, really good. Admiring that. How much detail.
What's that?
Looks like they turned it around for a sound. This one, they just. Oh. Yeah, so when did you?
Yeah.
They probably have.
Right?
Yeah.
But I get funny. Well, it's the only sound. Oh, it's great. Seared.
This is a public. It'll show where.
Oh, you're right. It's a
We were telling her.
We got a workshop. I should have downloaded this earlier.
And studied it.
This was a.
Yeah, okay, so it's. I don't, there's a one second. Are you going to talk?
You have nothing else to do.
I got lots to say.
Yeah.
How many people wanna listen? Yeah, they'll listen, but then I hear.
Speak yourself, like, I see all the names.
Yeah.
Oh, you don't have to.
It's fun having her home.
This is the big love match, dude.
You see all that.
What's the other man?
They're all.
No, someone's done.
Oh, my lord.
Yeah, mentally. We take a picture. We'll take a picture. I just got like something.
See you outside. Yeah.
There we go. And. Like, that. Nothing wrong in the waters. Right. Okay. No, stop. That's what I played. Show here. Yeah, maybe I'll come out later. So go bring her around.
Not here, you go.
I don't think they get that. Yes. Yeah, even
Oh, please.
Yes. It's a 50th anniversary.
No. It's all part of that, it's just. No, but the rest.
This is a whole, yeah. We can't get something like this, still.
No reason for it.
Want to go over there, or you want to go back to the guy?
Right, so.
Yeah, that's cool. The current one is our business, that's why, since I didn't select. It's not just your share, and this is, yeah. Yeah. I care what I here, I so. I was just gonna say it's true. Yeah, I figured that.
Oh, yeah.
Go, go get wrong.
Yes.
Basically, part of the hour is, it's getting really on the thing for everything. And like I would. Okay, no.
Yeah, this is our friend Kimberly that wants to do. Let's see, of course. Where tomorrow? Okay, I don't know.
So we did miss, okay. Yeah, one of the things I went to when I was at PSF last we had to talk about recovery rules of self, they also have. That she is an engineer for. Markets. And I essentially take urchin tests. And regenerate that into. Like ceramic house. The test and comments, like, so like, what is the urchin is actually done with it? Whatever extra scientific stuff that they do, they permission for that to try into. Early. And there's another one, Kim sells stuff from Sandbar Fisheries, and she. Similarly, I guess because it's in that.
Oh, it's great.
Yeah, she knows, but they have a which is a lot of them too.
What are you doing in here? Just for the
Big topic was something else, sustainably. Save here, satellite, coastline.
I'm on Jack.
There's uses a lot of. And feeling is for people.
And your friends. Your attention.
Leeway. Do something like this. I don't know that, yeah, so. All those values, relations, yeah. So it's right. So it's fine. You know, naturally, it's just like a big chest, you know, that. So it's just. Our ways pretty, yeah.
Yeah.
So it's not here, I don't think like communication is anything, yeah, but I'm trying to say. Something like you had this. Dumpster pile too, because that's on your stage, I see things. I grew up going to Monterey Bay, I used to go there. Never in my friends. And so the first ones I got, yeah, I was 22 and I was like, see, I like, I don't like that. I feel very humble. I like it. Been taking it for we're still here. But yeah, I read my. And also you should great. It's just too much, it's overstimulating. Yeah. Essentially, I was going to stay in Australia. Because I have to say, it's been our relationship. And I said, I'm right now, but yeah, I love that I'm still looking and love. Very hard to have started trying to make this so good. Relationship, but I also, when I was doing the class, we just slowly some many things. And I was not being able to say this to Vegas. I have to make a few to see what I'm doing.
Hi guys, I'm gonna wait a little bit longer just to see if anybody else trickles in.
But anyway, I love it real. That was sorry.
I'm sure. Yeah, I'm just gonna wait. It's fine.
Yeah, she did her undergraduate at Boston College and she's been trying to study purple. Without further ado, you can wake it. Pretty good, huh? Pretty good. Hi, everyone. Friend, perhaps. It's gonna should be shown.
Yeah.
Yeah. Because the only.
Oh, it's that one. Probably won't see them.
He would have.
It's right now.
I know, but I'll bring my opinion. Yeah, there. So I was thinking, Took it off. Order for you.
That's all it is.
Okay.
Video settings,
So I start our application, I set it to menu, and I turn on the order for you.
and then once it is.
Once you go back.
That's all there is. We need the 360 view panoramic. Only some wrong on that here anyway.
See if you have questions. See if it evokes questions.
Yeah, did you consider multiple collinearity in that regression model? So according to Tom's class.
Which first?
Your speaker mode.
And it's a speaker mode, and we'll search it on the manual mode, okay.
So there we go, we go to manual, right? And then. You see how it's moving because the auto it's turned on, once you turn it off.
Oh, yeah.
And that's not.
Input file. You wouldn't agree with that.
Good question. Well, you can hear about it. Really?
Wow. I don't think there's any rules. I know that, but
As long as you bring that to share.
Sure.
as long as I can have a bite.
I'm for everyone. Okay. Yeah. Yeah. You got it, yeah. Yeah.
All right, guys, well, welcome in. My name's Kayla Vidal, and I'm a graduate student here at Moss Landing Marine Labs in my second year. And I'm studying purple sea urchins. So today I'm just going to talk a little bit about them and their role in the ecosystem. I feel like they're misunderstood, but I can see how they're both seen as both a friend and a foe. So we're just going to review today what purple sea urchins are, why they're important, how they impact both humans and ecosystems, and some ways in which we can study purple sea urchins. So I'll talk a little bit about my thesis project and what I'm looking into. So what is a purple sea urchin? Simply, it's a spiny marine invertebrate. I thought our puppet show did a great job at showcasing our little purple sea urchins. And they talked a little bit about how urchins can form urchin barrens, but I'll get into that in a little bit. They have a really, really large range, so all the way from Alaska down to Baja, California. You can find them in the rocky intertidal zone as well as the subtidal. So if you're out tide pooling ever, they can be found hidden in little cracks and crevices. They're pretty cryptic, so sometimes they're hard to see, but they can also be found in great abundances, especially in the subtidal where they've eaten down a lot of the kelp forests. Purple urchin anatomy. I think purple urchins are really, really cool because they don't have a brain, but they can sense both temperature and light. They're mostly made up of their reproductive organs, their gonads, otherwise known as uni, which can be eaten in seafood. Their digestive tract, as well as some things to breathe, and their little tube feet that they use to both move around, to stick certain things on the outside of them to hide and camouflage, as well as to move their food into their mouth. This is an Aristotle's lantern. This is their little feeding mouthpiece. Purple urchins also have pental radial symmetry. So if you think of symmetry being like one side looks exactly like the other, purple urchins have that, but times 5. So they have 5 identical gonads, or nearly identical, and have that radial symmetry. So ecological importance. Despite the fact that we'll talk about the impacts that purple urchins have on ecosystems, they are actually essential to our kelp forests as important grazers and herbivores. They transform the bottom habitat, creating little crevices for other creatures to live in. They help control the different algae populations, creating higher biodiversity. And obviously they're important for different critters like otters, as well as other organisms like the sunflower sea star who eat them. Specifically, they're eating their gonads, which are the fatty reproductive tissue inside the urchin. Research importance. So the purple sea urchin is also used a lot in developmental biology. They're really easy to study and look at under a microscope. They're also invertebrates, so they spawn a lot. So you can do a lot in a very short time frame with purple sea urchins. And we've been able to learn a lot because we've been able to study them. They have a pretty well annotated genome as well. So while I'm about to talk about the ecological impacts, they are really important for both humans to study as well as ecosystems. They also have a really broad range, so it's really good to study them for climate studies. So they are exposed to this really large temperature gradient on the west coast of California. So they can be used to inform different, to get understanding on how organisms are changing with the different environmental conditions, which I'll get into that a little bit later when I talk about my thesis project. So unfortunately, urchins can just eat and eat and eat and eat unless they're stopped. And in California, especially where we are in the Monterey Bay, there's been a couple factors that have contributed to the boom in urchin populations and the decrease in kelp. So first was marine heat waves putting pressure on kelp, and the other was a disease called sea star wasting disease, which led into a huge reduction in their primary predator, the sunflower sea star, which helped to control their populations. Because of that, they've been able to graze down a lot of the kelp. And kelp is a really important ecosystem, both for fisheries, carbon sequestration. They provide a lot of ecosystem services. And these urchin barrens, while diverse in a different way, do not provide those same ecosystem services. And so a lot of research has been going into how we can control these urchin populations to bring back these productive kelp forests. This is a little graph about marine heat waves and sea star wasting disease and the change in kelp population and urchin population over time. If you see, you can see the exact switch. The first dotted line is the sea star wasting disease, where you can start to see an increase in urchin populations because of that wasting disease killing off their primary predator. And then you can see this marine heat wave, which created a massive reduction in kelp, which was just increased further by that increase in urchin population. And in the picture, you can see on the left, we have this urchin barren where there isn't any kelp anymore. It's hard to see, but there is diversity still on these rocky surfaces. But it's very different than that productive kelp forest. So one thing that people are doing in order to help with the urchin removal and bringing back these kelp forests is eating urchins, which I think they're delicious. That might be controversial. You have to try them because they're acquired taste, but I find them like sweet, and there's this fatty tissue, so it sort of melts in your mouth. But there's a up and coming aquaculture industry where people are trying to remove urchins from barrens and then grow their gonads in an aquaculture setting so that they can sell them. And if we can create a market for it, humans are really good at collecting organisms and bringing down the population. So maybe that can help bring back some of that kelp forests. There's also other restoration happening in places like Southern California, where they'll smash urchins in urchin barrens, which helps to relieve that pressure on kelp a little bit. And because the urchin barrens and kelp exist in this sort of patchy mosaic down there, there's enough kelp spores, not spores, but I don't know exactly what they are for kelp because that's not my territory. That they are spores? Yeah. Okay, cool. Spores that help to seed the rocky zone where the urchins were smashed to help grow those kelp forests back. And the restoration has proved successful in some areas. This is an example of work collecting the urchins, and then you take them into tanks, feed them a formulated pellet feed, and help them to grow their gonads. The gonads is a picture in that bottom right there. Those orange things are those gonads and what you would eat. So what more can we learn? My project specifically has to do with this climate stress aspect and how it affects purple sea urchin reproduction and physiology. I'm interested in how, as climate stressors increase over time, the urchin populations will change. It'll be good to know whether they're super resilient or whether it will lead to decreases in their population. It also will be important for in situ aquaculture systems. So how these conditions, ocean conditions are changing will change your aquaculture product. So phase one of my project, which is just to collect urchins, this is diving in Stillwater Cove. We create what we call a urchin lasagna to keep the urchins happy in these coolers. We lay down some kelp, put some urchins, lay down some more kelp. If you just put in water, it actually drops the oxygen condition in the water, and they're more stressed out by that. Phase two of the experiment is to put them in their treatment tanks. So I'm looking at specifically dissolved oxygen stress and temperature stress, and this sort of cross-factorial experiment design. So we have 3 different temperatures, 2 different conditions, stress and non-stress for oxygen, and each variation of those. And then this is sort of what our massive mesocosm system looks like in the tent. We have another facility a little bit farther south of here down the road called the Shore Lab, where we have these tanks and I can manipulate the conditions, and then I can collect the urchins and dissect them and measure certain physiological parameters. So this is me doing some data collection. I did a lot of different things. I measured the respiration rates of the urchins. I did collect these little pellets you can see all the chunks bitten out of them. That's me collecting the food after having fed them to measure feeding rates. This is just a picture of me opening up an urchin for some of these dissections where I'm going to remove the reproductive tissue and measure how much it's changed over time. And these are some of my friends doing some of the molecular dissections. I'm doing a differential gene expression analysis, which will basically tell me at the gene level what is changing based on these conditions. And I can get a bigger picture by tying the physiological data to these molecular mechanisms. So that's all for today, but I'm happy to stay and answer any questions that you guys may have. Great. Yeah, I'm the first in the middle.
Yeah, not specific to the research, but more of like a policy question. You mentioned there being a market down in SoCal for ways to mitigate urchin barrens, the culling and the culinary aspect. I understand MPA systems in Monterey are a little bit finicky around harvesting, but like in your research, have you learned like what makes it so complicated as to why Santa Barbara can do it and we are like still suffering like without like a very healthy trout forest?
So we can do urchin culling up here, but it's not as successful because of what I said with the spores and the kelp, and we don't have as many healthy kelp forests to help see those barrens back.
Sure.
I don't know exactly the policy aspects. I'm hoping to learn a little bit more about that this year because I'm partnering with the Kashaya Band of Pomo, who's an indigenous tribe that works with purple sea urchins, Monterey Bay Abalone Company. I'm doing a lot more outreach this year where I can hopefully learn a little bit more about that policy aspect because it's really unfortunate. There is only like 2 or 3 commercial divers that are allowed to collect urchins for aquaculture.
Yeah.
And so like Monterey Bay Abalone Company, for instance, like we collect all their urchins and give them to them and then they raise them.
Yeah.
And so there is a lot of policy is one of the big things that is like holding people up, specifically with urchin aquaculture. Going out and killing them is a different story. Sometimes you can, but it's not always successful, you know, and you need a lot of.
Incredibly successful, unfortunately, Fish and Game Commission has decided that doesn't matter. That they want to do 5 years of no culling in order to see how much of the kelp dies back. Wow.
That's great to know. Yeah, that's unfortunate.
Giant kelp restoration project.
What was that?
Keith Ridsart and the Giant Kelp Restoration Project.
Thank you.
And that's in Southern California?
No, that's here.
That's here in Monterey? Okay.
Yeah.
Thank you.
Yeah. And is the culling you were talking about at Tankers Reef?
Yeah.
Okay.
How fast does the kelp grow each day?
I don't know how fast kelp grows each day, but I do know that from some of these culling, like long-term monitoring studies, we've been able to see within like 2 to 3 years kelp grow back. But I'm not exactly sure how fast.
Somebody told me that they grow an incredible amount.
They do, yes,
And
yeah.
Are they annuals?
I
I understood.
The macrocystis is multi-year. The Nerio, the bull kelp, is annual, which is part of the problem. North coast, north of San Francisco, the bull kelp tends to be predominant. And because it's an annual, once you clear it, it's really hard for it to come back. The macro can live for many years when the hold fast and will regrow. So if you have a bad year, the macrocystis can come, the giant kelp, like that stuff there.
Or the giant kelp.
Yeah, that can come back more quickly and can be more resilient.
The bull
So-
kelp's not a good kelp to have.
It's an annual, so once at the end of the year, it just let's go and
does it reseed itself?
it will receive, but not from that same holdfast usually.
So it's harder,
Yeah, it's a,
for restoration, it's different.
that's why up in the north coast, they're actually growing bull kelp in the lab and putting them out to help recover the,
Oh,
yeah.
okay.
Is your proposal publicly available?
Not currently. No, but I do work with undergraduates at CSUMB, and my project is still listed under UROC if people were interested in like reaching out and helping at all. I've run my mesocosm experiment. That's what I did last semester. It was 8 weeks long. I didn't get too much into the nitty-gritty details because I wasn't exactly sure like the different types of people that we'd be here, but I mean, I measured respiration, I measured feeding rates, I took gonad mass and other body condition metrics. I also took the tissue for differential gene expression, which I'm working through right now. We also took tissue samples for looking at the microbiome. And so and then I'm also looking at the gametogenic stage. So how fast gonad maturity might change with different climate conditions. And that's this, I'm wading through data right now. But yeah, but if you're interested in reading more and talking, we can talk. Yeah. Yeah, a lot of questions. Okay, here.
2 questions, actually. What's the typical lifespan of the sea urchin? The purple?
They're a really long-lived species. Yeah. And yeah, forever. I don't know the exact.
Decade?
Yes, yeah.
Oh, wow.
But they're interesting in the urchin barrens, they exist in this sort of zombie-like state where they shut down all of their processes, but they still survive. So they're notoriously, I mean, physically not hard to kill, but hard to kill in terms of conditions. And even like seeing with my experiment, the thermal threshold the 20 degrees is kind of their top end before you start experiencing a lot of die-offs. However, a lot of them still lived, and it was only with the addition of the dissolved oxygen stress that a lot of them began to die. And so they can exist in stressful conditions for a long period of time. Not to mention like their range, not only from Alaska to Baja, but also like intertidal through subtidal. They exist in just such a wide gradient that they're extremely robust.
And the respiration, is it a continuous flow or a cyclic kind of a thing?
Yeah, we did continuous flow to measure the resting metabolic rate. And we had this sort of this tank system. We had 1 for background noise and then 3 urchins being measured at a time. The entire system goes into the tank. They're submerged underwater. We kind of unscrew the lid, put the urchin in. We have a little sensor. The water runs through the system, gets flushed, and the oxygen gets measured for over the course of 30 minutes. Yes.
Are there any specific months or seasons that include meat size?
I don't know about demand for uni, but they're depending on where you are, depends on like when they're growing their uni. So for the Monterey Bay population, at least in the fall, they're growing their uni a lot. And then they spawn in the spring and into the early summer. And so we've had difficulty, I mean, I'm not doing these experiments, but people have had difficulty getting them to grow their gonads out of season because photio period seems to matter a lot for them, but we haven't been able to really fine tune that aspect. But if you give them food, they will grow their gonads. It's one of those things that are very plastic, where if the conditions are good, they're going to grow their gonads and invest energy into their reproductive organs. It doesn't scale linearly with their body size. Yes.
You mentioned that there was no water in the holding tanks in the urchin lasagna.
Yeah.
Did that kind of set also a good kind of baseline for measuring respiration for all of them, since they were kind of in the more air exposed conditions together?
So we tried to standardize conditions as much as possible before the experiment, where we collected them. They were all collected at the same time. We brought them back to our facility. They acclimated to our system for 2 weeks, and then they were exposed to the temperature slowly over a week-long period of time. For respiration, it was only measured once, and it was at the 6-week time point in the experiment. But they were all starved for a 24-hour period before the respiration was measured. So that's kind of standardized it. Yes.
Hi, so I'm interested in the physiological and genetic aspect of your study. If you're looking at a particular physiological variable, do you already have genes accessed, or not really accessed, but you understand the genetics behind the gene and the physiology, or are you just looking at variability within one gene?
No, so I'm doing, I'm collecting RNA and doing RNA sequencing. And I don't know exactly what I'm gonna see yet, but there hasn't been a lot of knockout studies done to know exactly the function of certain of these genes. So I'm getting more of a broader picture. I think people tend to overclaim what you can learn from, RNA sequencing, and Esperatus, while it's, the purple sea urchin is said to have a really well annotated genome. I haven't exactly looked into that yet, and so I'm gonna look into a few more recently published studies, and to see how well it's annotated is, to see what we can actually learn. But hopefully doing certain downstream analyses through, after getting the gene counts and then some gene ontology stuff, we can see more broadly what processes are most affected by these stressors. Yes.
I'm curious about the biodiversity that you mentioned in the urchin barrens, because that's not something that I have found.
Yes,
What all are you seeing here other than urchins and non-stipitate kelp?
So on a smaller level, I'll have to look through my literature to find the study that was published if you're interested. But there was a study done looking at like Shannon Diversity Index, like those classic diversity measures, and there is still, it's lower. It's much lower in the urchin barrens than it is in kelp forest. Like, I'll be clear about that. But there is still a lot of this like prostose coralline algae, tiny little inverts, things that don't need big kelp to grow. Also, because there isn't this big kelp that's like shading the canopy and taking all that sunlight, there's opportunities for other like smaller algae and things to grow. But a lot of things obviously get eaten up by the urchins really quickly. So I think my point was just that there still is diversity, even though it is much lower. The communities look a lot different. And when you talk about succession and you talk about invasive species or species that are doing having detrimental effects on the ecosystem, like, where do you draw the line? At some point, we can't return an ecosystem back to what it was. And so, yeah, I just think there are interesting arguments and conversations to be made about things like that.
Yeah, part of the problem is that these ecosystems are bistable. So they're stable as a kelp forest.
Right, yeah.
And they're also stable as an urchin barren.
Yeah.
And it's really hard to get them to flip.
Yeah, talking about these phase shifts, it takes these massive events to either wipe out all the urchins to get the kelp to go back, and yeah. Yes.
I think it's worth noting that the giant kelp restoration was very successful in the area they had while they were allowed to do it.
In Tanker's Reef here?
And Tanker's Reef
Okay,
was
yeah.
successful. They grew they grew full kelp forest.
Okay, yeah.
At the time they were able to do it. But getting there was done, I think the average dive for a culling dive was certainly north of 200 urchins culled, possibly north of 500. And for a skilled, experienced diver, 1,000 was not an unusual number.
Yeah.
So that's a number.
And it's reducing down to a density of less than 2 per square meter.
Right, and so it's not reducing to 0, it's reducing to less than 2 per square meter. But that's a number of culled urchins that I don't think the scientists could handle.
Yeah.
Like, I don't think they had the stomach for that level of work.
Yeah. Yeah, and did you, I've heard about the Tanker's Reef project, but I haven't looked into it a lot, so I didn't know a lot about the success, but I definitely have heard about it. Do you know how long they monitored after before the efforts were stopped?
So they're still doing, Reef Check is still doing monitoring on that site.
Okay,
periodically.
they're still doing monitoring, but are they still doing culling?
No, the culling was stopped.
Okay.
And then the culling, when they announced the culling would be stopped, the leadership on the project decided they weren't willing to have divers out attempting to salvage a kelp forest. They were just gonna be forced to leave behind anyway.
Okay, gotcha.
And so there's a social engineering aspect that fell apart there. In that the science was being done on will divers. The divers were the experiment. And it turns out that divers, when they're the experiment, are don't like finding that out.
Gotcha. Yeah, that makes sense.
All right, thank you, everyone.
Yeah.
Now we're at your question.
Yeah, it's a little sticky here. I think they have the line that's so far along.
Yeah, you got to put yours and then.
Okay, my.
Yeah, there you go. No, yeah.
And is there here, I can.
Some plants grow, I mean, yeah, you might find
There you
it.
go. Yeah, let me try this. I think you might be able to press the space bar and continue, yeah.
Yeah,
But if not, yeah.
I like to wander.
Thank you.
Great job.
My advice is it's like something you. Especially if you always are. But yeah. I can't find it. Is that? Start looking at the family.
Oh, no, you're not gonna work.
I guess I'm published on the lab page, like I want some that, but.
It's not, it's doing something weird, but it's fine, like I don't need to, well, let me, maybe I'm doing it. Oh, there we go.
Okay.
Yeah.
So I'm gonna go ahead and get started, so we stay on time and we don't keep getting later. So I'm pretty good about talking over people as an instructor. A lot of times you have to talk loud. So today I'm gonna be talking about one of my favorite organisms, the emperor penguin. So this is some of the research that my lab has been doing. So over the last 6 years, we've been funded by NSF to study the habitat use and behavior of emperor penguins. So before I talk about our research, I thought I would just give you a brief introduction to emperor penguins. So there are 18 different species of penguins, and they're all found in the Southern Hemisphere. They range in size from a really small little blue penguin that weighs about 3 pounds up to emperor penguins that weigh closer to 80 or 90 pounds. So they're quite big. Emperor penguins have a circumpolar distribution, so they're found all around the Antarctic, and we actually did not know there were 66 colonies, although this number does change a little bit year to year, until about 15 years ago, when satellite imagery got so good that we can identify penguin colonies by their poop. So basically, they look at images all around the Antarctic, and you can identify where the colonies are based on big guano stains. So before I talk about the research also, I just wanted to tell you a little bit about their annual cycle. So this is one of the colonies that I worked at during chick rearing. So this top bar is the time, kind of the annual life cycle of a female, and the bottom is the males. And I'm gonna start here at kind of in April. This is when we often hear like they return to their breeding colonies. They have about a 6-week courtship period. And at the end of this period, the female lays the egg and she transfers it to the male. So what you see here, this white means that the animals are on ice and they're not feeding. So the females fast for about 6 weeks, transfer the egg to the male, and then the male continues to fast. It may fast for up to 4 months. As a physiologist, I find this fascinating, but I won't talk about that today. But then when the female comes back, the male goes to sea and eats for about a month. And then for the rest of chick rearing, they alternate. Initially, when the chicks are young, one parent is always there. By this time, the breeding season, you can see the chicks are pretty big and they're starting to be on their own. So at this point, when I've been studying them, we put tags on the birds. They go out to sea from anywhere from 5 to 15 days. They come back and they often are with their chick for less than 24 hours. So we actually are scanning for them 24 hours a day, just to make sure we get our tag back before they go back to sea. So my team has gone down to Cape Crozier, which is on the tip of the Ross Sea, so where McMurdo and Scott Base are, for 3 different seasons. This is the 2nd season in November of 2022. We instrumented birds during chick rearing and at the end of chick rearing and studied their foraging behavior. At the end of this season, I knew that we were planning on going back with the ship to this really remote area that is not accessed very often, the Eastern Ross Sea, which we know is a really important molting area. So at the end of this season, we put out some satellite transmitters that told us where the birds went to molt. Pretty much all of the previous studies found that they typically molted here. I have a box that shows that. This was kind of where we thought that they molted these birds. They went so much further. But this was a year that had record low ice. And so normally there's a lot of sea ice for them to molt here. Like this has been reliable for a long time, always good ice here. It did not exist in 2023.
So we saw the birds traveled so much further before they molted their tags. So just a little context about why we are studying the birds, and especially in this Ross Sea area where I work, is if we don't change our behavior and climate change continues at the rate that it is going, emperor penguins are basically going to be extinct in 80 or 75 years. Two of the colonies where they're predicted to be okay are the two southernmost colonies. And this is the colony Cape Crozier where I work for the 3 years. And then there's one over here in the Weddell Sea. I like to be a little bit more optimistic if we actually follow the Paris Agreement and only have an increase in 2 degrees. You can see that the Weddell Sea and the Ross Sea populations have a better likelihood of surviving. And it's even better if we can keep the increase to 1.5. So I like to think that if we do change our behavior, there are some colonies that are likely to persist. But these birds have just been listed as endangered this month, because of the impacts of climate change.
What does RCP stand for?
It's just one of the models. It's sort of the baseline model. I can't remember what the abbreviation is. And honestly, this prediction is 6 years ago. I'm going to show you some data at the end from a paper that I was part of that we just published last month that is showing us that this is happening much faster than we would like. So most of my work has been taking place in the Ross Sea. This is an area where about 25% of the world's emperor penguins live. Most of the colonies are on the Western Ross Sea, and then there is one colony that's been growing over the last 15, 20 years in the Eastern Ross Sea called Cape Colbeck. It had been thought that while ice was decreasing in the peninsula and Bellinghausen region, this region for a while was having increasing ice, but that trend has changed. And for the last 12 years, I think 7 of the last 10 years or something like that have had record sea ice, low sea ice. So this refuge may not be as good of a refuge as we thought. So for NSF grant, we're studying the foraging ecology and habitat use of Ross Sea emperor penguins. Today, I have students that are working with this late ship rearing. They're doing some habitat modeling and detecting feeding. But today I'm just going to share some of our early post-molt migration results. This is a time period in emperor penguins where we know very little, because we know where many of the colonies are. We can go to these colonies, tag the birds, but then they do this migration where they swim to that Eastern Ross Sea region to molt, and then they molt the tags off, and then we have no idea what they do. So very little is known about this period, and we were able to convince NSF to give us access to a research vessel to go and find where they molt. So just like, in order for us to study the migration, that's post-molt migration, it was quite a migration for us also. So we started in California, went to New Zealand. It was sort of during the end of COVID days, so we had to quarantine and wear masks. Our flights got delayed due to weather, so we were very happy when we finally got on the plane. This was our lovely transport from New Zealand to McMurdo. We were even happier when we got off the plane. I have to admit, I've been on this plane a couple times where it gets almost to Antarctica and turns around, so just getting on the plane is not enough. But we had good weather, as you can see here. We arrived. We then had to go into quarantine again. Then we got to take the bus back. That was really fun. So they have fun vehicles down there. Then we went to quarantine again and had to get tested for COVID numerous times before they'd actually let us on our lovely research vessel in our home for the next 6 weeks. So this was the day we finally got to take our masks off and get on the ship. And then that is us as we were leaving McMurdo and finally starting our adventure. And then we had to spend 4 weeks on the boat while geologists collected their data. We just looked with our binos and we documented any animal we saw, but finally at the end of 4 weeks, it was our turn. And we got to go to the area where there was lots of ice. Geologists found the ice annoying. We like it. That's where the birds were. So we'd go through this ice and we'd be looking at these ice floes to try to find emperor penguins. We saw a lot of ice that we weren't allowed to work on and would not be safe. And we also found other species. So these are Adelie penguins. What we were looking for are nice big ice floes like this. And here you can see 4 or 5 emperor penguins, or more than that, 6 emperor penguins that we were very excited to get out and try to catch. So once we saw that, it was time for us to get in our Zodiac and head out to the ice floes. And then we would try to, this is a different location, but it was a beautiful day. So I like the reflection. We would try to get between the birds and the water. And then we don't want to disturb that large of a group.
So we would then cut in and just get a smaller brood. And this is kind of what that looks like. So we have these large shepherd hooks that has like a little padding on them. And I don't show the actual capture, but we wait until someone can hook the bird. You'll see someone upstairs. And then we can go in and give it a nice big hug. Yeah, and so he's just about to go in, hook that bird right there. And then one of us would go in and give it a hug. Once we catch it, they actually calm down. So we put this hood on them, and once they can't see, you can see that bird is just basically laying across his leg. They every once in a while will try to flap their wings, but as long as they feel pretty secure in that. And then we just use super glue and tape to attach our data loggers. So this is an example of one of our tags. The reason we do this is we don't damage their feathers very much. So as soon as this tag falls off, their feathers might have a little bit of glue. But when they molt and get their new feathers, there should be like the glue does not touch them. We're just able to put this tape under their feathers and wrap it around. And most of the glue is actually taping our tape together. So this works pretty well. These tags transmit, so they're time depth recorders. So they collect depth data and then they summarize it. So they transmit their diving behavior to us over the satellite, along with just kind of course scale location. So we get we probably got about 30 good locations a day. But we broke it up. So we had a kind of a good location every 3 hours. This is kind of what our group looks like. The penguins were really not that concerned. You can see this guy here. We'd already tagged him and he's just waiting for us to finish with his buddy. There's Brazilian waiting to take us back to the ship. And then the next day we went out and we actually saw one of our birds hanging out. So we were pretty excited to see that it was collecting data. And we could go and verify that. So they don't seem too concerned about us once we let them go. So once we tagged our birds, so we tagged over 30 birds, then it was our turn to migrate home, and it was not very fun. Our captain was really amazing and managed to get us to go through the eye of like right between two storms, but even between the two storms, it wasn't very nice. But I managed to go to the gym every single day but one. I was very proud of myself because 12 days of this is not very fun. This is an example of some of the data that we got from the birds. So we ended up having to go a bit further east than I was expecting. And we did most of our tagging. We did some over here in Cape Colbeck where I did planning. But we ended up going to this Rupert Coast area. And this is just data from 16 of the birds that we tracked, that we tagged there, that returned to breeding colonies. 8 of them did what I had expected based on the only other previous study that had been done on breeding birds. They went back to these Western Ross Sea colonies. So Cape Roger, Woolman Island, Cape Washington, and we actually did have one come back here, but it was one of the ones we tagged at Colbeck, so I did out. Half of the birds stayed either at Rupert Coast or Cape Colbeck. And this was something we weren't expecting. We thought maybe 1 or 2 would based on previous studies. But what we think is happening potentially is that as the ice is no longer here, more birds are potentially trying to remain in that area because there is really big differences in their behavior. So the birds that returned to the Western colonies had to travel 2 to 3 times the distance. So they traveled a lot further and they had to do this at a much faster rate. So they traveled about 40 kilometers a day versus the birds that ended up staying in Cape Colbeck or the Rupert Coast to breed only did about 20 to 25 kilometers, which means that these other birds likely have much more time to rest. So we're looking into that now. And then one of the things that was most concerning is that these animals are really reliant on sea ice. So there has to be good sea ice when they return to the breeding colony. But the ice needs to stay good until mid-December when the chicks fledge. And so the birds that ended up traveling so much further arrived at the colonies 2 weeks later, and 2 weeks later than they usually do. So it does suggest that this decreasing ice is potentially resulting in changes or timing in their breeding. So just kind of to summarize that with some visuals. So the year that we were out there at that time, January, February 2023, that was a year of record low sea ice. There has since been 1 or 2 more years of record low sea ice. But in this year, the Western penguins traveled further than we had ever documented before to reach stable sea ice. They need this stable sea ice when they're molting because they have a catastrophic molt. They lose all of their feathers.
And during the 1 month that they're molting, they can't go in the water. It's cold, it's energetically demanding, it's very risky. So they really do rely on this stable ice, and apparently they will travel very large distances, long distances to get there. If the low ice conditions persist or worsen, I'm sure there will be years with better ice, at least I'm hoping, but if this trend continues, Western birds may start to incur increased energetic demands and more risk, spending more time at sea. And then additionally, these Western breeding birds may experience a mismatch in timing. So the ice might break out before their chicks are old enough to fledge. And this is just when I first went down there to study post-molt behavior, we went down in 2013. And there was actually quite a bit of ice. This is where we did all of our observations of the birds. But these pink lines this is what the historical average is. So this area has always had really good sea ice. In the last few years, there's no sea ice in this region when the birds need to molt. So it's kind of tough for them. And then just to show this in a slightly different way, these colors show so there are some areas in Antarctica where sea ice is increasing. So this is the percent change per decade. But in the area where it's the largest population of molting emperor penguins, there's been about a 15 to 20% decrease in sea ice in that region over the last few decades. This is a paper that just came out this week. Don't get too lost in all of the graphs. The main thing I want to point out is that Western Ross Sea colonies, so Beaufort, Washington, Pullman, Franklin, and Roget, and also Cape Crozier is one of those, but most of them have been declining. The two colonies that had an increase was Crozier and then Cape Colbeck. So Cape Colbeck is that eastern colony. So for a long time, if you ignore the last 5 years, the population was relatively stable, and we think the birds were just moving around. So maybe if the sea ice was changing, more of the birds were staying on the east, and that explains why Colbeck was getting so big. Crozier, maybe more birds were there because it's one of the most southern colonies. But unfortunately, in the last 5 years, even these colonies that were increasing, so these 5 years with record sea ice, we have started to see drastic declines. And so this is showing when they did the long-term data, there was actually a very gradual increase in the Ross Sea metapopulation up until 2019, at which point it has declined by about 30%. So recently, because of some of this evidence like this, these birds are designed to have some years of failure. They live a long time, they have a chick every year, but having this constant failure or bad ice seems to be having an effect. So like I said, just earlier this month, emperor penguins have been listed as a threatened species. I'm hoping with the world kind of recognizing this issue, this population actually spends most of its time in one of the world's largest MPAs. But climate change is something that an MPA can't necessarily fix. So we're hoping to keep doing more work to maybe identify areas that can be protected because the sea ice is doing better there. Yeah, and so I don't think I'm gonna have time for questions because I talk too much. So.
The Google slide. Is it up here?
No, it should be.
It's a Bible slide. It's a Google slide.
You didn't download it as a PowerPoint?
It's right here. No.
Oh, I like PowerPoint better.
Are they gonna see my notes? That's really odd. How do I get out of that?
Yeah.
I don't want my speaker now. Okay, there we go. You don't have to leave there, right? It's fine.
Try to change.
Do I bring it right there? You can go ahead and introduce me while I do this.
You're a second year or third year?
How do I bring you over? What's wrong with me?
All right, good afternoon, everyone. We're gonna start the next presentation. This is Ava. She's a third-year student at Cal State Monterey Bay under the direction of Dr. Cheryl Logan. And her primary focus for her thesis project are corals based out of the Galapagos.
Hi,
Hi, thank you, Javi, for introducing me. My name is Ava. I'm a master's student here at Moss Landing. Like he said, my primary advisor is Dr. Cheryl Logan at CSU Monterey Bay, and we're in the coral and fish ecophysiology lab. So I'll be talking to you today a little bit about the life of a coral scientist and how I specifically study corals. So to start off with the question for you, what do you guys think of when you think of someone who studies corals? Feel free to raise your hand. Yeah.
A diver.
A diver, yeah. Other folks? What do you know about coral science? Yeah.
Like modeling distribution changes related to like climate adaptations and climate change.
Okay, nice. So you know your stuff. Nice. Well, going off of, oh, I don't have access. No. Okay. Keep going. Well, you said diving. Most of the time people think of snorkeling, taking photos, doing coral restoration, like coral gardening, or just going on reefs, doing distribution modeling, doing health assessments, things like that. And you might be right. This is what I do. This is me in all of these photos. However, this is only what I do about 5% of the time. What do you think the other 95% of the life of a coral scientist is? Yeah. Computer coding. Computer coding, okay.
Chemistry with samples.
Yes.
Looking through a microscope.
Looking through microscope, exactly. So the other 95% is about 1/3 lab work, about 1/3 data analysis, computer modeling, reading lots of scientific literature, all papers that are posted online. Most of the time it's open access, so people have data available readily to them. And being stressed is about the other 30% of the time. So I take all of these things, I take the 5% of field work, I take all of the work that revolves around analysis and lab work, and I put that and I answer my specific research question, which is why do some corals survive stress, specifically temperature stress, while others don't? And so the first method that I use is taking field methods and taking computer modeling. So what I do is I take thousands of photos using a GoPro, and we go in about a lawnmower pattern. So we go back and forth, and we overlap all of that, and we get lots of photos that look like this. So you can see all of the different corals. The corals are laid out in colonies and rows. And so we snorkel throughout these rows. And so you may be thinking, okay, these are kind of horrible photos, like nothing is in focus, like they're out of the way. But these photos are important because we want to get all sides of the coral. And that's important because we use these photos to create 3D models of our coral restoration sites. So here's kind of an overhead photo. This is a model that I made using those photos. And this is kind of that 3D area where you can really see that coral as a 3D image. So what we're able to do is take these at different time periods. So taking it from an initial time point and a next time point, about 6 months to a year apart. And we can take those and put that into a computer software and layer them on top of each other to look at growth. So if I take a specific coral colony here and I take it out, I'm able to layer the 2 different time points on top of each other. So the yellow on the outside is the earlier time point and the red is on the outside. And so what we're able to do is generate these, they're called point clouds, and we're able to look at coral growth over time using these. So this is just another way to look at coral growth besides just going out and taking a ruler and measuring it over time. It's more precise and it just looks pretty awesome in my opinion. It's been really fun to do. And so from there, what we're able to do is look at growth over time. And so what my job is to look at which corals are growing the most and identify that and give that information to restoration practitioners. So here's an example. Here's a coral in July 2024 and one in July 2025, and you can see that it grow over time. But you might not be able to look at that and identify the exact growth. So what we did was that this is a ranking graph. And so the top is the fastest growers and the bottom are the slowest growers. And so we were able to see that, okay, P4, P14, and P13 colonies were growing the most. So we're able to take that and give that to restoration practitioners so they can outplant those in the future as well. So that's growth. And that's the photogrammetry work that I do. It's called structure from motion photogrammetry for those familiar. And this is kind of the lab component that I do. So we went and we scuba dived and we free dove to gather coral fragments via scuba. And from there, we swam to the surface where we sat on lava rock, chopped them into smaller pieces like this, put them in tubes to preserve the DNA, and then they were shipped back here to CSU Monterey Bay, where in the lab, I extracted the DNA and I did genetics work on them. We looked at which type specifically the corals were, as well as looking at their symbiont composition. And what coral symbionts are, is you may have heard of the word symbiosis. It's a mutual beneficial relationship between 2 organisms. And so corals have a mutually beneficial relationship with their symbionts. Specifically, as we know, corals are animals, but they have this algae that lives within them. And so if we take a look at a coral polyp, we can blow it up and look like this. And specifically, looking at the color, all of those little dots that are here around the coral, that is all algae that has that symbiotic relationship with it. And algae gives corals color. So if you heard of coral bleaching, that's when the corals kick out all of those algae and they have their white skeleton is remaining. So one important thing to note is that corals can actually change their symbionts. So here we have the symbionts represented as dots. And whenever a heat stress event or a stressor event occurs, they can kick those symbionts out and get them out of there. And from there, they can find a new symbiont and take it in. And so typically, this new symbiont is able to resist these thermal stress events or these heat stress events as well. And so the coral is so smart that it's able to do that and have resistance and gain that resistance by changing their symbionts over time. So what I found was that in the Galapagos, corals can change their symbionts. So to orient you, this graph is time. So we sampled in July 2023 all the way to July 2025. And on the y-axis here, we have two different types of symbionts, Platycopium and Durastinium. All you need to know is that the more resistant or the more resilient symbiont is down here, Durastinium by the muscle, and the least thermal resistant is at the top. So as you can see, in July 2023 and in January 2024, the corals were more of that Platycopium. However, there was an El Nino heat stress event in April of 2024. And after that heat stress event, you can see by the box plot is that it went down more towards this heat tolerant symbiont, meaning that these corals are probably shuffling and changing to that more heat resistant symbiont. And what was also interesting was that they went back a year after that heat stress event and went back to what they were before pre-El Nino, which is super interesting. And so, like I said, is that I'm able to take all of this data and use it to help inform restoration practitioners. So I partner with the Galapagos Reef Revival Nursery. I'm sorry my video isn't playing, but if you want to follow them on Instagram, they're a great place to watch cool coral restoration videos. It's the very first coral restoration site in the Galapagos. It was founded in 2022. Most of the time, you guys probably don't think of corals when you think of Galapagos. You think of marine iguanas, hammerheads, things like that. And that's because in the 1980s, an El Nino event wiped out 95% of all corals there. So the ones that are remaining are kind of sparse colonies like the ones you saw in those models I made. So this is a really important work that is going on there. And it's one of the very first ones or the first one that has happened there. And so I got to go there and help clean some of the reefs and help clean some of these sites that we restored. And all of my work will then inform the site as well. So just some key takeaways for you to take home if you didn't listen to anything I said, is that coral scientists combine all these methods. We're not just scuba divers to beat that stereotype. Additionally, corals and their symbionts respond to stress differently. So like I said, there can be more thermal resilient corals, but there can also be more thermal resilient symbionts. And so they're able to match those together, and we're able to figure out which are the most thermal tolerant corals and which are the most thermal tolerant symbionts, put them together, and then outplant those to restore the reefs that have been degraded in the Galapagos. And I just want to take a moment to acknowledge my advisor who helped me with this work, the Galapagos Reef Revival Nursery and Moss Landing. I was able to apply and got funded for scholarships to go down to do all of this work in the Galapagos and do all of my lab work. And I got also a bunch of money from all of the money we raised at Open House the last 2 years to be able to do this work. So if you're so inclined, I would love it if you donated to student scholarships as well. And I can now take any questions that you can have.
So with the symbionts, when they take up a new one,
yes,
are they selectively bringing in the better one, or is it just sort of random and that's what's thriving?
That's what we think, is they're selectively doing that. And it's different species are different. So I studied Passalapra, which is a branching coral that's found throughout the Pacific. And not all coral species are known to be able to shuffle their symbionts. Typically, lots of that work has been done in the Caribbean with elkhorn coral and stackhorn coral, which are the most common that are found there. And they have been found to be more selectively. But I'm not sure specifically about Passalapra, but I believe that it's a selective choice. Yeah.
For the specific corals you worked with, what were the benefits more of switching out the different zooxanthellae, and what benefits were they giving the corals and the zooxanthellae?
Okay, so the question, I don't know if everybody heard it, was what's the benefit of the corals for switching? And then what's the benefit for the symbionts for switching as well? So the corals, Long term, it's important for their thermal tolerance. And so by switching those symbionts, it can help increase their survival long term. And so we can look at growth and we can see how fast corals are growing. But if they're not taking in those more thermal tolerant symbionts, then they're not going to survive long term. And so we have found that there's a trade-off with the fastest growers have the least thermal tolerant symbionts, which is bad long term because they're not going to be able to resist hotter events that happen in the future as well. I'm not sure about the symbionts. I don't think anybody has done specific studies on looking at the benefits for the symbionts, mostly just focused on the corals. Good question. Yeah.
What's the mechanism for the coral booting out the symbiont?
Do you mean like the trigger?
Yeah.
For it?
Like
Typically.
like they change chemically or?
It's more of like a physiological mechanism for it. So they have that symbiotic relationship, but I don't know about the mechanism itself. I just know typically about the trigger, and it's typically a stress like pollution or temperature typically.
But the question is more, how do they actually exile them?
I'm not sure. I don't do more technical molecular work like that, specifically on looking at it. I'm more of an ecologist, but there's many papers online that I could refer you to. Good question though. Yeah.
So since a lot of coral species are going through like bleaching and physical degradation, how long can they withstand that massive heat change? Like, can they last and completely adapt to that heat change, or can they last for only a certain amount of time? Or
That's a good question. So specifically, I can speak to the Galapagos and the fact that the corals that are still there, they're the strongest and have resisted the most bleaching events. So I think looking at, if we're talking about future restoration efforts, looking at the ones that are still remaining in areas that have faced degradation, like the Caribbean, like the Great Barrier Reef, and looking at those corals and how they survived those events. So doing more work like this it's called corals at the edge of their limits pretty much, or in marginal environments, which is having been faced by constant stressor events like bleaching events too.
So would that lead to probably introducing that species of coral events since they're able to be most resistant?
Maybe not introducing new species, but definitely increasing diversity, I think will always increase survival long term. And potentially doing those like super corals of figuring out what the strongest coral is and the strongest symbiont and putting them together too. Good question. There's abalone races at 1 p.m. Everybody's interested. Yeah, I'll do one more question.
What was like the range of temperature that you looked, like the temperature distribution that you looked for in the symbiotic relation?
So during the cooler periods, it was about 20 to 22 degrees Celsius. And then the peak was approximately 28, 29 degrees Celsius during the heat stress event. So we looked at over the full 3-year period. So I did a year before I started doing my fragmentation and coral collections. And that was during a cooler period. And then we also measured during that El Nino period as well, the warmer.
Any other questions? All right, one last round of applause.
Just to stay like.
Yeah. Our moderator.
You'll just. Honestly, like, it had you. It's 20 minutes. But they'll be kind of this late.
Oh, you're in. Yeah, oh, yeah.
I'm the other.
Oh, yeah.
Sorry, I guess this is.
You can be nice. I can be in charge of all the labs.
Seventeen is on fishing. All right, that's great. Don't have to be final. I think that's where we were. Oh, no, that was. Oh, yeah, like for them, we can say that. How you do is great, then you're done. It's like. That's small.
Yeah.
Really, so that I don't think they're. What was it for that was less? Okay.
What does that? Oh yeah, I have to share my screen. Alright, let me see if this works.
Also, do you want me to introduce you?
Yeah, you can. Actually, I'm just trying
What do you want me to say?
Just right at the start, just say, hey, okay, we're about to start our presentation. If you have questions, just hold on to it.
Okay, and then introduce your name. Yeah.
Yeah, I need to figure this out. Trying to put this in presenter mode.
For sure.
This is not good.
I'm also on Thursdays and Sundays.
Hello, everyone on Zoom, can you see the screen? Just to make sure, give me a thumbs up.
Just to
So you're gonna go back on. I'm gonna go up to 101.
Yeah.
Then back. Yeah.
Working. Okay, yeah. Elsewhere? Yeah. Okay. Just trying to pull that up. Yeah. All right, so you go. Yeah.
Hello, everybody. Thank you for joining us or for continuing to join us. We have the next presentation here that's going to be led by Anthony. So if you have any questions afterwards, be sure to share them.
All right. All right. Hello, everyone. Thank you for showing up, spending time on your weekend to be here at Moss Landing Open House 2026. So again, thank you for the introduction. My name is Anthony McGinnis. I am a graduate student, a part of the Ocean Predator Ecology Lab ran by Dr. Salvador Jurgensen, and at Moss Landing, a part of the Ichthyology Lab, where I study juvenile white sharks along the California coastline. And I previously come from the CSUOB Long Beach Shark Lab, where we also study all of the nursery grounds in Southern California. So to start off, let's talk about some physiology and size cohorts. So there's different size groups. So we have neonates. Those are the babies freshly born, and they range from 3.5 to about 4 feet long. Then you have the young of the years, which are in between 5 to 6 feet. And then you have the juveniles are 6 feet to 10 feet. And then everything above 10 feet and up are subadults and adults. And then also some reproduction history about these white sharks. So white sharks are born living. So it is a reproductive process called ovoviviparous, meaning that they are not egg layers. And they develop all of the sharks or the embryos inside the stomach, where all of those embryos get the uterine-rich milk from the mother. And then they develop it later on in the stage, and they do something called oophagy, which is when the sharks develop to the point to where they start to eat on their brothers and sisters for survival. Yeah, it's pretty wild, yeah. And at one time, the mothers can have 2, sometimes up to 15 pups in one litter event. And you can assume like those embryos are eaten quite a bit if the other ones don't hatch enough in time. So, but just like our good friend Bruce, we have juvenile white sharks, where once they go off the coast to Hawaii, they mate, and then they come back to the shoreline, and then they pop out their babies, where we don't know where they go. And just like our friend Bruce from Finding Nemo, we don't know, or they don't know their parents. They don't have any parental care at all, so their babies just out to fend for themselves along the coastline. And we have a figure here that shows, fun fact, that juvenile white sharks, let me see if I can play the video for you. Yes, that juvenile white sharks within their first year of traveling, they can travel up to 3,000 miles. So we had a shark that was tagged in, I think, 2021 inside Monterey Bay, and it traveled all the way from Monterey Bay all the way down into the Gulf of California, or yeah, Baja California. Moving onward. So basically we have 2 decades, almost 3 decades of historical data in Southern California, and we noticed that it pretty much became a consistent, fully functional nursery ground for juvenile white sharks. And there are 3 major key components that nursery needs for the juvenile white sharks to thrive in. So 1, you need warmth. So they hug the shoreline because it's warm body temperature, and they don't have the physiological capabilities to maintain their own body temperature. The other one is abundance of prey items. So we also figured out in Southern California that 10,000 people in SoCal alone get stung by stingrays. So people were worried about all of the juvenile white sharks possibly attacking people, when really, right when you step in ankle feet water, it's the stingrays that are causing all of these injuries. And the white sharks help minimize those stingray populations. And then also protection from predators. So also adult white sharks can eat baby white sharks, and then also getting bullied. As of recently, you guys have noticed orcas have been coming in and eating all of the sharks for their rich fatty liver. So the most important key component for juvenile white sharks aggregating in a nursery or forming a nursery habitat is temperature. Is a main predictor. So in previous studies, obviously, like I just shown in the animation before, temperature is a major driver for where they like to go and aggregate, essentially. So they can travel all the way from Monterey down to Southern California and down to Baja, Mexico. And white sharks are the only warm-blooded shark that we know of that pretty much use the nearshore environment as a nursery ground. And just like, do you guys know what obligated ram ventilation is? Anybody? Okay, cool. So that's why I'm here. So obligated ram ventilation means that white sharks have to constantly swim in order to breathe, to get oxygenated water to pass over their gills. So they have to constantly swim in order to breathe, but they also have to swim to constantly stay warm. So they have something called the Reef Myriable, or the wonderful knit, to where it's a network of vessels to where all of the inner muscles, where's the pointer? Yes, so all of the inner muscles generate the internal body heat, and then that warm blood circulates to the outer regions of the body, and it can reach up to 0.4, 0.5 degrees Celsius, warming their entire body. So they have to constantly swim in order to breathe and also to stay warm. And how do we track all of this behavior of juvenile white sharks along the coastline? So there's a myriad of ways that we can do it, but there's two main reasons. So we use satellite tags, which I'm pretty sure that you guys are familiar with for like Shark Week and stuff like that. But we have another non-invasive way called acoustic telemetry equipment. So acoustic meaning sound, and sound travels 3 times faster underwater and 3 times further underwater. And it's more of a non-invasive way of tracking them. And the way that it works is we have acoustic tags that can either be surgically implanted into the sharks or externally implanted like this spot or this smart tag here. And cool thing is when we use radio tags like smart tags, spot tags, or pat tags, whenever that radio antenna pops up above the water, sends all of the information about the shark and the location to the satellite, and we get an email of what that shark is doing. Same with the smart tags where it has a magnesium clip and it's more of short-term tracking, to where that magnesium clip dissolves in salt water in 48 hours and it pops up to the surface. And the cool thing about it is it has a camera attached to it so we can see what the shark sees, if it's eating, if it's resting, so on and so forth. And then we also have these acoustic receivers and acoustic transmitters to where whenever this acoustic tag swims within 200 yards or so of this receiver, that sends a ping or an acoustic ping that gives us the ID of the shark, whatever sensor we have on it, depth sensor, temperature sensor, accelerometer, so we can see their movement behaviors. So, cool fun fact. So we have smart tags that we can deploy on sharks and they last again for about 24 to 48 hours. And it has an accelerometer, it has a pressure sensor and a temperature sensor. And this is a shark that we tagged in Belmont Shore in Long Beach. So we got some cool data from it. And what we can see is that whenever sharks were performing the circling behavior, their body temperature kind of lowered and then their speed slowed down. So what we're thinking is happening, what we think are happening is happening, yes, is that they are performing something that birds typically do. So long flight migratory birds have something called a unihemispherical slow wave sleep. So they're able to fly for long periods of time and they shut off half their brain in order to rest. So we're thinking, we're not sure if this is guaranteed, that they may be doing this at the surface on these circular patterns. So they will circle for about 2 to 3 hours and then they'll shut off the other side of their brain and then they'll go in the other direction. And some people may think, oh, the shark is circling me, you know, it may be wanting to eat me. No, sometimes they're just at the surface resting, possibly. Yeah, possibly. So let's talk about my project. So what I'm going to do is, since we have all of this historical data in Southern California, this is the acoustic tagging equipment that I plan to use here in Monterey Bay. So we have acoustic tags and then acoustic receivers, and then this is what it will look like when we are tagging all of the sharks. So it's just a quick dart tag, just like an ear piercing, and sharks have thick skin, almost 3/4 of thickness. And then the study site would be right in our backyard at Sea Cliff State Beach. So I just had a meeting with them yesterday, to where this is known as like a very well-known hotspot. It's a well-dominated area in Monterey Bay, but it's known in this area that the water temperature here can get up to 10 degrees higher as opposed to the rest of the bay. And seems like a safe haven seasonally where the juveniles like to rest. So just west of this shipwreck, we're gonna have all of these acoustic receivers set up in a grid. And the cool thing about the acoustic telemetry equipment is that whenever the sharks with the acoustic tag swim within this array, it's able to triangulate their exact geopositioning and the depth in the water column. So we can see where they're moving and we can see what warm or cold pockets of water that they're either trying to avoid or pursue. So what can this VPS data look like? We can get some cool 3-dimensional images of real-time movements of the sharks moving. So majority of the time, the sharks are moving at the surface, but also you can see they're also trying to go towards those warm pockets of water. So higher in temperature, the lighter the color, and then lower the temperature, the darker the color. You can see they're hugging within the shoreline, but whenever there's this warm spot, they're moving towards it constantly. And then the cool thing about this data set is this is all data historically for about 4 or 5 years collected in Southern California. And I will be conducting a similar somewhat experiment here at Santa Cruz or Sea Cliff State Beach and comparing it to figure out to where we can compare vertical, horizontal movement temperature and residency time to try to come up with a predictive model to see, is Monterey Bay gonna serve as a functional annual nursery habitat as opposed to just a seasonal habitat or nursery habitat? So the whole point of it is, juvenile white sharks are starting to become the new normal here in Monterey Bay. So the ocean is their home, and we kind of have to figure out how can we deal with them once we're in the water and not potentially harm them, because they are not here to harm us. And as some of you may know, we are in the midst of a El Nino event. So we are predicting that it's going to be a very sharky summer. I've already talked with my team at the Cal State Long Beach Shark Lab to where they already started to see juvenile white sharks in like the end of February, which is like 2 months ahead of schedule. And then obviously with this big VPS array, we're able to understand their aggregation. So we can see if Seacliff State Beach is going to be a new aggregation site, not seasonally, but annually. And then will Monterey Bay, I just said that, amazing. And then, yes, and I also just met with the state lifeguards as well from Santa Cruz. So what we're trying to do in the Ocean Predator Ecology Lab is once we collect all of this data, we plan on doing some education and outreach event. So not just in Santa Cruz, but also all within Monterey Bay, so that we can share this data with you. We can tell you whether, it's going to be a sharky summer, sharky season, what to do when you approach a shark, when you see a shark, so on and so forth. And then with that, I would like to thank all of my partners for this event. And my project, specifically Ocean Tracking Network, where I applied for a research loan and they gave me all of the research equipment that was worth $55,000 for free. Amazing. And then the Coral Mile Foundation, which is a nonprofit organization in Argentina where they are studying pumas, ocelots, and jaguars in Argentina. And with that, I will take any questions. Yeah.
So we hear about some like shark bites right offshore, for example, off of like Lover's Point.
Yeah. Recently,
Would those also be juvenile white sharks respectively?
So there's a live buoy system that Barbara Block's lab from Stanford or Marine Hopkins runs, and they get live updates on their website. You can literally search it up like Hopkins shark tracking organization or whatever, and it shows whenever sharks swim within that area, but they mainly focus on adult white sharks. And over the past few years, actually, it's been mostly like sub-adults that have been swimming around in that area. So like 15, 14 and up that have been swimming in that area. And I don't know why, because we have some data, actually temperature data, that shows that, let me go back. Let me see if I can go back. I don't have a micro or a macro scale image, but in Santa Cruz, this is like the warmest pocket of Monterey Bay. And then since there's like an eddy due to the California current, Marina kind of serves as like the secondary hotspot kind of. So we see like white sharks, sometimes sub-adults, sometimes juveniles as well. But Lover's Point is really cold, and the juveniles, they are not able to withstand really the colder water because they're limited physiologically. Yeah. Yes.
I had a question about the tracker that's on the shark, and if it emits a ping
Yes.
regularly.
Yes.
Is the shark aware of that ping? Is it picking it up with its auditory system as well?
So actually, scientifically, we don't know if they know that, but it's operating at 69 kilohertz, so super very low frequency. So we don't think that they do.
Yeah.
But probably not. Yeah. Yes.
Where's the nursery in Southern California?
Excuse me?
Is there a nursery in Southern California?
There's multiple nurseries sites. So we have, I think on record right now, tracking is about 14 different nursery sites. So all the way from Santa Barbara down to the Coronado Islands in San Diego. Yeah. Irina?
What similarities do you think there might be between Southern California and Santa?
Yeah, so the most prominent nursery habitat right now is Santa Barbara at Padero Beach. And that seems to be the peak to where they're there all year long, as opposed to like Torrey Pines, they may show up like last summer when we went out to go tag. There were no sharks at all, and that used to be an amazing spot for them to show up and aggregate. But there's differences in like turbidity. So there's a paper that the Shark Lab and Jack Elsner from Scripps, where he studies the nursery habitat there in Torrey Pines. There's differences in turbidity and there's differences in prey abundance, but they use the ecosystem there differently at different parts of the day. So like they'll show up and hover around the surface during the day at Torrey Pines, but they'll hang at depth in Padero Beach and they have similar turbidity. It's weird. We really can't predict where the juvenile sharks are going to show up. So that's like the whole point of this project, to where we can compare vertical, horizontal movement, residency time in relation to temperature. So we can kind of make predictive models and do a better job of trying to figure out where they'll be and how long they'll stay there. Yeah. Yep.
When the tag pops up to the surface,
You're talking about,
yeah. What's sort of the response time for the research lab, whether it be Long Beach or you guys? Like, if you get that message, do you guys go immediately that day if you can, or do you go the next day when the conditions are better, or like, how long can that tag be in the ocean for?
okay, so we had different situations. So like the pop-up archival tag, for example, we can program that to an exact day of when we want it to pop off.
Oh,
So like, say for instance, like, this shark swims a little bit offshore, like for Catalina Island, for example, and the current pretty much takes it further offshore. If we are still getting like, I guess, updates from that tag and it's just too far offshore, we won't go get it because we don't want to risk gas money pretty much into our safety. But as opposed to like the smart tag here, it only operates for 24 to 48 hours. And then whenever we tag the shark with that smart tag, it was typically within like the distance of the shoreline to where we can easily go capture it. Yeah. So it just depends. Any other questions? Cool beans. All right, thank you guys for showing up.
So that's gonna find house, maybe it feels like progress out now. You're gonna.
Of course. Not chill. So many people are just here to have fun. Yeah. Benson. You need speaker notes? No. Good luck.
Are you there?
Yeah, on the monitor.
Oh, I just.
Oh, I had it one too.
Yes, I know.
There was one.
Growing up in the fifties, I mentioned this.
That's where I'm scared of. We literally cannot. It's not recorded. There's been some times like moving houses. We don't know. Yeah, Arabs, everyone getting hurt, this juveniles are just caught out all.
As they both take what about this, what is trying to. I guess, but that's what we need.
Yeah.
Oh, yeah, like our last. Oh.
Are you ready? Okay. Hi, everyone. Thank you for joining us or for continuing to join us. I'm gonna start with our next presentation. We got it right here, and I'll just let it happen. And afterwards, if you have any questions, feel free to ask. Thank you.
Hi, guys. Thank you everyone for coming out today. Today is my favorite holiday of the year. So thank you all for coming. My name is Sarah Johnson. I'm a third-year grad student here in the Invertebrate Ecology Lab. And today I'm going to be talking to you about tide pooling around Monterey Bay. So first we're going to go over kind of when are the best times to go, where are the best places to go, And that'll get into some gear you might need, and then some safety precautions, and some best practices, basically how to treat our intertidal ecosystems with respect. And then finally, the fun part, we'll go over some of the critters that you can find out there. So first, when is the best time to go? And the simple answer is during low tide. So low tide means that the water line is lower than the average. And so that's gonna be when all those tide pools form. So when the water recedes, water is retained between rocks usually, and that's what creates the tide pools. And there are tons of animals that are specifically adapted to surviving in those tide pools. And there's a huge diversity on our coast specifically. So around Monterey Bay, there's tons of great places to go. So you want to go during low tide. And so the best way to figure out when that's going to be is to refer to a tide chart. And so I have two examples of tide charts here on the screen. So this bottom one, this wavy plot, is kind of more of a visual representation of the tides. And so on the y-axis here is going to be the tide height. And so the 0 is the average for the water line. So anything below that is going to be where you'll find the better tide pooling. As a rule of thumb, I think that a -1 foot tide, so anything -1 or lower is going to be the best times to go. That's when the water is going to be further out and there are going to be more of those tide pools exposed. So looking at this, we can see the pattern of the tides in Monterey Bay. So this is from the NOAA website. So this is pretty reliable. And I just pulled this one from yesterday through today. And you can see on here that Monterey Bay has what we call semi-diurnal tides, meaning that there are 2 high tides and 2 low tides pretty much every day, with a couple of exceptions. And so basically you double your chances of catching a good low tide on any given day. So you can see up here, there's a high tide at 4.43 feet, a little bit past 4 a.m. Yesterday morning. And then the low tide of the day was this -0.49 feet. And so that is a low tide, but probably not the best because it's not that -1 or lower. And then we also have another style of tide chart here on the right. And so this is showing the entire month of April. And so these first 2 columns are going to be the date and the day of the week. And then also lists those 2 high tides for the day along with the tide height that's predicted. And then this next section is those 2 low tides for every day. And then they also usually show when sunrise and sunset are going to be, and also the phase of the moon, because that will affect the strength of the high and low tides. So you can see during, or in the few days around this new moon, there were some really good low tides, like -1 and lower. Okay, I think I was on that slide anyway. So where are the best places to go? And like I said, we have tons of really good areas to go tide pooling around Monterey Bay. But you want to go to those rocky intertidal habitats because those are the areas that are going to form those tide pools. And they generally have more biodiversity than, say, a sandy beach, for example. So some of my favorite spots in the area are circled on the map in red. So there is Point Pinos up here. This spot is actually called the Great Tide Pool for a good reason. It's pretty great. And then there's also just south of that is Asilomar Beach. That's a really good spot as well. And then a little bit further out down south is Carmel Point. And Carmel Point is actually pretty famous for being an area where a lot of local nudibranch species were discovered and named. So that's pretty cool lore for those spots. But basically anywhere along these coastlines that are going to be rocky habitats are going to be pretty good tide pooling. So some gear you might consider bringing. I always wear my waterproof boots because it allows me to wade into the water where all those critters are going to be found. But if you don't care about getting your shoes wet, you can just wear an old pair of sneakers. And then sometimes we go tide pooling when it's dark outside, so you might consider bringing a flashlight. I personally like to bring a headlamp so I can have some hands-free light. You can also bring a field guide. There are tons of really good field guides for this area. Just anything like Central California is going to be great to use. But if you're concerned about getting the water damaged, then you can just bring a camera and then maybe check those field guides when you get home later in the day. And then finally, we want to consider like sun and weather protection. So if it's going to be sunny, bringing a hat, sunscreen, sunglasses. And then if it's going to be windy or rainy, bring a windbreaker or some waterproof pants. So next are some safety considerations. So you always want to make sure that you're checking the weather before you go out. Because if it's windy and rainy, you probably don't want to go out. That's going to create some unsafe conditions out there. And we always want to make sure that we keep our eyes on the water. Because even during low tide, some large waves can kind of sneak up on you pretty fast. And it's pretty easy to get knocked off your feet by those waves. So we always want to make sure we're not like too locked into the tide pool. We're always like lifting our heads and checking the water. And then some of the most common hazards are just slips, trips, and falls. So typically when we're out there tide pooling, you're crawling across rocks that are wet from the seawater and also usually have some algae growing on them that can get really slick. And so you want to make sure you're really watching where you step so you don't fall into a tide pool because that is not fun. And then finally, there are some critters that may poke, pinch, or sting you. Nothing here is going to be like too venomous or anything. So there's not any like real safety concerns, but if you're reaching under a rock or into a crevice or something that an urchin is hiding in, then you're going to poke your fingers. And if you're digging through kelp, there could be like some crabs under there that are going to pinch you, which I can tell you from personal experience is not fun. So I always want to make sure we're like watching where we're putting our hands and where we're stepping. So next is some best practices and how to interact with the wildlife as respectfully as possible. So the first big thing is to not disturb the ecosystem, right? It's that kind of leave no trace mentality. You can definitely go out there and flip some rocks over. There are a lot of cool things that live under rocks. But if you do that, make sure to put the rock back where you found it and always put it back down gently because you could risk swishing something that's underneath the rocks. And like I said, try not to step on live things. Some of these places have really dense patches of like anemones or hermit crabs or snails. And it's unfortunately pretty easy to step on them. So make sure we're watching where we step. Most things are okay to be touched, but make sure you do it gently and with fingers that are wet with seawater. Because that seawater will create a barrier between your skin oils and the animals that you're touching. And also really important is not to approach or harass any marine mammals that you might see out there. So we're really lucky to have a plethora of marine mammals around Monterey Bay, but they're actually protected under the Marine Mammal Protection Act. And so we are not allowed to approach them or harass them or engage in any behavior that's going to change their natural behavior. So it's great to observe them from a distance. And then finally, try to avoid picking up or removing any of the animals or the shells that you find. It can be really, really tempting to take shells home because they are really pretty. But those shells can create microhabitats for a number of different organisms. And so it's important to leave them out there so that they can have that habitat. And also the live animals, there are all important parts of the ecosystem. So we want to make sure that they're staying out there where they're needed. Okay, now we're going to get into the fun part. So there are tons of different animals that you can see out here. As an invert person, I focused mainly on the invertebrates, but there are some fish and mammals and birds that you can see that I have not included in these slides. But the first group is the sponges. So people often see these and they assume that they're plants or not living things, but they're actually animals. We have a couple little ones out in the courtyard in the touch pool actually. But these sponges can come in a variety of different shapes and colors. There's ones that can kind of bore into rocks. And then there's also some free living species as well. And then the next big group are the cnidarians. So these are the anemones, corals, hydroids, and jellyfish. So some of these hydroids in these two pictures here, they look like algae, but they're actually very small animals. And we also have a bunch of different species of anemones. So these are the two most common species of anemones that you'll see, especially these giant green anemones. These ones can get massive. If you are just walking around in the tide pools, you can see ones that are like this big and they're bright green and really easy to spot and a really cool find. And they're kind of everywhere. And then there's also some really small like soft coral species, like these tiny little orange cup corals. These things are like really small. Same with these strawberry anemones, which are one of my favorite species around here. So they're very small and very cute. So next we have the echinoderms. An echinoderm means spiny skin. So this group is the sea stars, urchins, and sea cucumbers. And we have lots of those around here. So we have various species of brittle stars and these bigger sea stars. These bat stars are definitely the most common species that you'll see around Monterey Bay. But you can also catch some ochre stars. I kind of rarely see the ochre stars around here, but the ones I have seen have been absolutely massive. So they're a pretty cool find. There's also these leather stars, also called the garlic star, because when you rub their skin and smell them, they smell like garlic. And then we also have a couple different species of urchins. These purple sea urchins are going to be the most common ones that you see. These guys are kind of everywhere, for better or worse. So you'll see a ton of them like boring into rocks sometimes, which is really cool. So if you walk past a big rock that has a bunch of little holes in it, if you look in those holes, there's probably some purple urchins in there. And we also have these red sea urchins. They're not quite as common as the purples, but they get a lot bigger, and they're always a really cool find. And we also have a couple species of sea cucumbers as well. So next is the mollusks. And so this group is one of the biggest groups of inverts. So there are so many different mollusks that you could find. I couldn't fit even a quarter of the ones that you can find on this slide. But just some representatives, we have a bunch of different species of snails and some limpets, particularly the giant keyhole limpet. These ones are really cool. This is actually one that I found when I went tide pooling at the Great Tide Pool a couple months ago. It was my first giant keyhole limpet that I saw out in the wild, and it's really cool. It was like this big. Yeah, so that's always a really cool find. These other kinds of snails are also cool, but they're kind of everywhere. And then we also do have a couple species of abalone. And these ones especially, these populations unfortunately aren't doing great. So these ones are important if you see them, admire them from a distance to kind of let them do their thing. We also luckily have some species of octopus that you could potentially find. These ones are really hard to spot because they're really smart and they're really good at camouflaging. In my last couple of years here, I've found one of these 2-spot octopuses, and it was the best tide pooling day of my life. It was a great find. But they often will hide like under rocks and will camouflage themselves because they don't really want to be messed with. And then we also have a bunch of different species of chitons. This species is my favorite. This is the gumboot chiton. Also sometimes called the football chiton, I believe. They get really big, like that big, and they almost look like a big meatloaf. So if you see a meatloaf out there, it's probably a gumboot chiton. And then also I decided to set aside a whole slide for the nudibranchs. These are also mollusks. These are sea slugs. And we are lucky enough to have a ton of different beautiful species of nudibranchs around Monterey Bay. Particularly these are two of my favorites. This is the Spanish shawl and the Hopkins rose. This one was actually, I believe this was one of the ones that was discovered at Carmel Point, and it was named after the Hopkins Marine Station. And they eat pink coralline algae, and that's why their bodies are pink. But we have a bunch of different species as well. We have the Monterey Dorid, or the false sea lemon, and the true sea lemon, and these opalescent ones, and these cloud nudibranchs. I think this actually was one of the biggest cloud nudibranchs I've ever found. It was like this big, which usually nudibranchs are around that big. So that was a really cool find. And the sea lemons can get really big as well. And then we also have the arthropods. So arthropods too are a massive group of animals. It includes all of the isopods, which are like really small, like bug looking things. Some shrimps, like the skeleton shrimp here, which are also really tiny, but they're a cool find. And then we have a bunch of species of hermit crabs, and like rock and shore and kelp crabs, all kinds of things. But these guys can and will pinch you, so be careful about that. And like I said, there are tons more that I just don't have time or the slide space to show you. But I highly recommend grabbing a field guide and seeing everything that you can find. And happy tide pooling. And I have a couple minutes for questions, so if anyone has any questions. Yeah.
Is there a specific time of year you find it better to have more diversity kind of thing?
I think it's less seasonal and more just dependent on the like phases of the moon. Because the moon is really gonna affect the gravity. The gravity between the moon and the water is the driving force behind the tides. So it's more related to the moon. But you can find really good low tides throughout the whole year.
And high tides, the animals, stay within the territories or they move about and then when the tide goes out, they just say, oh, I'm here now.
Yeah, pretty much actually. So, there are animals that are adapted to living in different zones, of the intertidal. So there's some that can live higher up and are more resistant to like drying out. But there are a lot of species that have to stay in the pools, because they're more sensitive to being dried out. And so, yeah, when the water is, higher, it, creates a lot more connectivity between those habitats. So they can kind of swim around, but once the water goes out, if they are reliant on that water, then they have to kind of stay in their little pool that they found until the water comes back in. Yeah.
Particularly interested in trying to find some brains. It's a good reason that like the muscular, maybe not. But there are like muscular is driving space, it's runs those sides and
Yeah, I don't study nudibranchs specifically, so I can't say definitively if there's a good like time of day to find them. But from personal experience tide pooling, I think that I have found more like later in the evening or the night. And you also want to check where there's a lot of kelp, because they're herbivores. So they want to hang out by all the algae in the kelp, so they can have lots of food to eat. So if you want to risk kind of digging around in some kelp, that's a good place to find them. But be careful of being pinched by a crab. Yeah,
So for like starfish animals that live on rocks, how part of they around?
So typically they're gonna try and stay where they are for the most part, because a lot of intertidal animals have strategies for retaining water in their bodies. And so the sea stars specifically, when they're hanging onto rocks, that allows them to kind of keep the water in their bodies more. But if they lose some of that water, they can like kind of fall off the rock. But yeah, so that's also why it's important to not like move the animals that you see, because they could be trying to hang on to like keep the water in their bodies. Yeah.
Easier to find at night.
I believe so. I can't say for sure, but I think I have heard that they're often nocturnal. Well, thank you, everyone. Appreciate it.
All right, thank you everyone for joining us or for continuing to join us. Unfortunately, the talk that was scheduled at 1:40 is not gonna be able to happen, but we have an impromptu shark Q&A session with a couple of people that are gonna present, so stay tuned for that.
Yeah. Where's the mouse? Oh my god. Bruh. Bruh. Oh my god. Yeah, you do that.
That should be posted over already.
Yeah, I was just gonna use the mouse. We could just have like an animation up. I'll just throw this up for now. Or have some, I don't know, like something that's reading this behavior. Temperature works, because that's a lot of my stuff too. Just can briefly touch on what I do in March. I give it a minute, I guess.
All right, thank you for continuing to join us, or if you're joining us right now. As I just mentioned, that the talk that was previously scheduled at 1:40 is not able to be happening right now, but we do have a Q&A related on sharks. So here you go, ask questions.
All right, good afternoon, everybody. Quick coverage here. Unfortunately, due to some weird issues with permitting and logistics, I wasn't able to give my original talk I scheduled for here. So as a replacement, I figured people always have shark questions, and I'm happy to just answer questions about sharks in as much detail as people want for the next 20 minutes. I'm sure there will be some. Anthony is also in the lab. I am in the Ocean Predator Ecology Lab at CSUMB. So my expertise is adult white sharks and physiology and physiological ecology with the sharks. Anthony studies the juvenile sharks and more of the behavioral ecology movement side of things. So between the two of us, we kind of got white sharks covered, and we know a little bit about other sharks in the bay as well. So I'm happy to answer questions about sharks, especially white sharks in the bay, other shark species, how we study sharks, anything of the like. So I will open the floor and see what happens. Yes.
Okay, so let's have some fun here.
Sure.
So what's the biggest white shark you've ever seen in Monterey? Yourself?
Inside the bay, I think I've seen a shark larger than 13 feet. That is a very small shark for a white shark.
What's the biggest white shark you've ever seen?
Outside of the bay at Año Nuevo State Park, about a quarter mile offshore, I've seen a 17.5 footer. That is a full grown female at that rate.
I think that she weighed.
It was pretty late in the season, so she had fed pretty heavily, built up a lot of mass to be able to migrate. I'd put money on somewhere in the 3,000 to 4,000 pound range. They get. I did not. My boat was plenty big. When they're at Año Nuevo, they're pretty docile. They're not very aggressive. They don't tend to come after things. In research, again, this is outside of Monterey Bay, where we have a very different set of permits and a very different set of methods for studying the sharks. Inside the bay, the sharks are the little guys. They're not aggressive. They're very calm. They're just here to feed and hang out in warm water and grow slowly. The ones at Año Nuevo are there to hunt elephant seals. They're there to build up mass, and they're feeding all the time. It's a very different environment, but they are very safe up there. Sorry, I have to be, as a shark scientist, I have to counteract the anti-shark propaganda and roll up the party line, right? So, answer the question?
Yes, thank you very much.
Yes.
Is this unusual that they're warm inside?
Yeah, so I pulled this up because this is a big component of my thesis. I don't want to get too much into my thesis right now, but I'm studying how their stomachs digest and metabolize food. And so the landed sharks, the clade of sharks that includes the white sharks, salmon sharks, makos, and threshers, primarily, are warm-blooded animals. They are mesothermic, which is a term that means that they are intermittently warm-blooded. So adult white sharks at their full size are warm-blooded animals. They maintain a constant body temperature between 25 and 30 Celsius. Juvenile white sharks will warm up when they are more active and when they're in warmer waters. It's a combination of all the same ways that every other warm-blooded animal makes heat. It's muscle expenditure, right? When they swim, their muscles make heat and they retain that heat. Their digestive system makes heat as things break down inside their stomach and as their intestines move material around. And they naturally produce a certain amount of heat just from bodily tissues. And this is, in most fish, this happens, but it is slow and it gets just passed out into the water without changing the temperature of the animal. In sharks, they intentionally retain this heat to make their metabolic systems more efficient. So their eyes work better, they get a little smarter, and they can swim faster and longer when they're retaining this heat.
And so can they change it? Like if they don't run out of food or something?
It does increase in temperature when they eat more. After a feeding event, we see a temperature spike. They go from about 20 Celsius inside the animal to about 30. And that's usually in 10, 15 Celsius water, right? So for Fahrenheit, that's 50-degree water and they're seeing 65 to 75-degree shark. So it's a lot of heat. And keep in mind, this is like a 2,000-pound animal, so that's a lot of energy. We do see that the temperature increases when they eat. We don't know if they're increasing the temperature to digest their food better or if eating the food is causing them to produce more heat.
Inside of the shark.
Put a thermometer in there. I literally, historic experiments, we've, like, they would modify an oven probe and they would surgically implant it into the shark. They put that was a 80s, 90s era experiment. They would capture a shark, surgically implant the temperature logger into it, and then have it broadcast a signal to a nearby instrument. In modern times, we just have various devices we can place on the shark to measure temperature or in the shark to measure temperature. Depends on what experiment, what technology we're using. We have different ways of doing this.
With the orca populations increasing at the moment, are white shark populations moving? How is that affecting data collection, if it is at all?
It varies based on the area. Both orcas and white sharks are globally ranging species. So what's true in Monterey Bay may not be true in Southern California or in New England, for example, right? We know that in South Africa, there's a lot of pressure on the white shark population. We do see some occasional predation events around here where the white sharks are eaten by the orcas, but it's fairly rare. Only certain groups of orcas go after white sharks. And the white shark population around here seems fairly safe. But we also see that white sharks, for some reason that we don't fully understand yet, leave the bay every time the orcas show up. They know the orcas are coming and they will leave in preparation for that. And they're very good at avoiding orcas. So not a ton of change, but it remains to be seen. We need more time watching this to actually observe any trends.
What's your favorite shark, or what's your favorite physiological bath?
Sure. Had to happen at some point, right? Classic question. I'm a white shark researcher, so I'm contractually obliged to say that white sharks are my favorite. I've done some work with Hawaiian tiger sharks, and they are probably my favorite. They're just beautiful animals. Just something about them. Shark physiology is the temperature thing. It has to be my favorite. It's just the weirdest thing. It's a completely independently evolved temperature process, right? No other warm-blooded animals descend from warm-blooded sharks. And there's a lot of long-term implications for evolution research from that, but we just don't know enough about it right now to really explain why and how these processes happen. So I could go on this for a while. I'm doing my entire thesis on it. I'm on year 3, but I'll let some other push on to some other questions for a minute and then maybe circle back to that if we have time. Sorry, I saw your hand first.
Yeah, what are the top 3 shark species in Monterey Bay?
Well,
in order.
Well, it depends on the time of year. In the summer, when we have a lot of juveniles here, like Anthony was talking about, it's going to be white sharks. We have by mass, probably at least. You know, there's the most of them. They're the most active. By population, I would say leopard sharks are the most common. There's just a lot of leopard sharks. They're little guys, and there's a lot of them in Elkhorn Slough and just in the local rocky reefs and kelp forests. But I could not tell you off the top of my head how many there are, or give you an estimate of that. I would say that number 3 is probably blue sharks. We see a lot of blue sharks in the bay. I know that their numbers have declined in recent years. There's a lot less sightings of them, at least. But there are a lot of shark species in the bay, and it would be difficult to tell you just like this is the most important shark species in the bay because they all do different things, I'd say. And there's a lot of them out there.
How do the physiological mechanisms for mesothermy, how comparable are they to tuna, who also have that capacity?
I am not enough of an expert on tuna to tell you for certain. There are people in the area who have made that their life's work, and I don't want to speak too much and get a corrected email.
Wasn't sure.
but
Lives there, very
Yes.
dense, of course.
from what I understand, please don't quote me on this. Most internal heat in a tuna comes from the action of their red muscles, right? So the same as any other fast moving animal, they have a lot of twitch muscles, these dense red muscle fibers that let them move very quickly in bursts. White sharks, they're a little flabbier. Their muscles are less dense and they're more adapted to long-term constant motion with migration. So we think that more of the heat in the white shark comes out of the digestive system and the other like visceral tissues, their stomach, their heart. But this is kind of an open area of research. And our understanding is changing with the white sharks. There was a lot of historic consensus that it was just red muscle. And then we started looking at the math on that and kind of doubting that, and we will see. Yeah. The shark in Jaws was supposed to be a white shark. It was a little bit too big. I think that in the movie, they say that shark is like 25 or 30 feet, which is much bigger than white sharks actually get. And it wasn't behaving like a white shark. White sharks don't do that. But I think that they modeled it off of a white shark and they intended it for it to be like a freakishly large white shark. And of course, they called it a great white. We don't call them great whites anymore because there's only one white shark. So it can't be the great white shark if it's the only white shark.
You know which shark it is for literally the logo of the movie?
Was it a tiger? I don't actually.
A mako shark.
Was it? That makes sense. That's hilarious. Yeah. It's been a while since I've seen Jaws. Sorry, guys. Yeah. Well, right now it doesn't feel like I'm ever gonna finish, so. No, in all seriousness, we will see, right? That's the scientific world is undergoing some changes. And the job search can be challenging. In an ideal world, I'd like to go into a PhD program and do more of the same of this research, but we will see. Yeah. I saw his interest.
What does the Elton seal predation with the white sharks look like? Like, how are they hunting them? What areas are they like eating them in?
Good question. So if you've been to Año Nuevo State Park and you've seen the elephant seals, you know they're on the beaches, usually during haul-out seasons. So there's just large collections of these seals on the beaches. The seals will swim for open water, kind of running a gauntlet. And there'll be white sharks at the bottom out there, and they will be looking up, waiting for seals to swim overhead. They will see a seal, they will bite it in kind of the rear area near the tail to paralyze it, and then they can eat it once it's let out. It's kind of a grim process, but it's not very dramatic. It's very surgical and very quiet for what you'd expect from like classic Shark Week stuff, right? The sharks around here don't do a lot of the classic like jump out of the water and eat the seal stuff. That happens more in like South Africa. We have seen them breach there. We have seen them do some dramatic stuff, but for the most part, it's just kind of a minute of splashing and it's all over.
Say a little bit about the juvenile white shark populations, like their behaviors and what they typically do here, how often they eat.
So it depends on the region. The juveniles have been showing up here more recently, since the heat wave in 2014, but they were pretty much more sparse, but now we're starting to see larger groups. And then comparing them to Southern California, it's been a historical nursery around multiple sites around Southern California. Like almost 3 decades. And since that heat wave, the thermal barrier used to be Point Conception, so just a few miles north of Santa Barbara. And that heat wave kind of weakened that thermal layer, so the sun was starting to scatter up and center. And typically what we know from the behavior from the juvenile white sharks in Southern California is that they hug the shoreline mostly because of 3 reasons. Either it's safety, it's temperature, because it's very little shallow, it's going to be warmer, and then also prey islands. So like the 3 top prey islands were stingrays. People in Southern California, 10,000 people basically standby stingrays alone per year. Because of the stingrays and the juvenile white sharks that were in the near shore habitat, helped minimize those incidences because they were eating the stingrays. Or at least Steve Irwin. But also they would eat things like perches, corvinas, produce, stuff like that. And I'm not sure if there's like much on the dietary research. You know, how white sharks eat hospital data.
We're still working on that. We know they scavenge more. So, we have more dead mammals around here, more dead sea lions, seals, whales, stuff like that than they tend to in Southern California. And so we see the juveniles will go to like, say, a dead whale in the middle of the day and they will tear into it a little bit and, get that nice tasty blubber. But, we think it's similar.
It would pretty much feel like, corvinas.
Yeah.
But it would be mostly like proteus or produce.
Yeah. Fatty, oily fish is what they want. Yeah. All right, we are running low on time. I think I have time for maybe one more question. Yeah. So you're asking when the sharks flip over, they kind of shut down and stuff? Yeah, so that's a process called tonic immobility. It does happen to all sharks. We don't know why. I know it's a boring answer, but that's basically it. Is we know very little about what causes tonic immobility. And we don't know much about it as a process, but I can tell you that you can take a full-grown white shark and you can rotate it upside down and it will be stunned until it gets righted. It's really weird. But we're still working on explaining that process. It's maybe something to do with blood pressure, but we're not quite sure.
Will it die? Could it die?
Yes, if it flipped over and it stops moving forward and it needs enough water flow and it's not getting it, it can suffocate.
Yeah, another question.
All right, one more question. Yes.
We're not sure why it's paralyzed by something down.
Not really, no. Are we out of time? Do we have our next person?
Yeah, 2 minutes.
2 minutes. That's one more question.
That tonic immobility, is that similar to what other animals go through, like rabbits? They go through something similar?
I think it's named the same and it may look similar, but because we don't know quite what drives it, we can't tell you if it is the same thing, right? Things can look very similar between animals, but be caused by completely different things. It's fairly common in nature, and that could be the case with sharks. We just don't know enough to really say for certain. Alrighty, you guys have been a great audience. This has been a lot of fun. I will be around, and if people have more questions, you can come and find me, and I will be happy to answer more shark questions. In the meantime, I think that I'm about to get booted off the stage. The shepherd's hook will be coming out soon. So, alrighty, and I will be passing it off to Ivano, Dr. Aiello, to talk about some rocks.
Yes, thank you everyone for that. Unscheduled kind of interruption for the QA, but I'm so glad that you all enjoyed it. And so now get ready for the next talk that is now.
Oh yeah, you got it. All right, get out of it. So, yeah.
Oh, okay. All right. Hello, everyone. Got a jacket. I'm a professor. And I don't know how to do. Should I change? Technical support.
No,
Hey, does anyone know here how this thing works? Yo. My actual presentation, I have no idea what it is.
Is it on, is it Google Slides?
No.
PowerPoint?
She tried, yeah, PowerPoint.
PowerPoint.
Oh, it's already loaded.
Yeah, but there's like multiple, so let's see. Which one is
Sure, I have my first one.
right here?
Oh,
Amazing.
that's beautiful.
Yeah, yes, it is.
Yeah, that's it. I know this one.
Yeah. All right, oh, yeah.
Cool. Okay, well, first of all, my name is Ivan Aiello. Thank you all for coming to our open house. And a geological oceanographer here. And I get to teach and do research with wonderful graduate students. And today I'm going to show you some brand new data, well, for the last century, so they're not that brand new, but a new synthesis of data from the head of the Monterey Canyon. So how many of you know that there's a canyon right offshore? The majority, so you're familiar with that. And so I'm going to show you some older and recent results. Actually, you can see in the picture, this is us going out there with a little survey boat, big waves, small boat. We did okay though. And so here's a map of the Monterey Bay. We are at Moss Landing right there. And as you know, there's a huge feature out there on the seafloor, which is called the Monterey Canyon. And one of the uniqueness about the canyon, not only is the largest canyon in California, but submarine canyon, but also the head of the canyon arrives all the way to the shoreline. So if you're a diver, you just swim a few minutes and it just drops several tens of meters. And the end of my talk, which at the end of my talk, I'll show you some new imagery, bathymetry from the canyon using one of our research vessels. But that's what you will see if you were to drain the water out there. And that's the Moss Landing shoreline there. And you look out, you'll see that the seafloor just drops off really quickly. And the purple, dark purple, it's actually pretty damn deep out there, like 50, 60 meters, or I don't know in feet, I have no idea, but it's probably 120, 150 feet, okay? So how do we know that? How we got there and what's happening out there? I'll always love to show this map. This is the first map of the Monterey Canyon by George Davison at the end of the 19th century, which was done with lead lines. And we'll talk about lead lines, which was a pretty cool way to measure the bottom of the seafloor. But it was clear that not just here in Monterey, but also along the California coast, there were several features that were very deep right out there, which were called submerged valleys. And they were really important because they were also important for fisheries and such. So, but the early exploration is a very, of the seafloors is a very exciting and pioneer work. And much of it was done with, as I said, with lead lines. So literally a guy with a long rope and a weight at the bottom and just dropping it and then measuring how far the rope went. So that was a way to do bathymetry back then. And, one of the major steamboats that collected information along the California coast, there's a Lidonia there that ended up in the Second World War as a support vessel. There were also other methods, which was the wire drag operation. There were two boats dragging a rope and literally looking at how the length of the rope was changing as they were dragging it as a proxy for the roughness of the seafloor. So everything was done manually, and the positioning, so to know where you were in the ocean, was done with sextants and triangulation. So all optical measurement. I mean, incredible skills. And one of the data set I was able to work on actually the earlier data set come from the early 1930s, when the surveyor vessel is a steamboat vessel mapped just offshore the head of the Moss Landing Harbor. So those are single measurement with lead lines. And after a lot of magic, and to bring it back to the more, and what you see in the background is the modern head of the canyon. I mean, it's a picture from the modern head of the canyon. When you look at the old bathymetry and you map it out, it's really interesting to see how the offshore area of the jetties here was much shallower than it is today. You could actually walk on it in some places. It was almost like a beach out there. That's only 100 years ago. So that got me really curious. And to put this in perspective, back then, this is actually Google Earth. It's such a cool tool. You can go back on time there. This is a picture from the same area. Moss Landing is down there. And you can see the mouth of Alcan Slough here. And then that's about the Salinas River. So the Salinas River used to empty north of Moss Landing, and then eventually was diverted in 1906 to the modern actual position. So there was a river that was dumping sediments to the ocean right there, that then eventually was cut off. So that's one thing that happened back then. And the other thing that happened in 1946 was the opening of the jetties to build out the Moss Landing Harbor. So these two major events probably had something to do with how the canyon responded to human-made change. So now, as I said, the earlier survey were done with the light lines dropping away and the rope. Then we went from ropes to sound. So one way to measure the depth of the seafloor is to bounce sound off from the seafloor and measure how long it takes to bounce back. Sonar. The first type of sonar was single beam, one beam, very much like one point at a time. And now we got to use the multiple beams, multi-beams, which really literally you sweep to the seafloor and you can create really high resolution maps. Plus we have GPS technology, which is amazing. We can find our position within 2 centimeters at any given time. So, from sextants and triangulation, we're very lucky. I'd like to acknowledge our friends and colleagues at CSUMB because for more than 10 years, they built a program of seafloor mapping, which was an incredibly successful program, also educationally speaking. And they had different, and especially Rick Vitek, you see they're driving the Kelp Fly. It was a really cool guy, cool technology. And what they did, they mapped the head of the canyon for about 10 years. And they produced really beautiful maps like the one you see here, that's from 2002. But I'd like to point your attention to the box out there because that's the very head of the canyon I'd like to talk about. And so this is one of the earlier maps. And as you can see, there's a lot of features. So the shoreline is out here. Those are the jetties up there. And you see there are a lot of features, but you can recognize some major features. Some of them are the channel, the main channel of the canyon. That is branched out, finger-like projections. So that's kind of like a river. And then there are like higher parts and slopes. So different geomorphologic features. And you have a time series. So the question is, did it change over time? I put together a little movie for you. And the first slide here shows this is the first survey they did in 2002. And I outlined the edges of the canyon here with red lines, okay? So let's see if this thing works. So I'm gonna play. So we start with 2002. Just watch what's happening here every year. It's pretty cool, huh? So you see something is happening to the canyon. It's getting larger. It's getting larger and is eroding mostly on the northern side. I mean, this is like up to 100 meters in displacement and also 10 meters in drop off. It's getting larger. The sand keeps kind of moving in channel, but the slopes are also getting shorter and more eroded. And right, it's a really an amazing data set here. So, and that's to show you the position of the edges of the canyon in 2002 versus 2014. You can see a jump. I mean, I did that in PowerPoint, really nothing fancy, just to show you how obvious it is. Actually, if you look at this data and you put them all together, and you play a game like, when you take photographs looking at for a long time and people are moving and they're kind of fuzzy. That's what this show here. The warmer color show places that have been moving over time, while the gray colors are more like stable places. And you can clearly see how the channels, much of the warm color are in the channels. That's where the sand is moving. But also there are parts of the slope, as you saw earlier, that are eroding. And also gullies, like, little trenches here that are also eroding. So we can look at all this quantitatively. And when you put all together, you can observe that almost 850,000 cubic meters of volume were lost in the canyon in those 10 years, between 2002 and 2014. And much of the erosion, really busy here, but much of the erosion was on the substrate, which is not the mobile sand that's coming from shore transport, but the actual rocks in the canyon. So that's permanent erosion. It's not going to come back. And that's 80%. And also the sand has been moving around. There's a negative budget, but it kind of comes and goes. But the canyon itself, the shape, the bowl is getting deeper and larger. Now we jump 10 years and we go to nowadays. This is our program. These are two of my students in my lab. One is Marcel Pilix, who actually we built together and we were supported by funding by San Jose State University to build a new mapping system that uses very high resolution. We're talking about 10 to 20 centimeters. So we can look at very tiny things on the seafloor. And it's a shallow water system operated from one of our whalers. And those are the kind of maps that we can produce now. I mean, you can see this is a pipe. Actually, this one takes. So we can see very, very tiny features out there. And of course, what we did, we compared the new imagery from 2024 with the older one from 2014, so 10 years. And when you put them together, our data set is from shallower parts of the canyon, so they're not completely overlapping. What you see is that the erosion that we saw earlier, so the red means for erosion, this is meters, -10 meters. It's mostly concentrated on the slopes that we saw earlier. Also, the sand kept coming and going with a net positive budget. In other words, the sand is something that varies in relation to storms and other events, but the substrate, the hard part is eroding. The canyon is eroding. And when you plot that in a very simple diagram like this is the data set from between 2002 and 2014, and you can see there's a net change in the substrate, both in the substrate and also in the sand river. When you go to this next decade between 2014 and 2024, the substrate kept eroding, although a bit slower. So the erosion continued, but slowed down somehow, and the net change in sand coverage was positive. So in summary, I'm going fast. That's amazing. I usually take like 12. Cool. In summary, so hidden, it's covered with water. We don't know what happened out there. We know what happened with Elkon Slough. Elkon Slough, since the opening of the jetties, was obliterated, essentially. It was covered by very shallow marshes, and now it's a pretty deep channel. There's a lot of restoration effort nowadays to bring it back to a more healthy habitat, but we visibly saw that, but nobody could see what happened in the canyon. So we know that once it was very shallow, you can almost walk on it because of the sand probably coming from the Salinas River, and then now it's much deeper, and it deepened rapidly. That happened in the last few decades, probably as a consequence of the opening of the jetties and the rerouting of the Salinas River channel, and the erosion is still happening, although it seems as though for the last 10 years it slowed down somewhat. And we need to keep monitoring. This is an essential part of the coastal system because we understand the canyon, we also understand the future of our beaches and what will happen in relation to sea level rise and other man-made changes. Okay, that's my presentation for today. Questions?
So I was just curious, do you think that the relocate, I'm just thinking of all the sediment transport that comes down the Salinas River. When the Salinas River outlet was relocated, do you think that had an effect?
Yeah, so the Salinas River was essentially emptying. I mean, again, Google Earth, it's so cool. I mean, it's such an amazing tool because it's very easy to access. And so this is a picture from 1931, right? And so the canyon head is here. This is the canyon head. I didn't put it here. I see. So the river was discharged. Much of the waves are coming from this direction, right? So much of the transport goes south. So probably it was getting constantly filled with sand. Yes. And sand does different things. One of the things it does, it covers or protects or shields the hard substrate from erosion. So if you subtract that cover, that might be a cause of increased erosion for the actual head of the canyon. And I mean, again, this is shallow. I mean, you're talking like 1 meter depth here. Right now it's like 10, 20 meters. So it was really, really shallow right there. Go on the side. Lady.
Yes, does the Monterey Canyon have several places where it comes from the coast? Because I was always told the Monastery Beach area, there is a place there, and when I was diving.
Monastery. Yeah, that's a different canyon. That's a Carmel Canyon. Yeah, so that's a Carmel Canyon that, that it's coming out here.
Right.
Yeah, so, but it's part of the same system.
It's part of the same system.
Yeah.
Thank you.
Yeah, of course, please.
Could you explain the process of upwelling and what drives it and how the changes in the canyon, the erosion might be affecting that?
Yeah, that's a great question, which my colleague Tom Connolly and I have been talking about. Yeah, so what is eroding? What is creating this erosion? So one hypothesis is internal waves, not directly upwelling. There's not much upwelling in the head of the canyon, but there are internal waves, so there's and also just wave refraction can be another factor, but I do not know the mechanism yet. I just observed the change. So, but yeah, that was a really good question.
On the East Cliff Drive, the cliffs are eroding because the waves have changed direction because of global warming. Is that clear, do you know?
So you'd say
Used to be, the waves always would go this way, now they're kind of tilted a little bit. Because they're blaming on global warming.
Yeah, I don't think there's two dots that you can connect directly. Obviously, the main direction from which waves come is dictated by the location of the low pressure area where the storms are generated. So if you're shifting everything southward, you might have a change in angle more, going from northwest to more like, westerly direction. So in future scenarios, but yeah.
What is substrate?
Sorry, I didn't complete it. What is this? Substrate, thank you. It's, so the substrate is the hard seafloor. So there are two types of seafloor essentially. There's the stuff you see on the beach, like beach sand, which is really mobile and soft. And that substrate is their hard rock. That's me, I need to stop talking. All right. Last question and then we go.
So with the increase in height of the water, are there any things that you predict over the years, either for the canyon or for the beaches, Moss Landing?
Yeah, I mean, yeah, they're gonna be gone. Yeah, rocket science.
So the canyon's gonna be.
Well, the whole shoreline will have to move, and the dunes will go. Yeah, sorry. Okay, I think it's time, all the time we have. Thank you so much. Puppet straight. Thank you.
Thank you very much. In regards to this erosion that's created, my name is Robert Fernandez and I own quite a bit of property here in Moss Landing on Moss Landing Road. And we're hearing anywhere from 30 to 40 years with the erosion and also with the rising sea level. Do you feel that is an accurate?
So one thing I was, I guess the good news is that I didn't see any headward in the signals that the position we have in this energy hasn't changed much. It's going deeper and the channel go on. But I don't see one thing that I really want to see to have is a definitely the total because we know that one of my colleagues we able to throw this very it's like that faction, so it's possible that having that trigger some analysis which might have you that has a beginning to but no, these are all simply mostly starting against. So yeah. Yeah, so I guess I really.
I think I don't.
So the Jackson Curtis.
Yeah.
And also the only one of the Here, most likely in house.
What an own.
John Smith.
No, what and more. I just like rushed out of my shop.
Last name is
Yes. No, we're gonna keep it on because we have some lyrics to put on.
Be here for. My I was here, yeah, I never, what are the signs? Yes. Oh, sure. I've no idea. I'm sure, yes.
Thank you.
I'll be fine.
Yeah, thank you. This is good.
Thanks, Ivana. Okay, okay, I just like one of
Yeah.
Yeah.
I told all the volunteers.
Okay.
Is this the last one? Yeah. Okay. I haven't seen it yet.
Can we turn this off? What do you think? Is this the Well, maybe they're gonna.
Like, I'm thinking.
It's in charge of the projector. It should be. So it's kind of connecting off.
Now.
It's a projector. Yeah. I'll maybe leave it on. Water. Yeah. Yeah.
Right. I never. So. Okay. Sorry about that. I know.
I mean, it's an.
Can you guys see okay if I'm standing here? Can you guys see okay?
Yeah.
Okay.
I have to, but I primarily. Yeah, it's just. Yeah. Yeah. Yeah.
Hi, everyone. Thank you so much for coming. I'm so excited to be here at MLL today. Before we begin, there's a few people I want to thank. I want to thank Dr. Amanda Khan for her encouragement in continuing this tradition. I want to thank the Quilt Guild for constructing some beautiful puppets for us to use. And finally, I want to thank James who helped us set up this stage. And without further ado, the puppet show.
Hi, everyone. I'm a scuba diver. Wow, it's so beautiful outside. What a great day to go explore the Monterey Bay. Are you ready to go exploring? Yeah. Awesome. Let's start in the intertidal zone. Who is that? The anemone. Can you say anemone? What is an anemone?
How it's going? I can stand the coastal habitats. This tide pool is my home.
This is such a cool place to live. What other animals live in the tide pools?
These tide pools are home to many amazing animals. We have urchin, mussels, barnacles, octopus, and sea stars. Here's one of them now. This is my good friend Asteroidia. Hey guys, are you enjoying your visit to the tide pools?
Yes, we are, but I almost stepped on an anemone.
Sometimes the critters in the tide pools can be hard to see. They can blend in with the surroundings using camouflage. That is why it is so important to be careful where you step.
I promise to be more careful about where I walk. I love the tide pools and I want to keep them safe. That is what I like to hear. I want to buy some tasty mussels for lunch, but I hope you enjoy your visit. Bye, Oscar.
We are so lucky to be able to see a small glimpse of ocean life these typhoons. We have to be careful to protect them. The best things to do are to watch where you step and leave animals where they are. It takes effort from all of us to keep this habitat the way it is.
Don't worry, I won't forget, and I will definitely be back for another visit. I can't wait to go diving in the beautiful kelp forest. Wow. Look, there's an abalone. Wow, look at all these fish. Oh my gosh. It's so nice how everyone works together to create this beautiful environment.
Is it just me or is it getting a little warm in here? Actually, it's making me pretty hungry. So hungry I could eat an entire kelp forest. Nom, 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 nom.
Make me feel a little sick.
Underwater, here we go again. If I survive you, underwater, she's coming in. That's fine, there's no other place to move to. Yeah, but we've been warm and hungry. Things like something thriving. Bye, we could never stay home. Underwater, here we go again. Bye, how can I survive you? Wait a second, am I in the right place? Where's all the kelp? Where did all the fish go?
Yes, that might have been my bad. I kinda ate all the kelp.
Oh no, why didn't anyone stop you?
The only thing I'm scared of are sunflower sea stars, and they couldn't handle the heat. With the sunflower stars gone, us urchins aren't afraid and we come out from our hiding.
Wow, I didn't know that a single species could make a big difference in making sure this kelp forest stays beautiful. Even the little guys like sunflower stars mattered in maintaining a healthy ecosystem.
Yeah, even the small guys matter. Big, small, squishy, crunchy. Oh no! Oh no! I see a sunflower star coming. Run away!
There's no place like home.
Okay, guys, now it's time to go a little deeper. Grandma Christius, I need to find my way to something called the mesopelagic zone, and I've heard you're quite the diver. Can you help me out?
Well, I'm glad you would ask. That's where all my favorite snacks are. Let me take a big breath. Let's go.
Oh, Grandma, you're pretty fast. Wait for me.
All right, Scuba. Some people call the mesopelagic zone the twilight zone, and that's because it gets so dark. I can use my echolocation to see where we're at. But you might need to use your flashlight.
Got it. Wow, Grandma Christius, it's really cold down here.
Sure is, but I've got thick blubber to keep me nice and warm. Other species swim to shallower areas during the night to stay warm. One sec. Squid.
Where did those guys come from?
Like I was saying, some species swim up to the surface at night. It's called vertical migration. Can you say vertical migration?
Why? Where's your sense of? At the end of the day.
So we have all this way to get ourselves out of sight. So we gotta slow down, it's all we've got, because it really matters if you make it or not. You see them from the open, they're playing on us. So what do we do? Is that the same on the golfing club there? So, the ladies. Is getting past you.
Sure isn't.
Grandma Christmas, I'm not sure I can go down much further.
Use the compartment dive this seat, so you can catch a lift on my submarine.
Whoa, nice ride.
I've got some squid calling my name.
Sweet, deep ocean, here I come. And I finally made it to the very bottom of the ocean. How does anyone see down here?
I can answer that.
Your eyes are, sorry.
What, big, enormous, gargantuan? Yeah, you wish you got orbs like these. Because my eyes are so big, I can see things in the dark that your puny eyeballs can't.
Cool. Do your eyes help you see on the surface too?
The surface? Why did anyone want to go up there? It's way too bright. I got all the light I ever need right here.
Lights? Down here?
Want to see something cool? My favorite place is the deep sea. There sure are some fancy lights down here. Look, here are some of my friends now.
Whoa, what makes you all glow like this?
It's a process called bioluminescence. Can you say bioluminescence?
That's right. Some fish use their lights to talk to their friends. And some, like me.
We like to use our lights for socializing. Disco never dies in the deep sea, my friend. I use my lights to party.
Wow, you guys sure do know how to party. You even have confetti.
That's not confetti. It's called marine snow. Tiny bits of plankton, food, and leftovers from the surface.
Oh. Dinner for us. This is delicious. Would you like some?
Look at the time. I think I'll just get the plankton poop for today.
Your loss.
Up to the So, what did you think?
I learned so much about our home in the Monterey Bay. I learned that every single species in the ocean is important, and I want to make sure I do my part to protect it. Thanks for the ride. I heard. Yeah, she's better than that Yes. Verified. Don't sit in the slide, and you say that. It's heard or like, is it a verified? Well, like if. That's not, I mean, if you want it. It's getting like. Yeah. Yeah, after I get my PSA submission back, I'm gonna create a specific ones in PSA, right? Specific in PSA. Gotcha. Yeah. What's up next? What's up next? It's actually. Yeah.
I was going to. Because I was. I have so many concerns here. Yes. But GameStop is. I get it. Is it like they here?
Yeah.
Yeah, wait, okay, they were literally just asking you want to do that. You guys just need on one paper, this can be a. You know, so I, yeah, really? You don't know. Okay, so it was literally. It was a little scared. Yeah. Got it.
I know. This is.
Yeah,
Sam Ziles. I think Zach.
Thanks for coming to the event. Yes, the most like.
You want to see very on the edge.
Got the giant cup.
I know there's songs like.
I know.
I want to. Yeah. I was like, I needed to, and I think there may be some that are like. Water. Oh, then I think those can be like 50 to. $100. Yeah, I wouldn't be surprised if there's subscription ones. Yeah, so, yeah, I was telling me to, it was like five. Yeah.
Yeah. Move the curtain.
Yeah. Yeah.
Yeah.
So a lot of these comments. Mine like that. Those like a line at the top. So it's different windows. Like you're information that's now. It's like. For sure, you like fire. You guys like now we don't. It's like. Not horrible. And like, it's very just like, very casual speaking, one word answer of a lot of the time, yeah, like, yeah, and about this. And like, I mean, when they close, like, it's about this, what do you want? Hey, really, good, it's not like, all right, it's all right, it's hopefully. Like, say something. It's like, it's like a wall. The idea is getting all of these kind of nights, very. Yeah, the emails are slowing down for like. Yeah, or they, we just type some typings too. Yeah, it's like it's just doing experimentation, but turning off grammar and punctuation. Yeah, that seems like it's just honest, but I'm sure I really people. I understand why I did the same, but I was all works, so you better get more, maybe a one now, you bought, I don't want. Oh, yeah, I'm sorry.
That is, well, if you're interested, you should stay for the talk. I work there, so I'm a part of the organization, but basically we're the Black Ocean Sample Scholars. Yeah, okay, so we'll talk a little bit about our programming, and some of the work we do, but if you're interested, you should attend the talk. Okay, if we have a booth down in Hemwood, it's kind of back down that way, I don't know if you guys made it on over that way.
Yeah.
With a bunch of like drawing and stuff on the tables. So-
Yeah.
Yeah, totally. So our talk is right before the student panel, as we're sort of transitioning away from like the hard science talks and more so into like, community involvement and outreach and what is it to be a student, right? So it'll be us and the student panel. There are master students here, they have a master's program, I'm not a student here, I'm also, but we have a lot of, years who are here. And I think it's a 4-year master's program, but they also offer.
Then, once you said he's down to come up here with us.
You'll come and present with us?
Yeah, you know, I don't know what to do.
Well, we're just to be nice on face.
It's not different than it used to be.
It's the same, what's that one?
How's it going? Good, nice.
Thank you, trying to survive. Yeah, the semester's pretty public.
Yeah.
I have,
Well, they didn't talk about the shark research project.
so, we say, the paper revisions are due in May. The other thing I'm doing is due in May too. So I'm just all over the place. I have a master's student now too.
Oh, wow. This is your student at Cincinnati.
San Jose.
That's sick, wow. Well, when we told people, we were like, yeah, we know a professor at San Jose, we were like, oh, yeah.
It's kind of worked out.
Yeah.
Apparently I'm the first like coastal marine ecosystems person.
Just like full stop.
Yeah.
Oh,
And he wanted to do that type of stuff, and so at first, the director was like, we don't have anybody, and then she was like, well.
It sounds like your story.
Oh, he's
We
gonna.
don't do that.
Yeah,
Oh,
exactly.
wait, maybe we do.
We do, yeah, exactly.
I don't know, is there a good tea person on it?
Things I have been recorded and from hygiene field with local birds. But other than that, love that. So he's trying to, wrapping up sequencing now. And it's just March is the plan, so now just a little level. So what happens.
Are you trying to stay here or is that? Are you trying to?
I would like to be in the same state as Matthew, but.
Makes sense.
Illinois or Georgia.
Hello?
Yeah, I was like, I couldn't tell.
Oh, it's over here.
Oh, okay, they're over there. It might be in the
Here.
folder.
Some people,
Maybe not.
society's expressed, maybe.
Oh, it's Olivia. Did you-
Yeah, I shared it with Olivia. So
Okay.
maybe share it with me.
Mm,
But I want to be careful what's it like.
You got options.
you shared with me.
Yeah, there it is.
Okay, perfect.
Yeah, perfect.
We got this.
Yeah, amazing. We did that. That was good.
Technology is not my forte. And then there's just a live stream.
Okay, so.
You'll be able to. Which one other side, so
Sounds
congrats. I'm where you were like,
Yeah, when we first met.
yeah, you're like, I don't even know what you.
Can you guys explain what BOS has to do?
I guess I'm not the president.
Exactly.
So basically, where information is, health information and Stanford focus on increasing access and equity along the ocean as a boss transfer of these blackfish and Stanford waters. So we do a lot about, each local high school, getting involved with stewardship of local, environmental resources, helps and rocky vitals, coasts. We're based here in the Monterey Bay area, so we're all PhD students, and then the Monte here is like. And then we'll just be doing a talk about sort of our organization, what we've been up to the past year, our future plans, and then introduce to what our stalls. Yeah, I feel like hopefully it's intriguing enough to get people to show. Yeah.
Yeah.
So we have some members based in on the main campus in Stanford and DePaul also. But we're all coastal ecologists, so we're all based out of and then we got in contact with people this year, not many since last year, we had a booth on the table. So that the prizes involved here, you can do another mass copies. So when we started doing it again this year, we're like, oh, well, we have a ton of stuff to show this year, so it's great to come back. And then September 12th will be an open house of podcasts. So we'll do that. Yeah, no, exactly, yes, we'll be there too. Sorry.
Why, you saw your email bounce the other day, the first time, they're like, Stanford email was not a good, I was like, oh my
They
god.
changed it as soon as the day hit, it was like, oh,
They were like,
you know,
Well, I heard from Jenny that they were strict about that. She said,
they got her too.
she said she couldn't even log back into her Sunday. She like her last name is like a Friday.
Yeah, well, that's the same thing happened with me, luckily.
Steve's just gone, maybe he's like.
I was like, that's crazy.
Well, he didn't have to worry about anything, he stopped paying for me way back in, March or whatever.
You at that point,
Yeah.
Julio.
Oh, I also, because I know we can rely on you, to maybe do some research.
In the sloop?
Yeah,
Totally. Yeah, you need some people to tag along on a boat or something.
Yeah, because I figured probably just to make it easy to see if there's a lot of someone like that feels the way.
Yeah. Yeah. Like in the sloop?
Yeah.
It's just me. Like at Hopkins.
Yeah.
What's that other group that you sort of work with, right?
Oh, but they're based in Santa Barbara. Yeah, so they commute here to go to Chamberlain.
Barton.
Oh, yeah, Barnabas.
They're doing field work in school now, they're just getting started too, so it'd be a good time to chat with them, you make sense for like how you guys answer. No,
Yeah, it's. We packed up all of our stuff, wheeled it all over here, set it up, and then no one ever.
Shout out to the person who has me.
Yeah, exactly, and I'm so disappointed.
Well, yeah.
It's also so wonderful that like mattered, yeah.
To be fair, you and Stephanie do it, but I would just like to, we have to get it.
There, I should probably also, I think you got a solution, you, especially if it's.
Yeah, well, I got, I changed our information over the there, especially for that.
Oh, yeah.
So I really love to get.
Are we?
Yes, that's also. It's just, this is like sharing.
There's a Zoom crowd.
Yeah.
Go webinar, live stream.
Yeah. So this is all.
The camera, yes. Get started.
Three. Yeah.
What's it? I'll go to the other side over here.
Make it happen. Maybe we can close the door.
We got secret loud.
Yeah.
I think that's fine.
That's smile. That's so funny.
I was there first. You want me to do the screen?
Is that okay?
Yeah, that's fine. As long as you tell me when to go.
Yeah, sure. It's pretty chill. Cool. All right, how's it going, guys? Hopefully you've enjoyed the Moss Landing Open House so far. It's been really busy, full of a lot of talks and booths. And so we're here to share a little bit about an organization that we're a part of called BOSS, the Black Ocean Stanford Scholars. We work at the Hopkins Marine Station in Monterey, right next to the aquarium. And so we do a lot of work around ocean equity, access, and belonging, specifically in the Monterey Bay area. So we take a lot of high schools out on tide pooling trips like here, trips to the Hopkins Marine Station, which is where we work, because, you know, in order to get future generations to feel responsible for the earth and to steward the earth with a lot of intention, they have to have a connection to place, right? And so we were really struck by a lot of our work in local high schools, teaching sort of science that people who grew up here, born and raised, within 15 minutes, 10 minutes to the beach, had never been to the beach, right? And so if we think about a future where climate solutions are going to be really important, we're going to want to make sure that everybody feels involved in those solutions, right? And so that's basically what we're here to talk about, is what we have done historically, what we currently do, and how to sort of get in contact with us. And so I am Ryan Rogers. I'm a third-year PhD student at the Marine Station. I work in Theo McKelly's lab. We'll talk a little bit about that later. And I'm the president, acting president of BOSS.
Yeah, hi guys, I'm Sienna. I'm a fifth-year PhD student in the same lab, Theo McKelly's lab at Stanford. I'm last year's president of BOSS, so I'm handing off the baton now, but really excited to be here. It's been so great to see all your faces come through. It's such a diverse audience here, which is really exciting. And we're talking about ocean equity and access, and people just being excited about our ocean and our ecosystem. I'll hand it over to our ex-vice president.
Hi, everybody. My name is Dr. Devontae Dawson. I was a postdoc at Hopkins. I currently am a lecturer at CSUMB, and I was recently hired as a tenure track professor at San Jose State in environmental studies. So I'll be starting there in the fall. So.
All right. Cool. You might have to. There we go. So we talked a little bit about our mission, right? Situating us here in the Y for things like equity work and access. And I'm not really just gonna read this off, but I will emphasize, right, that we focus on fostering belonging, promoting retention, and expanding mentorship and resources. So specifically Black marine scientists in the Monterey Bay area, because we sort of had this experience as starting our PhD, talking to other high school students, that there was really this disconnection from the coastlines here that felt sort of unique. I mean, I'm from North Carolina on the East Coast. And there are plenty of communities where I'm from that are close to the shore but haven't interacted. But I think it just felt so jarring in California, given the proximity and like how much of like outdoors and like wilderness activity and nature that there is. And so we also talk about the disproportionate burden that low-income communities and communities of color face in regards to climate change and climate impacts. And so when we think about climate justice and conservation, we have to acknowledge that these will always be incomplete if we're not having an honest conversation about those impacted and ensuring that, you know, they are present to say that, to say that narrative. And so we take advantage of the Hopkins Marine Station, which is a small little outcrop by the aquarium, but a powerhouse to create opportunities for professional development, outreach, and recruitment, empowering a next generation of Black marine scholars. And so this picture is from our annual event last year. We do a summit at the Marine Station in partnership with the aquarium, where we invite about 85 or so undergraduates, master students, people who are interested in getting to know more about the ocean, climate science, to Monterey for a day at the aquarium and a day at the station. And we talk about careers in science, demystifying pathways to marine science, so grad school, professional careers and opportunities, but also belonging to the ocean, right? And ocean interactions. So, you know, going tide pooling together or learning how to surf or learning how to swim. And so all these things sort of tie into these themes of belonging, retention, and mentorship. And so that's really where we are sitting as an organization. And so from this, we'll talk a little bit about our programming. So we have a diverse set of people. Most of us are at Stanford. That's how we sort of got started. But we've really sort of spread on out from there. So we've got Dr. Dawson, who was recently hired as a professor at San Jose State. We have career researchers from the Monterey Bay Aquarium who are partners with us. And so we are called BOSS, like the Black Ocean Stanford Scholars, but we're not tied specifically to Stanford. And so the bulk of our membership is there currently, and we use like the Marine Station to host our event. But when we promote our events to, you know, other groups and other organizations in the Bay Area, we often emphasize the fact that, you know, you don't have to be at Stanford, obviously, to engage with our events. And we study a bunch of different things. So we're all PhD students in, you know, marine science or biology or earth and planetary science or social systems. And so it's really multidisciplinary. But we recognize that regardless of what discipline we're studying, we still find these conversations important to have and also sort of lacking in the field. And so we really sort of came together and said, we have a lot of energy here. Maybe we can at least put on this event and see what goes from there. And so we tend to say when we work in sort of like these three broad themed areas. So education, mentorship, and nature. So with education, we do both scientific education, so like a poster session. This is an example of one of our mentees at a scientific poster session from the event last year, but we also put together a historical exhibit of artifacts around Black American history and the ocean, as well as African history and the ocean. And we displayed that in an exhibit that was walked through at the event, but also two, in sort of a presentation style format at future events. And then maybe Sienna, you're in that picture.
Also interested is in mentorship, and Ryan sort of touched on this already, is this idea that there's a lack of equity in just ocean access and ocean recreation, but this is also especially glaring in ocean research. So when I got to Hopkins, that was like 2021, fall of 2021, I was the second Black grad student to ever get into Hopkins. And for reference, Hopkins Marine Station has existed as a part of Stanford since the 1800s. So there's this glaring disparity. And so it's not just about making sure that our Black communities are interacting with the ecosystem and sort of aware of conservation issues, but also making sure that we're able to be, you know, in the rooms where decisions about conservation are made, or in the rooms where decisions about accessibility to coastlines are made. And a lot of that is into, that's our advice. A lot of that sort of comes about when we sort of eliminate these pathways into academic careers or careers when it comes to ocean policy and conservation. And so what mentorship looks like is we have this program where BOSS members are sort of matched with interested students, undergrads from universities around the area. So I have some mentees from UC Santa Cruz, which is actually where I did my undergrad. So anyone who's been at SLM, Hello, hi. Love to see you guys here. We also have some mentees from CSUMB.
Yep, I was gonna say my mentees from CSUMB.
And he's actually here
The
today.
Strategy Department, exactly.
Out in the hallway somewhere. We have some mentees from Stanford main campus as well. And a lot of it is just giving them an idea of like, okay, what does ocean research look like? What are some of the careers that exist? How do you get from undergrad to grad school or to a master's or to a professor at San Jose State University, right? A lot of these pathways aren't super obvious. And if you don't know someone who's done it before, it can be really hard to figure out as you go along. And there's a lot of extra work associated with that. Like doing a PhD is hard enough without having to learn how you apply for a postdoc or for a faculty position, right? So making sure we're sort of giving them an idea of what these careers look like, whether it's something they might want to do, even if it's not something they want to do. Exposure is really important. So the mentorship program is really geared towards that. And this last bit here, I know that's Ryan pictured up there, and so I'll toss it up to him because he's our resident tide pool extraordinaire. But this is sort of picturing a Black undergraduate group at Stanford called Nature by Nurture, and they're really interested in getting Black undergrads together in community and out in nature and using nature as a form of wellness, as a form of healing. It's therapeutic in that way, and I'm sure a lot of people in this room can sort of vouch to that. But a lot of times Black people aren't super confronted with the opportunity to sort of access those spaces. Sometimes there are cultural barriers to accessing those spaces. And so they're really deliberate about that. And I'll let Ryan talk about this event a little bit more because it was really fun. It was the first time they were kind of doing submarine stuff.
Yeah.
So Mr. Tide Pools.
Yeah, exactly. So part of the piece we talked about earlier was this idea of belonging, right? And I mentioned this sort of story, right, of us going, asking high schoolers, you know, how do you guys engage with the ocean to sort of crickets, right? And they're like, oh, well, we don't, you know, really go to the ocean, or I don't really go to the beach, or tide pooling, I don't really do that. And that made me a little sad because I love the tide pools here and I think that they're so beautiful and cool, and that, you know, every local kid, you know, if you have access to them, I think it's cool to be able to go out and see them. And so that I personally said, well, that's unacceptable. We will have to get you guys on a tide pool with someone who knows, you know. So we took them out down at the station. The Hopkins Marine Station is a biological reserve, so it's some of the best tide pooling on the peninsula. And we took them out to just see what we could find. It was a great time for a lot of for most of the people in the group. It was their first time in a proper coastal habitat in California. And so it really just emphasizes that while we do talk about education and mentorship, fundamentally, one of the things we're trying to do is also connect people to place, and that involves getting out in nature and doing things. So to talk a little bit about a specific thing we did, last year we put on a workshop called the Black Faces in Ocean Spaces workshop in 2025. And it was a 3-day sort of symposium, funded by all the logos you see here, Hopkins, the Monterey Bay Aquarium, and the People's Climate Innovation Center, an NGO group that gave us some money. And it sort of came together to these 3 sort of thematic days, where the first day was on research and mentorship, right? So we did career panels with people from NGOs and academia. We also did student talks. So there's me giving a research talk on my thesis work. And then on the second day, we actually had Dr. Kiki Jenkins, who is a marine biologist from Virginia, who came and walked us through this exercise about creating and commanding our personal narratives. And we thought this was important because a lot of times we sort of passively subsume narratives that we sort of see, but we don't really think a lot about the ways that we're maybe intentionally developing or driving, our own sense of self. And so it was a really powerful workshop that I think a lot of people took a lot out of. And then we paired that with our historical narratives exhibit. And so that's where we worked with the Stanford Library System to bring together a bunch of historical artifacts around Black aquatic histories and the water and display them in the library as sort of a walkthrough exhibit. And I think people really liked that. And then
yeah, the only thing I'll add is that one thing that I think can be a barrier sometimes to diverse participation in these careers is, I know I experienced this with my family. When I said I wanted to, get a PhD in marine biology, they were supportive. They're like, great, that sounds awesome. But also like, how do you make money doing that? Like, how do you support yourself? What does that look like? I had to explain the difference between a PhD and an MD to some people. And it's a conversation that happens a lot, right? And so in this career day, what we're really focusing on is giving people an idea of like, this isn't just something that you're excited about. It doesn't just have to be a hobby. There are ways to support yourself. There are careers, and not only do they exist, but day two is really about belonging, right? You belong in these careers, showing people representation, not just of us as grad students, but of faculty members that we have at Stanford, people who work in ocean policy, people who work with the Marine Mammal Sanctuary, really allowing them to see someone that looks like them in these positions and ask them, what does daily life look like? What does the job entail? How do you get to that position? And so it becomes a little bit more tangible for people. And the last day, of course, we love to ground it in place and in nature. And so Ryan led one of his tide pool excursions, which are always so fun and everyone learns so much. And I think it really connects people to the idea that, like, yes, we've heard about this in theory, we've heard about this from a research perspective, but the ocean is also there for recreation, for us to interact with just as community members. And I think Ryan tide pooling with people really gives them a way to say, okay, there's now this way that I know how to access the ocean on a regular basis and learn from sort of those activities that I'm doing. So we're hoping to do another one of these, and maybe we'll talk a little bit about that later.
Yeah, but sort of pivoting away, we're also sort of like Sienna was saying, researchers. And so we are both PhD students. All of the people in our organization are working marine science researchers, ranging in fields from people in human dimensions to pure like ecology and natural systems. And so we all do feel like all the three of us here do field work.
We're actually all pictured here. That's me in the goggles in the bottom there. And of course, here's Ryan up here, and a couple other members. So that's Kemi in the middle, Stephanie here. Not that they're here with us right now, but Kemi does a lot of paleoecology, which is super cool. And Stephanie is sort of the one that's really interested in these paired human and ecological systems, really thinking about fisheries and fisheries management, which is really cool. But yeah, a wide range of research sort of fields, a wide range of systems, from corals to seagrass to penguins and bivalves, all sorts, physiology to microbiology to laying out transects and counting what you see, right?
Yeah, exactly.
So tons of different methods, a wide range of research, which is really exciting to see all this disciplinary work, interdisciplinary work, and to just get to learn from each other as well. There's so much rich content that we all learn throughout these PhD processes, so it's good to be connected in that way.
Yeah, and then just a little bit more focused on Sienna and myself, who are here as representatives. We both study seagrass ecosystems with our advisor, Theo McKelly. Theo, do you wanna? And so I study those in Elkhorn Slough. I specifically study how the algae communities on the grass affect the composition of the grass and its response to stress. And I do that by scuba diving at Elkhorn Slough. So it often looks like super hazy, low vis. I'm like this close to the ground. But it's really cool work. And then
yeah, so I also work on seagrass in Palau. So it's different from Elkhorn Slough in that it's great viz. A lot of but beyond that, Ryan's really interested in the epiphytic community or the things that live on seagrass. I'm really interested in the fish and the microinvertebrates that live within seagrass beds. So thinking about as the health of seagrass changes, how does that affect the fish and the things like sea cucumbers, giant clams that live in seagrass. And also you can see here, there have been some protected areas. So those red outlines indicate sites around Palau that are protected, meaning that people can't go into those areas for tourism, boating, anything, and they can't fish or groom any animals from those areas. And in some sites, these protected areas have been up as long as 15 years, but as little as 10 at others. And so part of what I'm doing is sort of assessing, have these protected areas been useful? In what ways have they helped the community reach their goals as far as conservation? And in what ways might they be falling short? And so I do that again with transects, visual surveys. Visual surveys are easier when you can see. So I can vouch for that. And also some environmental DNA. So really extracting DNA from seawater and using that to tell you what sorts of species are in the area and creating a community assemblage within species.
And so, yeah, that was a short detour through the work life that we have, but then we wanted to really circle back to underscore this point about future programming as an organization. These are all pictures from our past event, but that picture, that one in the top, in the bottom corner is a new picture. That was from the Climate Dance Party book signing with Dr. Ayanna Elizabeth Johnson, a Black marine biologist who hosted a climate dance party at the Monterey Bay Aquarium and has been going on a tour of aquariums along the coast. We got the opportunity to go to the event and talk to her and invite her out to our annual event this fall. So hopefully she will be keynoting day one of our event this year. And so we're super excited to put on another annual event. It was really well received by the Monterey community, by the aquarium community. And we really got more emails asking us what's next than like, we weren't really prepared for the. Like, we're still sort of
With
like-
this event, like, it was so good to see people come out from Moss Landing, from San Jose State, from CSUB, UC Santa Cruz, Santa Clara. We even had people come from SoCal.
From Southern California, yeah, exactly.
And so I think that really illustrates how important this community is to people, how much people are longing for this level of connection and networking within Black marine spaces. And so we're hoping to, grow even more, expand this year. There's some people we met today who hopefully will become a part of this community and join into our network. And so, yeah, we're sort of riding on the momentum and trying to make sure that we can provide as many opportunities for people to connect and sort of find their way through marine careers and hobbies and recreation as they so please.
Yeah, just a shameless plug, our Instagram is on that QR code, and that's my email, our vice president's email, and then Sienna's email. So if you guys want to contact us or hear more about what we're up to, that's right here, but we will call it there.
You can take a few questions and then we do have the next round of seminar room schedule with the student panel.
Yeah.
When did you first begin creating this collective?
Yeah, so I was the first of the boss group to be at Hopkins. There was a Black student before me, but she graduated my first year in 2022. Nia Walker, she's now what? She's great, look around. But yeah, I was sort of prepared to spend the whole PhD program. I mean, it's like 5 to 6 years. I was sort of prepared to be the only one for the whole time. Because that's just sort of at every marine station what it sort of looks like. And then I remember there was a student who came the year after me, and I was like, okay, there's 2 of us, we should start a Black student union. And we make jokes about it, and we just like hang out. And get and then Ryan's year, there was 2 Black students, and then Devontae came as a postdoc. and I was like,
200% increase.
Class, and then we've sort of been collecting more people throughout different departments as we go along. But it was really great to start that community, and when I started to see Black faces around, I noticed how much happier I was, how excited I got, how much easier it was to just sort of go into lab and get through the day. And so I was like, okay, I think we should keep this together and expand this. I can't be the only one who's felt this way, not just at Stanford, but throughout these other institutions as well. And so that's really what it was born out of, was just being excited to see someone else that's like, oh my God, you're too, like, what's your number, what are you doing here? And then it just kind of grew from there. I think the more we talked about it, the more people resonated with the message and gave us funding to try stuff.
Any other questions? Awesome.
All right.
Thank you. Cool.
I don't know, it's like currently screen sharing. Yeah, we'll stop it.
Just kind of, yeah,
Oh, well.
stop the share.
And I'll-
We'll set up the chairs up here from the panel.
Just like me and the owl. You can still student panel, yeah.
I'm Ann Wynn, by the way. Ann Wynn.
Yeah. They see what happens sometimes.
Well, and when that, like, definitely reach out, like.
I got our email.
Yeah, I did. Yeah, I got it.
Yes.
Is it not that one?
I don't know if it's just my.
Laria, say hi. Say hi. No, you're on the owls.
Is it ever? It is, or is it?
Yeah, it's just a can portion.
That's how. Yeah, I don't know how it's.
If you put a black image on it.
All right,
Hello again.
let me try one more thing.
Yeah, it's like right now, right now. That's what this.
All right, I'll just go ahead and get started with the,
Status.
are we missing someone? Carrie?
I'll just do it.
Okay.
Sarah.
Go ahead. I guess I could.
I don't know what.
It's really guys later.
Yeah, sorry. So we'll go ahead and get started with the student panel. There are 5 panelists and we'll just kind of talk about our background a little bit, each of our experiences here, our projects, our theses, and then it'll just kind of be like an open floor for any questions that you guys have, whether it's project, whether it's journey, whether it's future plans. And that way we can kind of inform you guys of our experience as Moss students. Yeah. Cool. Does that work?
Thank you.
Yeah. Kayla?
Sweet. Yeah, my name's Kayla. I'm a second year graduate student here at Moss Landing Marine Labs. I'm in the invertebrate and ichthyology lab. I guess maybe we'll just do introductions, short introductions first.
Tell me you study.
Yes, I study purple sea urchins, and looking at climate stressors affecting their reproduction and physiology. I do some molecular work, some mesophones studies, some diving, kind of spanning a bunch of different things.
Hi, everyone. I'm Ilaria Bardini. I'm in the invertebrate ecology lab here at MLML. I'm from Massachusetts originally, so yeah. My current research is on animal communities around hydrothermal vents, so specifically looking at the dynamics and community structure of those animal communities and how they're influenced by hydrothermal vent fluid that's being expelled in those areas.
My name is Leah Corbo. I'm a second year here at Moss Landing. I'm in the vertebrate ecology lab, and my thesis is focused on seasonal variation in sea otter abundance, distribution, and raft size in both Monterey Bay and Elkhorn Slough. I'll be doing most of my observations with a drone.
Hi everyone, I'm Javier Mata. I'm a first year graduate student at Moss. And I'm in South Jorgensen's lab at Cal State Monterey Bay, but I'm also part of the ichthyology lab here. My project focuses on competition of 2 different shark species in the Galapagos and kind of understanding how they use nursery habitat with implications for management.
Hi, I'm Carrie. I am also enrolled at CSUMB in Dr. James Lindholm's image and analysis lab, and I'm also enrolled in the ichthyology lab here at Moss Landing. My primary research is in a naval exclusion zone and seeing how it acts as a de facto marine protected area, and it is providing benefits to the fish communities in the area. So we can get started. Anybody have any questions? Or we'll just go through maybe our journeys and our experience in ground school. Oh, we got my word. Oh, true. That's pretty simple. Oh yeah, no, it's that guys.
Oh, that's way better.
Journeys. Well, I guess, does anyone have any questions to start that we can discuss?
How did you decide what you're gonna focus on? Was it the professor saying, this is what she wants? Or was it something you were in love with, or something you hated, or like?
We get the mellow line.
Yeah, all right, sounds good. So when I applied to Moss Landing Marine Labs, I applied to both master's and graduate programs. I knew that I wanted to study marine science, and I knew I was passionate about physiology. Wasn't sure exactly what form that would take. And so I was open to a lot of different things. So my process looked like a lot of cold emails to professors and their labs to see who might take me anywhere. I had originally actually applied to work with Cheryl Logan at CSUMB to do coral research. That position didn't ended up being filled by someone else, and I reached out to Amanda Kahn here, who was doing deep sea sponge reefs, which I thought was really interesting. And she was like, well, you know, there might be some potential for some physiology research, but But we're really looking at diversity. We have already collected this data. And I was like, you know what? I know I want to study marine science. I want to get my graduate degree. So I'll take the position. So I started out here, my first semester, starting to work through the data, getting to know sponge reefs. They are really fascinating and cool, but I was feeling sort of disconnected from my project. And then I ended up talking to a postdoc here who was studying sea urchins and sea urchin physiology. And I was like, oh my God, this is what I wanted to study. And all of the professors here are really supportive of pivoting projects if that's what you're more passionate about. They want you to feel connected to your project and they want to be able to help you facilitate that in whatever way they can. Understanding that a lot of students are not funded. And so if there wasn't money to pay me for the original project anyway, it's hard to keep me on one project and convince me to stay. So she was supportive in helping me switch, and I ended up focusing more on physiology, and that's sort of how I ended up into purple sea urchin. So it was a combination of some things I was already interested in just starting and talking to people to find opportunities and making the best of whatever situation I was in.
So my background was in like copepod research, looking at lipid content of Calamassum arcticus in the Gulf of Maine, and then also in like the use of eDNA for the detection of beluga whale population. So I had kind of a very varied background, but I knew that I really wanted to study invertebrate species, especially in extreme environments. And so when I applied to Moss Landing, I applied to Amanda Kahn's lab, the invertebrate ecology lab, just because that's what I was interested in. And so luckily for me, another science group decided they weren't going to go on this research cruise to the Juan de Fuca Ridge off the coast of Washington and Oregon. And so then there was space available for some other scientists to be able to go on the research vessel. And so my advisor applied for a last minute funding grant for looking at hydrothermal vents along the Juan de Fuca Ridge and the kinds of environments that live there. And so she offered that up as a project that I could come up on. And I was like, I would really love some at sea time. I'd really love to spend some time in an RV, and that was what I got to do.
So for the vertebrate ecology lab, I found out about Moss Landing actually when I was in high school. So this has been a goal of mine since I was way younger. I did my undergrad at UC Santa Cruz, and that's where I got interested in marine mammals. Came to Moss Landing under the Ida McDonald, she's the advisor for the Vert Lab. She kind of had two projects in the works when I started. There was a harbor seal project and an emperor penguin project, and she had two incoming students that year, so one of us was going to be on either. We ran into some funding, or no, not some funding, some permit issues with the harbor seal permits, and it was going to take too long to get them for my master's timeline. So we swapped projects and ran into some permitting issues with otters as well, but persevered through, we're over that hurdle. But she had kind of told me that there was no real backup yet, so if I had any ideas, I could come to her with the ideas. And I had gotten my start in marine mammals and otters, and so I had some contacts, and this project was developed with multiple different collaborators kind of talking things through. So I got very lucky that my project switched to something I was so passionate about, but.
Yeah, so I did my undergrad at Cal State Long Beach, and I worked with Chris Lowe, who's very big into white shark research down there, looking at nurseries and stuff like that. And similar to Aaliyah, I kind of started in the field that I wanted to go into, and I just kind of stayed in it and kind of created those connections that followed those people around. So Chris and Sel had actually worked together on previous projects, and I was just reading through papers and I kept seeing the same name over and over and over again. And so I emailed him and I came to visit, and my original project was going to be on white sharks. Salmon actually, and then similar to Aaliyah, some permitting issues with the Fisheries Science Center that's up in Oregon. And then kind of pitched like, hey, I have this project in the Galapagos, like that kind of stuff, do you want it? And I'm like, darn, like I guess I'll take it. And so that's kind of how that came to be. He had this huge data set that he had with another collaborator down at the University of San Francisco in Quito. And he pitched that to me, and they needed someone that spoke Spanish, but also someone that had experience with acoustic telemetry. And fortunately for me, I had experience with both of those, and I just kind of inherited that project basically.
My story is actually most similar to Kayla's, although in a weird way. I am from Maryland, so I'm an East Coaster, and then I went to school in the University of Rhode Island. And there I was doing work with a shark professor actually. And I was doing all of this volunteer work and helping out in his lab and doing data. And then when I graduated, I was like, I don't really know what I want to do. I don't know what I want to go into. So I took 2 years off. And I just did a bunch of random internships, wherever I could find them. So I worked in Nantucket, Massachusetts, on an island, like in an aquarium, completely different than anything I really desired to do, but it was a lot of fun and I'm really glad I did it. And then I worked in Florida as a sea turtle research intern. And I was kind of like doing this random stuff and I thought to myself, I really don't like turtles. And I didn't really. I was just like, I don't really know what I want to do, but and then I realized after a little self-reflection that I kept gravitating towards fish stuff. Even when I was working in Florida doing sea turtle research, I was always out on the fishing boat. I was always helping the shark professor down there. I was always helping my friend who was the shark intern, and I was doing all of this fishing stuff. I was looking at these fishing projects, and I was like, I think I like fish. So then I started cold emailing people. And I knew that I kind of wanted to move to the West Coast. I really hate the cold, so I needed to get out of Rhode Island. And even, this is controversial, but I even think Maryland is too cold. So I was emailing everybody and I was like, I'm just really interested in like these projects, and I also am passionate about the intersection between management and science. I think that policy needs scientists in order to really advance it. So I wanted to do a policy adjacent project, but in the sciences. So I was emailing all these professors and then I spoke with James. And he was like, I have a project down in Southern California. It's going to be you're going to be looking at the fish community structure in a naval exclusion zone. So nobody is allowed to go there, basically. It's only the Navy. Prohibits all fishing efforts. And then he was like, and you're gonna look at the rest of the island and see how that's acting and see if there's a difference between the two. And that was the perfect project for me. Combined my love for policy, my love for management, I got paid and I get to work with fish. It was all of the things I enjoyed. And I am building these naval research connections because we're in collaboration with the Navy, which is really exciting as well. So I'm actually writing a report and I'm going to present it to the Navy and hopefully they're going to use that as a way to fund the future research in that field as well. So I love my project and I'm very happy where I ended up and I still don't really like turtles. Controversial. All right. Anyone?
What's something that you know now that you wish you knew when you were either first starting your, graduate journey, or maybe about Moss or CSUMB or, anything in that realm?
Yeah, that's a good question.
Yeah.
Yeah, why, you gonna make us go down the line again?
Yeah.
I guess I could start the solution. Yeah, I wish I knew how small the community was, especially within any subfield of biology. Everybody knows everybody, and I wish I knew that if I wasn't so scared to just ask someone like, hey, I'm interested in this, do you have anything that I can help with? I wish I would have known that they would have more likely than not been willing to help me out, because there are kind of professors and researchers and postdocs and assistants and field techs that they need the help, but science as a whole is extremely understaffed right now. So I wish I would have known how willing people were to help you out if you would just ask. And I think that's a big thing. Just that, worst you can say is no. Yeah. And I found that to be very true so far, so, yeah.
I think mine's off for Bobby's a little bit, the community is very small, I knew it was collaborative, but I didn't know how collaborative. Everybody that we talked to in this field is most likely more than willing to lend a hand when needed, or I've had people from the Monterey Bay Aquarium offer up data that they have, or people who are also monitoring in certain areas want to combine projects, so it's a lot easier on both of us, so it's very, while it's a small community, it's incredibly collaborative at the same time. So there's always somebody who needs help and there's always somebody willing to help. And I think coming to Moss where all the students are working on their own projects, but yet still helping out on others. I got my dive certification, not for my own project, for
everybody else's, I wanted to kind of help with that or things like that, so I very much enjoy and love how much collaboration happens in Marine Science too.
Unfortunately for me, I don't get to personally go to the hydrothermal events every week, so. I have like, I don't know, like 2 weeks worth of like ROV video footage, that's just like piles up. And I extract all the still images from that video footage, and so you can just 8,000 photos. So I think like I knew that was gonna be the case. I knew it was gonna be a lot of computer work. I don't think I just like mentally prepared myself for how long I would be sitting in front of a computer for, like pointing at like brittle stars and being like, oh, 1, oh, 2. I mean, what's cool for me is like, there's also, I wish I had known like that, like the breadth of like knowledge when it comes to deep sea organisms is actually like it's really small. Like, so most of the organisms that I'm seeing are actually unknown to science, which makes it impossible for me to identify them and like thus be able to like give you any like idea of what's really living down there. So there's lots of difficulties that come with that, but yeah.
Man, I had all this time to think. I don't know, I think maybe I'll just talk and I'll find my way. But I think that there are a lot of things I did not know coming into a graduate program. I think everyone tells you a graduate program is a lot of work and is going to be hard. And I was like, but for me, it'll be fine, right? And then you get here and I would say that the skills that I've learned the most are soft skills, like being resourceful, asking for help, being your own biggest advocate, like not being afraid to say no, and like standing your ground as like, we're still young, we're still newer scientists, and yet like our opinions matter. And so, but saying no to a professor or saying like, I've done the research now, like I am the expert at what I'm studying, and I might not be the expert, but like I have knowledge and potentially more knowledge than my professor whose breadth is like far bigger than my own. But you focus in on one area. And so you can stand your ground and about certain things. And it's really hard to kind of navigate and figure out how to do that. So I think it was a little bit hard for me to begin with in graduate school because so everything is very overwhelming. And so I think each semester has had a different challenge. But I think leaning on your community and your peers and just trying to foster community outside of the lab has been really good for me and helpful this year. And I almost wish we did more of it in our first year. So maybe that's something that I would tell my younger self, is that fostering some of these connections outside of the lab and potentially in the community that might be marine scientists, but not at Moss Landing, or other aspects of your person are valuable as well. So just some thoughts, I guess.
I genuinely love it. Coding. Nobody told me about coding. Oh my gosh. Not a single person mentioned I need to know how to do that. So that was a new one for me, but we're getting. That's a good one. Yeah. I didn't really realize how intensive I would have to understand how to code for my field was. Like, obviously I knew it's a science. At the end of the day, I'm doing science, but most of my entire project after I did my field research, I have been locked in my little office in the dark usually, and I watched fish videos, same kind of similar to Ilaria, analyzed thousands of photos, videos of fish. And then all I do is I sit there and I code it. And I'm trying to create graphs, and I'm trying to like show how the science works and like show you how I got to this process. And I didn't realize, like, not that I didn't realize it, but it's a lot more intensive than I thought it was gonna be. But I actually enjoy coding. It's just sometimes I need to touch grass.
My graphs are there. They're not pretty, but they're there. Yeah. And to add on to that, like the whole AI thing is becoming even more prevalent in our fields for sure. Like, I'm creating like machine learning models to identify some of the more common like animals and megafauna that I'm seeing in my video footage, and that just helps me when I'm trying to like sort through like all my video footage that has like 1,000 bird stars or like 100 brittle stars in one image, and I don't really have the time, I know what that is, so I don't really need to spend the time to identify it, and so it allows me to give the space and time to see the animals that are less common, so I can spend more time identifying those versus, you know, clicking a dot on every single brittle star.
That's so true. You have to be, I almost think that a lot of people think marine science or any kind of ecological field is kind of like a soft science or like not a real science is like something that I've been told before. And it's like, no, we actually have to know so much background science in order to even understand our research. Like we have to teach our coding softwares how to like machine learning to create all of these programs. You're basically a computer like programmer, you're like a data like scientist. You have to understand basic physics, chemistry, geology, oceanography. It's this giant intersection of all of these things. And sometimes you're just like, oh my God. I just wanted to look at fish all day. And that's the dream, you know? But
Right, like I'm an ecologist, but in order to understand like how the animals are being influenced by this unique chemical fluid that's coming out of these vents, you have to understand the water rock interaction. So the geology of the area and how that influences the kinds of fluid that's coming out of the vents and like the ratios of like the heavy metals in it. It's like you have to know everything. You have to have a good grasp on a lot of aspects of the area. So you just have a huge intersection of a bunch of different kinds of knowledge.
yeah, that's.
Ocean science is really interdisciplinary, and you find that in ecology, a lot of people bridge their ecology with a hard science, right? Like molecular work or chemistry or geology or something like that.
Physics.
Physics.
I do a lot of hydrodynamics and tag design and stuff like that, so that's kind of the hard science that is referring to. The geology part, my field site is made out of volcanic rock, so, you know, magma has chemicals in and of itself that affect water chemistry, so just kind of re-emphasizing the point of interdisciplinary work within any subfield.
Yeah, that's so true. I am taking a class at CSUMB with Hobby's professor. And we get to class and he told me, he was like, oh, you're not gonna need any background knowledge at all for this course. And I was like, great, I can't wait to start. We get there on the first day and he's like, do you have basic engineering knowledge? And I was like, no. Basic engineering knowledge. And he was like, well, we're just gonna do some very simple 3D rendering of this tag design. It's gonna go on a real life shark, and then you're gonna build it in this coding software called CAD, where we will then print it with a 3D printer. And I was like, you told me I needed nothing to take this class. But actually it's going great. Yeah, and the tags that we designed, were designed through Hobby and many other students in the class, and also his lab in particular. His lab is very intensive. They actually put these tags on the sharks when he went down to the Gulf Coast. And then we also go in the field and we tag leopard sharks for them. So it's like all of these things, it was really cool to see like all of them actually interacting and working. And I was like, oh, this like actually matters.
You have any other questions?
Yeah. Silly question? Yeah.
So you mentioned a lot of collaboration. Do you guys have a lot of collaboration like between yourselves as well? So I know you're all studying something different, but do you guys work together on certain things, or do you reach out for questions between each other, I guess?
Yeah, I think I'm gonna speak for a few of us here. So Aaliyah is using drones. You have to be a certified drone pilot through the federal government. All drone pilots have FAA licenses as drone pilots. And so I'm on her flight plan, she's on my flight plan. We're both divers. Kayla's a diver. Kayla and I can dive for a different student's project. And so that's kind of where it gets kind of tricky between institutions. But that's really, I think we see the collaboration of students helping out with others' field work. Some of the analysis, like I've done some bruv stuff that uses video analysis. So if they need to get some code for that, be like, here, yeah, take the code. I want credit for it, but if you can use it, absolutely, it helps put their paper out, it helps get my name out, it builds their skills, it builds my skills. And so I think a lot of the collaboration, at least for I'll speak to megafauna specifically, really come to the field. It is incredibly labor intensive. Everybody sees, then with sharks, it's like, you don't see the 95% of the time you're on your hands and knees scraping mussels and barnacles. You know, you're pinning boats upside down, you're in full hazmat because of toxic fumes with anti-fouling paint. It's incredibly labor intensive, laying chain, tying knots, all that stuff is, you know, I would say the bulk of marine science and being in the field that most people don't really get to see. And that's what facilitates the collaboration to eventually get to policy or publish papers and things like that.
Unfortunate, it sounds bad, but your best free labor are your friends. And honestly, like we want to support one another. We want to see each other succeed, and we know like firsthand how intensive this work is. And so we want to be able to make sure each other are supported, because it's not like we have, like, this is our team. We don't have another team of people just designated and paid to help. So like I had to dissect urchins, and half these people definitely didn't want to get their hands dirty in an urchin, but they helped me anyway. And like, I would help them for whatever things they need. And it also like helps us with our skills. Like you become a better scientist by being like knowledgeable in different subject areas besides your own. And you want people that can bring different perspectives. And so I think I've said it once and say it again, like the things that Moss Landing does well, it does really well. And there's things that are challenging about Moss Landing, and those are really challenging, but the things that it does really well, partially is collaboration. Like we want to help each other, we want to support each other.
And that applies to professors too. Like the
Yeah.
professors here are very good about bringing in students and, hey, you know, one that's off the top of my head, Tom and Mike, the physical oceanographer and computational oceanographer, they're always together, always sharing code, you know, talking about the coding skills, reproducibility of the code, and allowing students to use each other's advisors' codes and stuff like that. So the collaboration is through across the hierarchy here at the Moss.
Yeah, personally speaking, my scans, I'll be doing 2 scans a week for an entire year, which doesn't leave me a lot of time to do much else. So like Bobby said, he's on my flight plan as a backup pilot. So on weeks where I can't go out or I need to go see family or whatever else, he's gonna be taking over my statement, my scans, my research for that scan. And it just makes it easier knowing that they're certified, that they've got the skills needed and that they're willing to help. I need about 3 to 4 people per scan normally, so it's a lot of people to just wrangle up all at once. So thankfully, I've got a lot of volunteers that have already jumped at the chance to help out, which is very, very nice. So takes the pressure off of finding people last minute, but.
Yeah, I think the greatest strength of our lab in particular is the fact that we are all so interconnected with one another and close. And it's just because of how it's set up. Like, we're all in the same building. So we interact in all of the same classes every single day. Like, you guys walk the hallways. Like, we can just pop into whatever lab we want. Like, oh, our friends in fight, let's all go to the fight lab. Like our friends I used to sit in Aaliyah's lab in the vert room all the time, just because I wanted to get away. Yeah. But stuff like that, it's just like, that is one of the strengths of this facility, and I know other people in different graduate programs who just don't have that same level of community. And they still have a lot of collaboration and a lot of like, obviously respect and like cross institutional like relationships, but it doesn't have like our strong community. Like, we have a great community, like this entire event is community run, like all student run. Like, we're the ones doing it, and it's all of us together, like pulling our weight.
Here's the open house coordinator. She made all the. One more point to that though, to bring potentially the other side of the coin. Part of the reason why we need to collaborate with what you want to know so much is there's no money in this. And so we're really scrappy here. In our warehouse is full of rope and
they can make anything.
And cages and glass and this is a more positive one. Our facilities people, fantastic, wonderful people. They're not professors, but they are so helpful. Like, yeah, I can make you that. We just gotta take these PVC pipes and then this plumbing and they're like, cool. But we do everything ourselves. Like, we don't have the money to buy things new. So we consistently revamp things, and it's good that we're not like throwing things out, but it does make things more challenging sometimes because things are broken all the time. We don't have endless manpower and we don't have endless money to send out samples for things. So we do a lot of things in-house, which means we have to support one another because otherwise we're not going to get our projects done at all. So it's part of it is a necessity too.
And then sometimes if you're helping with too many projects, yours start to get delayed. So it's also important to stay on top of your own thesis research.
Yes,
If you're waiting on her.
yeah.
Questions?
I apologize if this was asked earlier because we missed the beginning, but if you could share a little bit about how you came to marine science, as your path.
Yeah. We can go down the line again. Yeah, so
You did it earlier.
it's okay, we'll do a quick recap. Yeah, I'm Kayla, I'm interested in physiology. I was originally an environmental geoscience major in my undergrad, took some years off, did some internships, cold emailed a bunch of people, knew I wanted to do a graduate program, but I didn't know exactly what I wanted to do. Once I came here, I wasn't super sold on my project, so I actually pivoted projects halfway through. And that's sort of how I landed with my project now, which is looking at purple sea urchins. But I think mine is slightly squiggly, but pretty linear path. I studied science in undergrad and I proceeded to study science in graduate school, but I focus mainly on more geoscience-y related things like sediments and things like that. So it was kind of a different pivot to more organismal biology. Me.
I had a varied background, so I studied copepod lipid content, like their fat content for a little bit, and then I moved to Alaska and studied the use of eDNA in beluga whale populations and how we can use it to detect them. And so during that time, I emailed Amanda Kahn because I was really interested in vertebrate species and also extreme environments and how those animals survive in extreme environments and basically what community function. And she had a project that happened. She had applied for like last minute funding for a project that was going out to sea. So we got to go out to sea for a couple of weeks. And I was like, absolutely, I would love to study hydrothermal vents, which was what the project was for. And so I'm a deep sea ecologist right now.
Mine was a very linear straight shot path. I grew up with, my dad loved the ocean, big swimmer, got me invested in the ocean at a young age. I loved sea turtles was my first love, sorry, Derek. But there wasn't a ton of sea otter, sorry, that's what I'm doing now. Sea turtle research, in California. I grew up in Long Beach, volunteered at the aquarium, did my undergrad at UC Santa Cruz, took a mammals class, fell in love with it. Got involved with the Sea Otter nonprofit, found out about moss in high school and knew if I was gonna do my master's, I wanted to do it here and kind of full circle moment, got here on a harbor seal project, had permitting issues, switched to sea otters and now stuck with it, stayed here, so.
I did my undergrad at Cal State Long Beach, officially my degree title is marine biology. I knew I wanted to specialize in something. I asked my advisor for some of his contacts and some of the people that he knew, and he connected me with my now advisor. I reached out to him, I was gonna do some white shark and salmon stuff up here. Ended up pivoting partially due to permitting, but definitely there's someone else to take this other project. So I looked at competition in the Galapagos between two different juvenile shark species and a couple of the super quick.
I went to school in Rhode Island. I was doing shark research there for a little bit. Then I graduated and kind of didn't really know what I wanted to do, so I started doing a bunch of different internships. Lived on an island, then I moved down to Florida, did sea turtle research, hated it. Pivoted back to fish, and then when I was applying to masters, I was like, I want to get out of the East Coast, so I started applying to only West Coast schools. Got into Moss with my advisor, Dr. James Lindholm. He handed me basically a fully functioning project of going down to Southern California, thank you. Southern California, there's an island there owned by the Navy, and they have like a zone that nobody can enter except for the Navy. And they let us in, I dropped cameras, I watched fish videos, and I'm studying how it acts as a de facto herd.
Yeah, I was curious, after you get your PhDs and you're all done, What do you see yourselves doing in your near future? What are you gonna be doing with that?
The age-old question.
Well, okay.
My mom asked.
So.
I think I'm just gonna stay grad student forever.
Seal, masters. So we technically all get masters.
Oh,
degrees.
masters, I'm sorry.
No, but it feels like a PhD.
It
to be honest
sounds like
with
a
you.
PhD.
We call it a PhD light, because most master's programs are designed to be 2 years. Ours is designed to be 3 years.
And I
have it.
We're actually 4.5 to 5
Yeah.
years.
Right.
That's average, so,
The flip side though is like, the people do turn out really robust projects here.
Yeah.
They're
PhD level.
PhD level projects. It's, yeah, I mean, none of the credit, but all the work.
Ideally, I'd like to stay in the area. I also I would go travel to wherever the jobs are. I would ultimately love to end up in sea otter rehabilitation and research, at the Monterey Bay Aquarium or in the nonprofits in the area, but I think Moss is stuck with a skill set that's very well developed for diving projects or drone projects. There's opportunity, they're doing whale projects with drones right now too, so it was piloting them. I've also done some aerial surveys for planes for a company in Monterey, so there's opportunities. The one thing I will say about grad school that I also didn't know before I started was the networking opportunities here are incredible. I've just being in grad school, it was the first time where instead of me having to seek out opportunities all the time, it was the first time where I also found opportunities finding me without having to scrounge around and fight tooth and nail for it. So it's very nice to have people go, oh, this is your skill set, great, I think I have something you might get used to. So it's very, very nice time.
Unlikely, I don't believe. And it is nice here, it's beautiful, and I'm originally from Southern California, I like the Southern California kind of pace of life, there's something slow here for me. It's a little too cold for me up here too.
You say slow?
Slow.
Slower than
Pace of life.
Long Beach.
Really?
That's why I thought beach.
But anyway, my goal is to eventually go work for NOAA, they have a lot of, not right now, but usually, they have a lot of positions open where individuals that have a master's degree are able to go and kind of work at the policy science interface. That seems really appealing to me. I did an internship working with Sam actually doing that. I currently have no real desire to do a PhD unless I absolutely need to. But that's kind of the goal is work for the federal government, kind of legislation, science adjacent, fieldwork analysis, kind of combine everything and synthesize it into one career path.
I also wanna leave. Like I said, moved from Rhode Island and I was like, God, I really get cold. And then I moved here and I had never ever visited before. So my first time here was when I was looking for apartments and I was like, it's kind of cold. So found out that it is in fact too cold for me. So my plan is to hopefully either move south to San Diego or move away. I'm currently looking at PhD programs in Hawaii and also Australia and New Zealand. So trying to go somewhere warm and sunny. And like I said, I'm really interested in a PhD. I have always known that I wanted to do a PhD. I just like really want to be a doctor. I think it'd be awesome. And kind of savvy, but like my dad gave up pursuing a PhD so he could raise me. So I would be Dr. Johnson first. But I'm just like similar to Javi, I'm really interested in the policy science interface, but I really want to be up in a managerial level. So I think I'm going to need a PhD in order to really advance to the high levels that I want to be working in the management, and I want to be the one designing the management plans. I don't want to be the one implementing them. I want to be the like final say, stamp of approval, this plan is going through. So I think I'm gonna do it. Yeah. Sorry.
And on a more serious note from my previous answer, I am interested in staying in the deep sea hydrothermal vent fields. there's not too many of us. So there's a pretty like straight shot to a PhD from there, which is nice. But honestly, I've been really fiddling around with the idea of becoming an ROV pilot. So any other questions?
Yeah, I don't know.
Did you already go?
No.
Oh, sorry.
No. I don't have any like super special unique answers, but but I think that I would be open to pursuing a PhD. I really do like research. I like science a lot and I like what I can learn from it. But because we wear so many hats as scientists, I think a position where I can do a lot of different things would suit me best. Luke Gardner here has a Sea Grant liaison position and he has a PhD and he basically helps to bring together the right groups of people to tackle a specific problem. So he's someone who facilitates a lot of that communication. He helps with outreach and he works on management plans. He works with policy stuff and he also takes graduate students now and again, but he's not a professor. So he doesn't have, and he has his own funding from Sea Grant and is paid that way. And so I think some sort of position where I can do a lot of different things and work in a communication research aspect, do a little bit of everything, I think would suit me best. And I do think a PhD will probably be the best for that. I'm open to going to a lot of different areas. I've also looked abroad for different programs. I'm someone who is really big on traveling. I like learning about different cultures and experiencing the way they do science there. And so I think it would be cool to learn from a different place. I feel like I've jumped around to a bunch of different places and I've learned something new at each one. And so a lot of people tend to stay in one place, build a network and just continue here. And so I think it would be valuable for me to leave, and even if at one point I come back. So well, all that's to say, I don't know. But we're gonna see how it goes and graduate first. That's the goal. Other questions?
I know Santa, but.
Yeah,
Whatever.
but we're here all day. So,
If you want to find one of us for an extra question, you
yeah.
probably will be able to, right?
Yeah. What's your guys' favorite sea creature real quick?
Yeah.
Yeah, sure.
Turtle.
My mortal enemies.
I'm biased, obviously, but I'll say the Mako shark. I really like high performance things, jet, land, motorcycle, and a Mako shark and a tuna are the high performance fish. Really, really cool.
I'm gonna have to say turtles, but also otters. Both of them.
Shocking. Horseshoe crabs.
Oh,
Crabs.
that's a great one. At all.
I like a good cephalopod. Octopus are cool. Orcas are really sick. Seahorses are cute.
It's hard to like one whale shark. Just a giants. Well, just I'll get answers.
What's the most difficult, I guess thing to get into? I was thinking animal to start researching, but I think in general, like what's the hardest to get?
Quick sidebar, it is 4:08, so if any of you guys need to go, feel free to leave. Well, I'm happy to stay for a bit longer and chat more questions, but I'm just saying, if you need to leave, you're totally good to go.
Can I speak on this one to the side?
Yes.
Yeah, I'm.
Sure.
So there's a lot of stereotypes and there's a lot of kind of stigma. Obviously in my subfield, we deal a lot with public perception, right? And dealing with media and pop culture and social media and news outlets. And going back to this one about what do you wish you would have known? I wish I would have known how much that affects you as an individual, right? And people coming up to you and, did you hear about this and that? And it's like, yeah, we did, but we're scientists, right? Our job is to look at the data, analyze the data, and try to draw conclusions from the data. It's our job to stay removed emotional from people's perspectives, so that you can't manage that, right? But we deal with data. Sharks and maybe Leah can speak to marine mammals are notoriously difficult to get into, and I'll be the first to admit it is extremely gatekeepy. A lot of the kind of higher level advisors that kind of started it, they're kind of on their way out in terms of careers, so most of them are getting ready to retire. But I think generally marine mammals and sharks are the most heavily sought after, just as it's megafauna, right? Due to the pop culture and media, everybody knows what they are. And so that makes it inherently popular, which is great, right? Like, we want the interest, it makes it easy to kind of advocate to you guys, and, you know, going back to the money thing, it's easier for us to go out and say, hey, this is what's important, you know, you see a white shark here, this is why they're important, this is how we deal with them, this is how we study them. But yeah, I think sharks and marine mammals are the two kind of subcategories of species that are super saturated and notoriously difficult to get into, because everybody's kind of like, they want all the credit for all the data and all the outputs that they do.
So you hit a wall with your collaboration then?
Potentially.
Yeah, I think, I would agree with a lot of the points Javi made, with marine mammals, my first experiences were rather. I ran into a lot of walls because there's some gatekeeping. However, I have noticed that the new generations that are coming in seem to want to squash that, which is great. I've been doing my part to try to make sure it's more collaborative, inclusive. I make it easier for the people after me than it was for me. I will say, with being marine mammals, another aspect of hard to get into is permitting. They are super heavily protected, which is a great thing. It just makes it hard for people who want to do research or implement new methods to get past some of those hurdles, because they don't know what the outcome will be. So it's a little nerve-wracking for the government for the protective measures. But thankfully, we figured it out. But yeah, I would say I agree with a lot of what Bobby said. There's very, very, because of charismatic megafauna, they're right there, so that there are a lot of perspectives about things that happen with those species.
And to go off of this, but like, we are getting better in terms of, why are we putting unnecessary barriers, you know, we're working on the same project, yeah, things that we have similar methods. Come all, you know, you get credit for the paper, I get credit for the paper. Like that.
This is a science problem generally too.
Yeah.
This idea of publish or perish and people being very gatekeepy about their data and things like that. Like, we're trying to publish it. Why are we all reinventing the wheel? We don't need to do, because we're reinventing the same thing, you know, like sharing is. I'll also throw in corals and turtles as two other subcategories that are hard to get into. And one aspect that they didn't like talk a ton about is like nepotism, sort of, where like a good example is grandfathering, the coral lab in Hawaii, for instance, like that chance you can get into that lab unless you work there first, or you went to the school as an undergrad and you worked in the lab, and then you can get in as a graduate student. Like, there's a lot of like programs that are sort of like that. And part of this is connections. Like, you're always going to be able to get to better places by making connections. And I hear that, but these particular programs will almost like never accept people unless it's that way. And so it's almost like funneling the type of person that they have in the lab, which I think limits the diversity in perspectives, which I think is a little bit harmful to science.
I definitely agree. Also, like, one of my best friends, she's South African, and she went to university like in South Africa. And she has a master's from the University of Cape Town. And she was telling me that there's like 0 shark projects there. And I was like, it's South Africa, why aren't there any shark projects? And like, obviously she's a shark.
There's a lot of sharks there, if you didn't know.
Lots of white sharks there, yeah. Actually, there are causing these problems, but it's maybe a lot. And yeah. She had a lot of problems when she was like getting into science, because a lot of these programs, like in her own country, they're like, oh, you should pay us to take you and do research. And so
That's a
instead
big
of
thing too.
them paying you, you're paying them for the joy of research. And it's like, that seems kind of backwards, no? And it's a really big problem, especially in like
In
other
sharks and
countries.
corals in other
Yeah.
countries.
Sharks and corals especially, because it's just like, people wanna do it so badly that they're willing to pay the money for it, and they know that. They know that people are willing to pay the money in order to pay out the white shark and do any kind of research on a white shark, even if it's not real research or meaningful research. It's just like the idea that I can pay money and do this thing. So it limits even people from that country being able to do this work. Which is really sad, and she's applying to, since she just graduated from her master's, she's applying to a lot of jobs in America. And abroad, in like Australia, Bahamas, obviously the US, Mexico, all of these places. Cause she's like, yeah, there's nothing here in South Africa, they've monetized all of it, and it's sad.
I almost applied for one of those until I found out it's $3,000 to be
Yeah.
a 3-month field season.
Mm-hmm. I had looked at like coral restoration ones and like, Indonesia or like things of that nature, like there are ones that you can pay for that,
Yeah.
that will pay you, but they're few and far between. For the most part, you like look and there's fees and suddenly like you have to pay a lot of money. Which gets also into like ethics of like, when you're looking at someone's resume and trying to pick someone,
Yeah,
how do you?
I think it's inherently predatory.
Yeah.
Which is sad and a problem in science, but. Yeah, I think it doesn't push us.
It just limits accessibility.
Yeah.
Just.
If this is too much of an invasive question, you don't have to answer. But what GPA did you have when you got into graduate school?
Oh, like my undergraduate GPA?
Yes.
Well,
If that.
Yeah.
my lab, when I emailed my advisor, just for information on the program on her lab, she would not accept anybody with under a 3.4. I had a 3.45.
That's really good.
I had a 3.8 GPA and a 3.7 actually, I think. I don't remember exactly, but I can say that the vast majority of the time, your experience matters well more than your GPA. So you're like, you need to do well in your courses, but I think if it's a good program, they're looking at experience as opposed to just like how you do well on tests in certain respects, and you know, to a certain degree. But everyone's different. So like ask them, the professor is, you can ask them for transparency about it, or if you're worried about it, you can be like, hey, I have experience in some of these things, I excel at some of these things were hard for me, what are you looking for? And how can I like, yeah, what are you looking for?
I unfortunately had a 3.98 and I lived in the library, so. Yeah.
I graduated with a 3.8, but a lot of it, school always came naturally easy to me. That's just the way it was, but to reiterate Kayla's point, the experience is far more important. My advisor told me, you know, I have a GPA, look it up, but I didn't know that, you know, GPA didn't matter. Relatively speaking, and so I put it at the top of my CV, and he just kind of like got a piece of paper and put it over it, and he was like, tell me about your experience, you know, why, his results to me, essentially. So that, yeah, let's go about the experience, and to the point about the capitalization or the monetization of it, true experience. That, you know, you're doing real science, analysis, the quantitative thinking, the critical thinking, the synthesization. The synthesis of information, that's your job as a scientist, to take all this information, synthesize it, and create an output that is relevant to the public, is, I think, what our job is as scientists, and communicate it properly. Communicate it properly. Scientists are terrible communicators. Proper communication for the general public, so that what we do is digestible by the general public.
I will also say, like, one point before Carrie talks on it, to that is like the experience thing. On the one hand, you're like, yes, experience over GPA, because like classes are hard for some people, apparently not hobby. But some of these competitive things like sharks, marine mammals, they're looking for people who have that experience. And so it's really hard to get into these things if you didn't already do it in the past. So that is an unfortunate thing. It's pivoting within this field sometimes can be challenging.
Yeah. Mine, I actually don't remember, it was so long ago now. I think I had a 3.75. I think, and kind of similar, I was a little lazy in school. I knew if I applied myself a little bit harder, I'd get better grades, I just didn't really feel like it. It's the unmedicated ADHD, but. So there. But yeah, definitely experience, I've hopped around so many different things, and I will say though, when I got into Moss Landing, I had one of the like advisors at CSUMB, which is the one that I'm actually held in, tell me that one of the reasons that I was accepted over somebody else was because of my impact. And I was like, cool, I didn't know that. So that's nice, I guess.
Grades are not.
When I had like interviews for grad schools, like, no, they never asked what my GPA was, they didn't really
No.
care.
They cared about who you were.
Yeah.
Like
They cared about my course experience.
your character also really matters. Like another thing is if you make enemies in this field, they will know they don't.
Don't bring
And
it up.
other people will then know you. It is a smaller field than you really think it is. We know way too much about some people. Just from word of mouth of some advisor on the other side of the country. We know things that they've done to other students based off of what other people have told other people. It's a small field.
A really good example, my undergrad advisor. Was this
Say it?
was her undergrad advisor.
Yeah.
And we had no idea until 5 or 6 years later. And I was like, hey, you know, Brad was like, yeah, great, oh, yeah, cool, and they had like dinner at each other's house and drinks and.
Yeah.
You know, we were all partying and stuff in Hawaii and everything in Keys. So it is a.
Small.
However small you think it is, make it small.
Yeah. They say everyone in the world, you're only like, what, 5 people removed from, six people removed from any person in the world. For marine science, it's maybe 2.
Maybe 2, maybe 3.
Like, it's like, I applied to a job the other day, already rejected, it's fine. Par for the course. But in the, one of my references in the email, I did an internship before I started graduate school in St. Croix in the Virgin Islands in the Caribbean. And in his email back to me, rejecting me, he was like, sweet to see Kristen on your as a reference though, I did my dive certification with her a few years back. Totally different, like.
Yeah.
Crazy.
I needed a last minute recommender for grad school because one of mine had backed out 3 days before the letter was due. Checking with your recommenders. Months in advance, weeks in advance, that was gonna happen. I got a last minute recommendation from somebody in my nonprofit, ended up being a lead student of my advisor, so. Carrie and I interviewed the same.
Yeah, Leah and I interviewed for the same exact position in the same exact lab at CSU Long Beach. And he told us word for word verbatim the same exact thing. And so.
And then neither one of us got the position.
Yeah, so.
And the person that did is my close friend, because we did our undergrad together.
He Rhode Island that I was doing research with from conferences. Like, he was like, you know, you follow this person on Instagram, but I know this person. And I was like, how do you know this person? And you
Well, I was just saying, thank you guys so much for highlighting all of your experiences, and I think like the idea that your experience matters so much more than your grades, I think that's such an important thing, and I just was wondering if you have any advice for young people who maybe love science, like how do you go about getting those experiences so that you have those types of experiences, like how do you chase down those opportunities?
I had mentioned it earlier, one of the things that I wish I knew was just ask people. Like if you go into the aquarium, hey, taking anyone? You see someone wearing an aquarium t-shirt, you never know. And just ask, first they can say is no. If they don't know, especially within science, if we don't know the answer, more likely than not, we know someone that will know the answer. And because we are so connected and, collaborative, it takes 2-3 connections and email and that's your foot in the door. And then I have just a personal opinion. If you're willing to pursue it, tell them, and if you're not, tell them also, and that way they're not expecting you, and then, that could kind of be your name a little tainted, like this person didn't follow through, they didn't tell me. So I would say just ask and follow through, and if you're not gonna follow through, totally okay, you realize it's not for you, you're not interested. Life happens, you have to move, like anything can happen. Tell them, hey, I appreciate the opportunity, unfortunately, I can't take it right now, but thank you for your time, and that's it, just move on.
Yeah, another thing is,
Just that.
don't be too picky. Like, you can be picky at some point, but like, it's so valuable to learn what you don't like, and I've heard this before, like, people say that, it's so valuable to learn what you don't like just as much as it is to know, like, what you like. Like, asking people, advocating for yourself. The second I got to Moss Landing, like, I didn't have funding, I asked every single person in this hallway if any of them had a job. And like, I thought my advisor would be giving me money basically from the start for like some of the funding that she had. It takes administration and back end stuff way longer than you would think to get their money together. And I was anxious, so I was just asking, just ask people, and like to Javi's point, and eventually, like within a couple weeks, like I had a job, and it was a job that was not posted, not available. They were like, maybe we're looking for someone, and then they just were like, sure, we'll take you. Like, so just like asking around, and you know what, that job, I will not be doing that job again, you know, like, it's just that, like, you just learn from every experience, and like, when you're young and have time, if you can volunteer, but it is definitely valuable to shoot for those paid positions. It's okay to get rejected, try and apply again, you know, like, try things that seem like they're adjacently related. One of the jobs that my first job post-grad from my undergrad was a freshwater position in Oregon. And I was working with Oregon Fish and Wildlife, nothing to do with marine science. But it was still like adjacent skills, and people look for these environmentally science skills. So just yeah, try different things, ask people.
Sign up for everything and anything.
Yeah, sign up
Get
for-
involved, get involved, get involved.
It's so true too about like just asking. We know a girl who walked.
Oh my
into the
God.
aquarium.
Yeah, sorry. This is
She
crazy.
walked to the aquarium in Hawaii and she
On
woke
now,
up,
walked in.
like walked.
On
in,
vacation.
genuinely on vacation, walks into the aquarium and she goes, hey, are you guys hiring? Has a job in Hawaii. Moved there.
She's in her last year of undergrad, was vacationing in Hawaii her senior year, walked in that aquarium on her vacation, asked them if they had a job. They were like, yeah, we have an opening. You want to start this summer? She said yes. So she's moving to Hawaii post-grad.
Based off of just one conversation with these people. And that's just how it goes, honestly.
Who you know.
It's all
I
about who you know.
got an
Don't be afraid.
internship at the New England Aquarium in Boston, on the giant ocean tank internship, diving in the tank. I don't think a single other person applied that year, actually, and that's why I got it, because it was COVID. So I actually didn't really interact with visitors. I had to like waterboard myself to go underwater. I had to wear a buff coming out of the diving thing, but just like ask people if they're still having positions open, follow up with people. That's a big thing too. Like, you get rejected, email being like, thank you so much for the opportunity. And like, keep me in mind if you have anything available. Like, continuing to follow up, continuing to maintain connections, and being diplomatic about things.
I think most of us, well, actually, I won't say most, I know Aaliyah and I, we like, just like volunteered at aquariums, but that was by us,
Oh yeah, my internship was unpaid.
Yeah, well,
So,
exactly.
like-
Oh, just unpaid, but that's just one of the things you do in your time.
Yeah.
I am from Maryland, I was asking the Baltimore Aquarium all the time, hey, you guys have, you know, just constantly annoying them.
Yeah.
Exactly. And
Yeah,
then I lived
Sweet Green
in-
does get the grease.
When I lived in Rhode Island, I was like asking, I worked at an aquarium there, and it's like, I don't like working in aquariums, really. And I did, because it was experience. And then now, like, I know how to pivot it in a way that's like, yeah, I worked in an aquarium and all I did was clean a fish tank, but actually I had all of these other cool experiences too, so you should still really hire me, because I learned all of these things from doing this one job. So this is also about how you sell yourself.
Yeah, high school, I volunteered at the Aquarium of the Pacific, and I was just an education volunteer, but it also required opening up some of the exhibits. Animal husbandry, making sure that the animals were well taken care of. Public outreach, communication. I was the one on the microphone speaking to groups of people in high school, which is very intimidating when you're 16. Then I took a gap year, went to San Luis Obispo, did the Central Coast Aquarium there, was an aquarist, chopped up squid every morning, but that's animal husbandry. I did system maintenance checks, I did water quality checks, I did main system checks, just it gave me a plethora of skills I could use elsewhere. And then came here, and the Monterey Bay Aquarium has 7, 8 applications on file with my name on it, so eventually they're gonna get tired and say yes, but fingers crossed.
Opportunities do come to us now, but we don't necessarily get them.
Yeah,
Still
I
competitive
just
field.
submitted one 2 days ago. Yeah.
Yeah, I mean, high school for me was also a lot of unpaid animal husbandry internships, was just great. But I think that like for me, I kind of started to let my love of marine science percolate into other aspects of my life, like art or like videography things or like any artistic pursuits I was also doing. Like I figured out what I loved and I went and ran with it in every aspect of my life. And I think that really comes through, especially later on when you're like applying to college or applying to grad schools in the end, because it's like I think it's just so important to be true to yourself and also just if you love something, let it take over your life, I guess, if you want it to.
A counterpoint to those, I don't know what I love, so I just keep trying different things, and that's okay too. Something will stick.
And I always joke, but I tell my students this all the time, I'm a TA at CSUMB, and I tell my students all the time, it's like, it's so much easier to find out what you don't like. And the only way to do it is to try. Like, one of my really good students, was here volunteering, and originally he was like, I really like oceanography, I wanna go into engineering, I wanna go into like some kind of ocean engineering. And I was like, that's great, have you ever done an internship? He said no, and I was like, okay, well, like, you should apply to these ones, and then he did it, he goes, I hate this, this is awful. I could never imagine the rest of my life being like this. And I said, well, at least now you know. And now he's switching gears. He's in the biological oceanography class here, and he's still an undergrad. And he's taking the bio-ocean class, and he was like, and he was at this event in Stanford, and he starts chatting up somebody down there, and they were like, oh, I have an opening coming up for a PhD. You should apply to it. And now he's applying to a PhD program at Stanford. And he's like, I'm totally switched to biological oceanography. I'm totally done with engineering. I hate engineering. I was like, well, at least you found your passion. So it's easier, I think it's a lot easier to find out what you don't like. Like I joke, like the turtles, that really sucked. And I was there the whole summer being like, wow, I'm never doing this again. But it was a great experience and I'm incredibly grateful for it. But yeah, it's so much easier to find out what you don't like, because you will notice right away if you don't like something. More so than when you're like, oh, I'm like kind of passionate about that, like, I kind of like it. Yeah. Cool. Other questions, any other questions? Thank you.
Thank you.
I didn't find you to us off.